The first three speakers were all heavily involved in that discussion, and we are very eager to hear what transpired. We are also eager to hear other things about what does access and benefit sharing mean. What are the negotiations about? What is the development, policy development, and infrastructure development? What does it mean for the smallholder farmers who are custodians of genetic resources? How are they going to start getting the benefit of some of these finances that are coming out and actually enrich themselves after or even as the technology companies and drug development companies are reaping the benefits from their genetic resources. So welcome to this first session where we're going to talk about access and benefit sharing, digital sequence information, biodiversity governance in Africa. And our first speaker is Dr. Priscilla Mumo from the Wildlife Training Institute. Dr. Priscilla Mumo is a scientist based at the WRTI in Naivasha. She has extensive research and experience in wildlife biotechnology, microbial bioprospecting. Her research focuses on the discovery and application of beneficial microorganisms. More importantly, she has played a significant role in the development and implementation of access and benefit sharing agreements and advances in the objectives of key environmental agreements, including Convention of Biological Diversity, the Nagoya Protocol, and CITES, the Convention on International Trade in Endangered Species of Wildlife, Flora, and Fauna. So we are excited to have Dr. Priscilla Mumo with us. And Dr. Priscilla Mumo, if you're present, the floor is yours. We're really looking forward to hearing from you. Karibu sana. Thank you.
Thank you, Professor, and good morning to everyone. I hope I'm audible enough.
You're audible. If your bandwidth allows, you can switch on your camera so that we can see you. If not, it's okay.
I have.
It's not showing. No, we can't see your face.
Let me try. My camera is on. I don't know what the issue could be.
No problem. I think we need your information. So we're loading your slides, which you shared with us.
Yes.
Yeah, I think, Professor Ann, the camera is on. Maybe there's something blocking the camera because it's dark. I can see it's on, but it's dark. So maybe there's something blocking the camera, the laptop camera or something.
You wouldn't have put some masking tape in front of your camera,
No.
would you?
No, it's not there. I don't know what the problem is.
No, don't worry. Daktari, we have your slides present.
Yes.
We are ready to hear from you, and please go ahead.
Okay, thank you. As Professor said, my name is Dr. Priscilla Mutungi, and I work with the Wildlife Research and Training Institute. The institute is a government parastatal that was established to coordinate and undertake wildlife research in the country. And we are based in Naivasha. And now I'll just take I will take you through briefly on biodiversity, sovereignty, and policy foundations for African DSI infrastructure. Next. So when did all this process start? In 1988, the UN and the UNEP set up a committee because of the challenges that were happening. And one of the challenges was the unprecedented loss of biodiversity and the inequalities from the developed world, which was accessing materials. And basically the major complaint was from farmer seeds. Farmer seeds could be accessed and developed into hybrid seeds and then commercialized, and the farmers were left out. So that inequality created a lot of uproar, and UNEP wanted to set up a policy framework, a global policy framework that would oversee how benefits would trickle down to the owners of the resources. And so that committee was set up in 1988, and by 1992, they came up with a convention, and that is the Convention on Biological Diversity, which now was open for parties to sign and accept. This convention had 3 objectives, and the first objective was to conserve biodiversity. The second objective was to sustainably use the biodiversity, and the third objective was to equitably and fairly share the benefits that arose from utilization of the resources, now to solve that uproar that was coming up with the farmers' seeds. Whatever I'm saying, yes. So now after that, the convention was ratified by parties and entered into force. Implementation started. Implementing the first and the second objective was not a challenge, but the third objective took time, a lot of time, on how would the benefits be shared. So in 2002, the parties to the convention adopted what was called the voluntary born guidelines that were basically putting in place how access should take place and how benefits would be shared. But they were voluntary guidelines, and they stated that before access takes place, you need to have what we call the prior informed consent, the mutually agreed terms, before access took place. So now, since these were voluntary guidelines, some of the countries took them and put in place national legislations, like Kenya. Kenya in 2006 came up with the access and benefit sharing regulations that affected what was put in place. Then, since they were voluntary guidelines, countries now went further and negotiated now a legally binding protocol, that is the Nagoya Protocol, which came into effect in 2014, to now obligate parties to actualize those effects that were put in place under the Bonn guidelines. But now the challenge came, the ABS procedures, the prior informed consent, the mutually agreed terms, and the material transfer agreement did not foresee how to regulate the digital sequences that were coming out of the resources. And so that was a legal loophole, and it has brought an uproar because with time, science put into use the digital sequence information that were generated from genetic resources. And now that is what has led the parties now to negotiate what we are calling the Kali Fund. I will explain to it below. Next slide. So implementation of the convention objectives took a coordinated approach. The African Union developed some continental policy frameworks to help countries in Africa to implement the 3 objectives of the convention. And one of the aspects was the African Union and developed practical guidelines for the coordinated implementation of the Nagoya Protocol. They are online, one can see. They have model contractual clauses that parties can adopt and change based on their national circumstances to enhance the implementation of the ABS tools, the PIC, the MAT, and the MTA. We also have the coordinated and unified African common position during the international negotiations at the Conference of Parties, which is held every 2 years. And also they organize high-level forums like the African Biodiversity Summit, which was done last year, to align regional economic communities and member states on policy execution and resource mobilization. These are just part of the policy frameworks the AU puts in place to assist countries in Africa to implement the objectives of the convention. Next. So the current issues with the DSI and the convention is that, one, the convention provides sovereign rights over biological resources and obligates parties who are bound by the convention, one, to maintain and organize by any mechanism data on components of biological diversity, which are important for its conservation and sustainable use. And part of these components of biodiversity is the DSI, the digital sequence information that is generated from the resources. And this is found in Article 7 of the Convention. It also obligates parties to adopt measures for ex situ conservation of the components of biological diversity, that countries or like Kenya, if a country, since it has sovereign rights over its resources, they are obligated to establish and adopt measures for ex situ conservation outside the natural ecosystem. We are supposed to have that system of keeping our resources and maintaining them, the facilities, and also establish facilities, such facilities for research and conservation on all biodiversity, not only plants, animals, and microorganisms, but all your biodiversity within your country, within your national jurisdiction. And this is found in Article 9 of the Convention. Next. Yes, so in access to the resources, several procedures have to be put in place. And the first procedure is to put in place the connection between the resource provider and the resource user. You can see from slide 1 to 5, it's about the procedures and processes that should be put in place or are put in place before access takes place. You negotiate the contracts, the mutually agreed terms, the prior informed consent with the resource providers, between the resource providers and the users. Then once that one is put in place, then access follows the conclusion of the contracts, because the prior informed consent and the mutually agreed terms are legally binding contracts. So once access takes place, now utilization follows. And this utilization can be either in the country of origin where the resources were obtained, or an external country which has already negotiated and is party to the contracts. Now what happens after the Nagoya Protocol was put in place, many countries put in place these procedures for access, the negotiation of the contracts, the prior informed consent, and the mutually agreed terms. But now, it did not see or foresee the utilization of the sequence information that is generated. That is the sequences. Once they are generated, what happens? And therefore, most of the countries did not regulate how sequence information could be utilized, could be accessed, and would be stored, and where it would be stored. Because when you look even at most of the African countries, they do not have these sequence databases. So all the sequence databases were shipped to other countries out of the country of origin. And therefore, with time, now the resources have been, what has been accessed is not the resource per se, but the sequence information that now follows the protocol for production and development of commercial products. And that is where, from step 9, the challenge started. You can see, because from step 9 is where we have the sequence generated, deposited, and now accessed for utilization. Next, something is happening. I can't see my slides well. I can't see my slide well. I don't know what is the issue. Anyway, so what is this sequence information concern? Do we call it a mirage? I don't know, but it is a global concern. And the first concern is that the term was first discussed at the Conference of Parties meeting in 2016. It's now 10 years down the line since COP13. And it has been an agenda item during every Conference of Parties meeting. And up to now, the term still remains a placeholder. There has been no agreed definition, including not only in the CBD, but also in other international UN and specialized forums. And the key issues that have been raised that have hindered the definition of the term is because there has been no agreement on whether the term should be defined narrowly to include only DNA and RNA sequences, or broadly to include, in addition to DNA and RNA sequences, to also include amino acid sequences, proteins, and metabolites. Next. And some of the UN agencies that are using DSI include the BBNJ Treaty. This is the Ocean Treaty. That also places a DSI as a placeholder because it has not agreed. The parties have not agreed whether to use the term broadly or narrowly. The FAO Treaty also on agricultural crops places it as an evolving placeholder and waits to see how digitalization would impact agricultural research. The other UN agency is the WIPO Treaty on intellectual property, genetic resources, and associated traditional knowledge, which has provided no definition of the term and does not also provide any policy measures onto how intellectual property generated from utilization of digital sequence information and associated genetic resources could be handled, and remains silent on the use of DSI. The only international agreement that kind of has put some regulatory measure is the Pandemic Access and Benefit Sharing Agreement that only describes the use of a sequence data from a tracked pathogen should attract benefit sharing obligations from the users of the resource. And so this has remained a big challenge. We are negotiating, we are discussing, but still the definition of what we are discussing has not been agreed yet upon. So this DSI, which has not yet been agreed on the definition under the Convention on the Biological Diversity, has now created a lot of challenges, and that's why 10 years down the line, we are still discussing and negotiating on how it should be utilized, how communities and resource providers should benefit from its utilization. And some of the challenges is because access largely centered on physical and biological resources, that the governing frameworks took little or no caution on sequence information, generation, archiving, and potential future use to provide policy directions and guidelines. Also, national frameworks overlooked the link between the physical resource and its sequence information and did not provide for that regulation. Deposition of sequences also in databases went ahead without any proper source of material description. Like if it is a student depositing before, sequence information would just be deposited without proper description. This sequence was obtained from this particular material. This material was obtained from this particular region. These are the GPS coordinates. These are a lot of information about that sequence was just deposited into databases without all that information. And therefore, databases just kept sequences which did not have a lot of information to tell where the information came from. Things have changed now. Before you provide that, you need to provide all that description. But the previous sequences did not have that. And this posed a lot of challenges, even in the negotiations. Open access sequence databases were also left out of benefit sharing obligations because these were not foreseen. This commercial utilization of sequence data was not foreseen before when the policy frameworks were being put in place. And parties to the CBD also failed to establish national sequence databases. Like in Kenya, we don't have a national sequence database. And this was left to private sector, which would just collect that sequence information and keep them. And through negotiations, now that the private sector has utilized the sequence information they have in their databases to produce commercial products, discussions on how benefits would be shared with the providers of those resources, because the sequences come from the resources, I've ended up with what we call the Kali Fund. And this is a multilateral system of benefit sharing, where all the benefits that are put into that fund belong to everyone and are shared with everyone globally. This global sharing complicates the sovereignty of ownership of resources, because the CBD establishes that sovereign ownership of resources, and it complicates it further because all the accessed materials have now been sequenced and the sequence information have already been deposited in the databases. But now, the benefits that arise from utilization of those sequences are shared with everyone. No one owns anything. We all own everything. And these benefits are to be shared 50% with the local communities, and local communities are, it is proposed that the local communities are to access the benefits directly from the fund. But now the challenge comes even for Africa. Who is this local community that can access the benefits on behalf of Kenya, for example, can access the benefits on behalf of Uganda? Do we have a one system of a community which we can call this is a local community for Kenya, this is a local community for Uganda, this is a local community for Ghana, which would be able to access those monetary benefits from the Kali Fund? So these are the challenges that we are facing. We are still negotiating, but now, what is the way forward? With all those challenges, with all those issues, and the research still continues, we are still depositing sequences, we are still dematerializing our resources and depositing the sequences there. We still do not have databases that can keep that in the country of origin, as the CBD says. So one, parties are to review their legislation to cover DSI or negotiate agile access contracts that cover DSI generation, archiving, and use, so that whoever accesses resources, the physical resource, is guided on how to proceed with the DSI that they generate. Also encourage development of national ex situ facilities that would act as repositories for our sequences and also our resources for future use. We don't know what tomorrow science will take us to, where it will take us to. Today we are discussing DSI, tomorrow it will be something else. But since it all revolves around our resources, we need to have these resources at a national level so that we can have our resources anytime we are interested in using it for research and development. Also developed robust policy frameworks for capacity building on genetic resources, and this includes the know-how and equipment also, because when you have the know-how and you don't have the equipment, that know-how goes to drain. So we need very robust policy frameworks. Also sustainable financing options, especially for ex situ conservation, because even if you initiate an ex situ facility, if you don't have sustainable financing options, then the startup will just end up being a useless venture. Develop legal and regulatory tools on biodiversity that cover the unseen future utilization of biological resources, mainly to cover the changes in scientific discoveries and knowledge. Also establishing a system of local communities that would be able to access the monetary benefits from the Kali Fund. And I would wish to thank the African BioGenome Project because it has really initiated this process. And I hope it will work with governments to help them establish these systems at a national level so that they start country ownership and start negotiating the sustainable financing systems so that we can have a sustained system of utilizing our resources. Thank you very much.
Dr. Priscilla, thank you so much for that very, very insightful presentation. It's set the ground for the conversation that we'll have throughout the day. And now I open the floor to questions, both online and virtually, for Dr. Priscilla.
Yeah. So Professor Ann,
Yes.
I have a question here. Should I go ahead?
Yes, please, go ahead.
Okay, yeah. So I wanted to ask, one of the pain points that Dr. Priscilla highlighted is on, having the infrastructure, the database, especially in African countries, particularly Kenya. So it would be nice to know what the bottlenecks are and if is it the design or what is the main issue in terms of obtaining it? And if an entity does have that provided for Kenya, who has the mandate in Kenya to make it operational, and will this be welcome, particularly in Kenya as well?
Dr. Priscilla, you can proceed.
Okay. Thank you very much. I think one of the challenges is because many countries did not foresee how important sequence data was, and therefore many countries did not go ahead and develop the infrastructure for DSI hosting. This was a challenge. It is a challenge now, but now that we know, countries should put in place these systems. And now you've asked a country like Kenya who should be the right person. It can go in various directions. And one for Kenya, we have an initiative as an institution, Wildlife Research and Training Institute, based on our legal and national mandates on coordinating wildlife research and training in the country. We have taken off that challenge and initiated the development of an ex situ facility in the form of a biobank. We are starting, we've initiated the development of a biobank at our institution as a government to preserve all wildlife biological resources we have in the country. Digital sequence information will be part of it. We have the procedures for that, and that is only for the wildlife sector. We also have CalRO for agriculture that can be also a repository and can be approached for the agricultural sector, because Kenya has now institutions that have direct mandate on particular biological resources. So for the wildlife sector, we have the Wildlife Research and Training Institute. For agriculture sector, you can have the CalRO. For the human and medical purposes, we have the Ministry of Health and we have KEMRI. This is this has been a discussion that has gone on for some time. There's been a discussion which was going on time, but it didn't come up with one unified ex situ facility that could host these resources. I think Professor Ann was part of that discussion, those discussions, which were initiatives that started earlier. But for now, we have those institutions that could collaborate and coordinate to establish such facilities. It would also be good to have, if resources would be available, to have like each institution, like each university, having a small repository for what their researchers are working on.
Yeah.
If they would still keep that, in addition to also sharing with the national institution, that would be very important.
Yeah. Okay, thank you, Dr. Priscilla. So I would have a chat with Professor Ann Mugai. I want us to take this as one of the outcome of this. Professor Mugai knows what we're working on. So I would have a chat with her, and then she can maybe arrange a meeting, and then we can, I mean, I don't have to be in the meeting, but for us to kind of say, how do we explore this further, particularly? So maybe it's something we take out of the current meeting, and then Professor Mugai will have a chat to see how this can be explored further. Yeah, and basically, I also agree with the last point on having laboratories and universities also keep their own record as well. Okay, thank you so much, Professor Ann and Dr. Priscilla.
We have two questions in-house. First here, go ahead.
Thank you. My name is Peter Bogo from CME, Center for Malaria Elimination, Mount Kenya University. I have a question, or maybe a concern. What challenge are you facing inside that it's leading to these discussions, or to take such a long time to come into a conclusion of establishing such a national body? And what policies do you think should be implemented to make sure that if certain biodiversity commodities are established from Kenya or any other country, remain part of that country in such a way that they can receive royalties? And also, what measures are you taking to ensure that there is technology sharing between the research institution in one country? For example, in Kenya, there is technology sharing between maybe KEMLI, ILDI, and other research institutions. Thank you.
Dr. Priscilla, hold that question. We will follow it with another one, and then you can answer the two together.
Okay. Good morning. My name is Josephat Geshure, CME, MKU. The first question, the way the response came out, for me feels like that may not be the best approach, where wildlife hosts differently, agriculture hosts at Carroll. It will create a very big conflict. We know right now, if we have, let's say, an issue with food safety, you will find that almost 10 institutions, NEMA, all these institutions will be involved, and there will be no address, even for researchers. If I want to do let's say, I want to do a follow-up of an issue which I assume it's related to wildlife, how will I know specifically which place to get the DSI? For me, I felt that the best place to host this will be in an institution which is government-owned, but it's central, it covers all these other institutions, rather than have wildlife to have its own, food to have its own, agriculture to have its own, everyone, veterinary will want their own, so it will create a lot of confusion in that. But that's my observation. Thank you.
I think just to add on what he has just said, Dr. Priscilla, there are some cases where you can deal with crops, and these crops also have their own wild relatives. So in that case, will they be deposited in the agricultural facility like in Karo, or will they be deposited in the WRTI where it falls under wildlife? And I think that's what he was trying to say, or just to bring to attention, where if we have different institutions databasing or having these different categories of plants or data that we have, molecular data that we generate, then in the long term, I think there will be a lot of problem or issues unless we define these things fast before we establish the infrastructure. But just for the last question, and then you can add, you can answer all three. There's a hand online from Hewan. So I think Hewan, you can also shoot your question, and then Dr. Priscilla, you can answer the three, and then we wrap it up. Thank you.
Okay, thank you very much. My name is Hewan. I am here in Ethiopia, Hawassa University. My question, and specifically with this thought-provoking presentation my sister presented, is when we look at the biobank or this database storage, that definitely we really wanted African base database. But do you think that we Africans are really ready? I am asking this question because when we look at the different aspect of this database, like, for example, infrastructure and technical capacity, in Africa, we still have a lot of frequent power interruption, limited ultra cold storage condition facilities, even at government high institution level, fewer high throughput sequencing hubs, which is not well. And I think because of the BioGenome Project, I think we are really growing better. But also my question is also on governance and database sovereignty, and specifically with ethical control. Are we really there in the governance frameworks still emerging? So, but no, there is risk of external institution, they might control the African samples. And because of this historical pattern of extractive research that we have and weak enforcement of benefit sharing and data sovereignty is there. And another aspect is funding stability. In Africa, still we are relying on external donors. I don't know how the policies are really influencing the African Union, to give some funding specifically for this infrastructure development. So reliance on external donors with limited national research budgets and difficulty maintaining long-term operation without this external support might harm also this database. And the other one is the scientific utilization and local research capacity, which really worries me a lot. Because when we look at in Africa, we have fewer trained bioinformaticians and limited access to high performing, competing and lower utilization of these African samples for African-led researchers. And the other one aspect is representation and equity. So when we look at the global biobank, like specifically Europeans, they are really strong on that aspect. So if I summarize all and to bring to your attention my question, in given all this global concentration of biobank in Europe, in North America, what structural and governance difference must Africa address to ensure that in Africa, an African-based biobanks strengths data sovereignty, long-term sustainability, and equitable scientific research in Africa? So how much we really trust each other as an African Union? That's my question.
Thank you, Dr. Priscilla. Please, we can answer that.
Okay, thank you. Let me start with the first one. The first question was on how well prepared we are. Why it has taken so long to get the benefits. We have some countries in Africa that have received the benefits, and we've received a lot of benefits because the Nagoya Protocol does not provide only for monetary benefits, but provides for both monetary and non-monetary benefits in terms of training, equipment, and capacity building. So there are benefits that we have perceived as a continent. And this can be seen. You can go to the ABS clearinghouse on the website, and you can see the reports that countries have provided under the Nagoya Protocol, because we submitted the first national reports. There are benefits which are there, but now when it comes to technology sharing, the challenge comes if the access of the resource did not have a very tight and well negotiated, legally binding contracts with the user. That is where the first challenge starts. So it is very important to note, as a researcher, you need to have that contract very well stated, very clearly stated, and in a form that it is legally binding. With that, you will have benefits coming back and technologies being shared well. When it comes to the conflict on institutions and hosting of these resources by every institution, you understand that the law, every institution, every government institution is established based on a particular act. And therefore, the act provides for the extent to which you are supposed to operate. Like the Wildlife Research and Training Institute is essentially established to oversee, coordinate, and undertake wildlife research and training in the country. So we cannot go to another institution's mandate. And that's why we are because our mandate restricts us to wildlife, we keep our wildlife. That notwithstanding, we've had initiatives initiated to bring all these institutions together that are working with biodiversity. Because the challenge we have is, unlike Ethiopia, Kenya does not have one biodiversity law that oversees all biodiversity in the country. It is fragmented. The forestry is on its own, fisheries on their own, wildlife on their own, agriculture on its own. So we had an initiative to bring on board all the ministers responsible for these resources, because you also understand the Ministry of Environment has the forestry resources under it. Wildlife is in a different ministry. Agriculture is in a different ministry. And therefore, you need a coherent and a uniform position from all these ministers. It has taken time, but we have not given up. We are still pushing that we can have all biodiversity under one resource. It is at that point when we can have one biodiversity act that controls and regulates all biodiversity resources in the country, that we can have one common resource, the way you are saying. But there is no conflict, because if Wildlife Research and Training Institute keeps what it is mandated to keep, then there is no conflict with agriculture, because we cannot go to agriculture. That is not our mandate. But there is that coordination. If you need anything for research, you are able to access the particular resource you want from the institution that is responsible for that. So as a researcher, when you want wildlife resources, you need to know which institution to go to get what you want. The other question was on why we have the governance issues in Africa. Are we ready as Africa? I think we may be ready and not ready. Ready in the sense that we have a coordinated approach. We have a unifying coordinating institution, that is the African Union. And not ready because since this includes resources from every country, we need a policy framework that would govern how do countries deposit. If it is an Africa-wide repository, how would countries in Africa deposit their resources? How will the governance of that facility be done? It needs a policy framework that would govern how the facility would be funded, how it would be established, and how it would be managed, and governed. So that should not be an issue. It only needs a push from the African Union, a coordinated push, and how the facility would be financed and sustained with national finances of the African countries, not external factors. If we bring an external partner, you expect additional inputs, and you've now broken the chain of the Africa system. So if it has to be Africa system, from my opinion, the AU should develop a policy framework and a financing mechanism from Africa itself, the countries themselves. That way it would be sustained, and it would, I think it would work well, because there would be that policy framework to govern deposit and use, release of the resources. Now there was this issue of the world relatives and agriculture. The laws are very clear. If it is agriculture, what is an agricultural crop? If it is world relative, what is a world resource? So the Wildlife Act defines what a world resource is. The Agriculture Act defines what an agriculture resource is. So the two of them are very separate. If it is a world relative, it is a world resource. It is not an agriculture resource. So you need a concerted effort. But we've not had any issue where there is a link between two resources. We would work together. Like when you would want to access a wildlife resource in a forest ecosystem, we work together with the KFS to make sure the researcher does not get through all the legal processes. If it was started with the Wildlife Research and Training Institute, then the KFS would accept what we work with, what we have developed, the system we have developed. If it started with the Forestry Act at the KFS, the Wildlife Research and Training Institute would accept, because we are all government, what system has been developed and put in place to access the resource through the KFS. So we work in coordination since we are all government. Thank you.
Thank you very much, Dr. Priscilla. That was a very insightful presentation and also discourse. Could we give Dr. Priscilla a round of applause?
Yeah.
We have to move on to the next session, unfortunately, and Dr. Priscilla also has to move on to other official matters. But we thank you for taking the time to be with us this morning, and we appreciate it very much. We're now going to have the next two presentations following one another. And the next presenter is Professor Douglas Miano, who is a professor of plant science and crop protection at the College of Agriculture and Veterinary Sciences, University of Nairobi. He holds a PhD in plant virology, which he got from the Louisiana State University in the US. He holds a master's in plant pathology and a bachelor of agriculture from the University of Nairobi. He's an accomplished scholar with high number of peer-reviewed presentations. We are extremely honored to have you, Prof. Last week I was looking for you and you are in these high-level negotiations. So we appreciate the time that you have taken to interact with us now, especially having come out of a very, very busy week, and the floor and the mic are yours. Thank you very much. Welcome.
Thank you, Dr. Muigai. I hope I'm clear.
Yes, you're very clear. Do you have slides, or are you going to just have a conversation with us?
I'll present the slides if I'm allowed to do so.
They're sharing the rights right now. Try now and upload your presentation.
I hope you can see it.
Not yet.
Yeah, I think we can see on this side, Professor Mugai.
Yes, we can see it now
Okay,
here.
okay.
Please proceed.
All right, thank you. Allow me also to switch off my video so that then I can be able to reduce the bandwidth. All right, I hope I'm clear now and the presentation is clear.
Yes, you're clear and the presentation is clear. Please go ahead.
All right, thank you. As Professor Mugai has said, my name is Douglas Miano. I'm a professor of plant pathology at the University of Nairobi, the Department of Plant Science and Crop Protection. I'm a scientist, so I'm not so much into policy issues. So most of the things that I could have to do with the governance on DSI and biodiversity has been discussed by Priscilla. So I thought of looking at it from a different angle and looking at how did we come to discuss about the issues of DSI. So I've been in the discussions through the CBD, the Convention on Biological Diversity, since the negotiations began, that is in 2016. And what I wanted to take you through is what has been these discussions about all those things that Priscilla was talking about. So how has it been progressing so that we can reach to where we are today? So what I wanted to bring to your attention is that when you're talking about DSI, especially in line with conventional biological diversity, these discussions came when thinking about the objectives of the convention. And as Priscilla said, the conventions are three. There is a conservation of biological diversity. There is the sustainable use of those resources and how to share fairly and equitably the benefits that are arising from the utilization of those genetic resources. So the discussions on DSI basically rose out of these three objectives and the concept of what DSI is. So if we look briefly at the history of the DSI, the terminology and how we are talking about its governance, the discussions started in Cancun in Mexico in 2016. This is what we call COP13. And developing nations argued that the use of genetic data without sharing its financial or scientific benefits constituted a loophole in the Nagoya Protocol. And I think the presenter before has highlighted the key loopholes that were being seen. Because what happened is that the CBD had envisioned the biological resources as a physical genetic resource. And they had not seen that there will be a time when you can be able to use the genetic information to come up with products or services that are supposed to bring in benefits. So, and the question was, is information that is generated from a genetic resource also a part of genetic resources? And this has been the big discussion, and the question of how to access, how to share those benefits that could be arising from the use of digital sequence information. Do we use it? Do we use the multilateral or the bilateral system that was envisioned at Nagoya, or how else can we go about it? So the thing that I would want to bring across from the word go is that the digital sequence information is still under negotiations. The access to information, the benefits that are coming out of it, how they should be shared, where do they go, all of these are still under negotiations. They are not necessarily only at the CBD. They are also under negotiations in the different other bodies like VBNJ and the FAO and WHO. And I think these, as Priscilla has already mentioned. But the point I wanted to bring across is that the question of how are you able to say that the benefits that are coming from digital sequence information are being shared equally, are being shared to the people who are supposed to be benefiting from them, and how do we trace back to where those sequences may have come from, so that then we can be able to share benefits. So there are key decisions. If you look through the history of these negotiations on DSI at the CBD, and so every 2 years, the parties meet and discuss different aspects of this different agendas under the conventions. And as I said, the first discussions were in 2016, what you call the COP13. So, and what was decided then is that, and what was realized is that digital sequence information is a cross-cutting issue that may concern all the 3 objectives. It came to discussions under the Nagoya Protocol. Remember, the CBD has the convention and has 2 protocols plus 1 supplementary protocol. There is the Cartagena Protocol that deals with biosafety. There's Nagoya Protocol that deals with access and benefit sharing. And there's a supplementary protocol, which is also what you call the Nagoya Kuala Lumpur Supplementary Protocol on Biosafety, which is kind of complements the Cartagena Protocol. But this DSI was brought into the negotiations through the Nagoya because of benefit sharing. But because of its nature and realizing that the digital sequence information was still part of what was being used in, for example, in genetic engineering, and the fact that it is touching on genetic resources as a resource itself. So it was decided that it be discussed not under the Nagoya Protocol, but it be discussed under the convention, or the CBD convention, the mother convention itself. So what was also decided, it was to consider the implications of the use of DSI on genetic resources on those 3 objectives, and also the aspect of the scoping. What it constitutes the digital sequence information. Is genetic materials part of it? Is it only the sequences? Does other information that could be generated from the materials also constitute a DSI? So a technical working group was developed so that they can look for this information, to have started to look at these details. Some of them was to clarify the terminology. When you talk about DSI, what do we really mean? And so also looking at the concepts on how to assess the extent and the terms and conditions of the use of DSI on genetic resources. So it was kind of a scoping study so that to look at all the information that we needed so that we can be able to negotiate in a way that we are saying that it's all inclusive. So the first point that was critical was that we could not just leave these negotiations on DSI to only access and benefit sharing protocol, that is the Nagoya Protocol, but we needed to look at it from the wider scope of the convention itself. So the other decision that came in COP14, this was held in Egypt in Sharm el-Sheikh, and this to me was a key and fundamental in terms of being able to look at the whole concept of the DSI. So the first thing was that the term DSI was considered that it may not necessarily have been the most appropriate term to use when you're talking about the information that is being generated from the genetic resources, but it was agreed to be used as a placeholder. So even up to today, that terminology of DSI is still being looked at as a placeholder, meaning that the information, at some point, the terminology may change. So the other point that was critical is that to consider that when you are thinking about DSI, it is not only about the genetic resources, it's not only about the access, the benefits themselves alone. The information is important to researchers, and you appreciate that we may not necessarily be getting all the benefits in a commercial way. There could be also non-commercial benefits that are coming out of it. For example, if you look at the FAO, their sharing of information on genetic resources was kind of multilateral and not necessarily bilateral because of the benefits that it was getting from food for the safety of humans. So again, the COP decided that there is still more information that needed to be generated on top of what information was generated earlier. And they formed another group of experts to look now deeper into the issues of how far can we go and associate the genetic information to the genetic resource. Where do we say that the genetic information ends and where do we say that genetic resource starts? There was also the question of if we do the sequencing and you want to say that you're going to have the benefits arising from the use of those sequences, coming up with products and resources, how will you say that they can be able to reach to the source from where the genetic resource came from? And the question of how to govern the issues of DSI within the country, what Priscilla was bringing to, and some of the questions we are coming on to. How can you say that you're going to be able to govern the issues of DSI within the country? So what are the policy options that we can have so that we can be able to utilize? So all of these formed the study that was established from the decision 1420. And some of the concepts that came up is trying to define the scopes. Where do we start saying that there is information? And for example, the issue of DNA and RNA and the issue of the proteins was considered like that could be part of the information. But as you move forward towards the formation of the combination of the proteins with other metabolites and all those. So then that may go beyond what you may call the digital sequences, but it is still part of information, which is important and relevant to when you are considering the issues of benefits and the coming up of products that may be coming from the information that is generated. So these are some of the a wide view of some of the areas that we are considered that we could look at. How do you come up with different policies that would be able to inform or govern the issues to do with DSI? This was a big discussion, and some of these concepts now have helped us to come and start looking at how do we manage the issues to do with DSI. So the next decision was the one that came 15.9, and this was the decisions that were made from Montreal in COP 15. And some of the issues that came up is that the issue of scope of whether you are calling the resource itself, the genetic resource as a part of the DSI, we are still having people who are still discussing on whether we should link or associate DSI with the genetic resource as one and the same thing. So, but we still agree to continue with the terminology of DSI, using DSI for discussions. And one of the fundamental things that was also agreed is that it is not possible necessarily to trace all genetic information, all the sequences to a particular genetic resource in a particular country. So tracing back, especially for some of those sequences that have already been placed in the gene banks, may not be an easy part. So the other thing, decision that was also important in COP 15 is accepting that there is some benefits that could be arising from the use of from the DSI that may not necessarily be traced to a particular country, or for example, genetic resources that are from different geographic regions. And because of this, a multilateral approach of sharing benefits was proposed. And this is part of what resulted to the creation of the multilateral mechanism of benefit sharing, and especially the global fund, what you are calling the CARI fund. And this decision was eventually agreed upon to create the fund in COP 16. Part of these discussions was to look at, when you talk about the fair and benefit sharing of digital sequences, especially when genetic resources are going beyond one particular country. So then coming up with a system of sharing those benefits. So we had to look at different aspects. What are we looking for? If we are going to have a fund our system of sharing the benefits, our system of putting the resources together. So there were some considerations that had to be made into place. And if you look at decision 59, a solution for fair and equitable sharing of benefits arising from DSI should be efficient, it should be feasible and practical. It should be able to generate more benefits from both monetary and non-monetary. It should be effective. It should provide legal certainty, provide clarity, certainty and legal clarity. It should not hinder research. It should be consistent with open access to data. And I think this question has continued coming now and then, to especially on whether Africa is able to access, to keep these, to have data banks. So it has to be compatible with international legal obligations. It has to be mutually supportive for other ABS instruments, and also take into account the indigenous people and local communities, including their knowledge. So at the moment, we are looking at the decision that was made in Colombia, that is what you are calling decision 16.2. And the critical thing here is that this decision established the CARI fund. And the question is, how are these funds going to be taken from the people who are using DSI and deposited into the CARI fund? And how will those resources be coming out of the fund? What systems or what mechanisms do we have to have those benefits streaming out of the fund, even as we think about how we get the funds from there? So looking forward to COP18, there are a number of things that we need to discuss about. For example, the governance of the fund. This is a big discussion that we had in the last meetings in Nairobi, so that as we prepare for COP17. So how should we be able to enhance more contributions to the fund? How will those funds that got the contributions that got the CARI fund, how will they be distributed to different users? And also how do we capture the non-monetary contributions and other modalities of how we can be able to manage the mechanism? So in conclusion, what I would want to say is that the issue of digital sequence information and its governance, even before we come to the national level, even at the international level, the discussions are still going on. There are still many areas that needs to be agreed upon. So then and of course, in COP17, it will be important for us to make the decisions that will help us to make those contributions even as we continue on. So I think that's what I have for now. I'm ready for questions.
Thank you very much, Prof, for further clarifying a conversation that was begun by Dr. Mumo. And indeed, we are ready to ask you questions. So questions from the floor, we have one here. We'll take 3 questions from in person and also virtual, and then we can allow Prof to answer those, and then we take the next round.
Okay, good morning. I'm Dr. Mandela from Pan-African Institute for Genomic Sciences. Thank you so much, Prof, for this insightful presentation. I got curious a little because I realized that most of these policies are being developed in hindsight. Is it possible that there are materials that had already been maybe taken from our environment before the development of these items, and probably they are already being used somewhere? And is it possible, or are there ways we can go about tracing for the benefits? Because I have remembered that there's something called horseshoe crab that used to be indigenous to some place, and its blood cells were used to develop some in vitro diagnostic test. And I realized that there was no framework for sharing benefits. Is it possible that now that we have conversations around this, is it possible to trace some of these benefits to the indigenous communities? Thank you.
Thank you. Do we have a question online? As we're looking, Prof, you can answer this question that has just been asked.
Yeah, thank you. And I think that's a very important question because I'm a victim of that. I isolated a virus that was supposed to control the insects, Lepidopterans, many years when I was a young scientist. And then those sequences went out, of course, where we deposited them in the samples. And after that, they were commercialized. The virus sequences were commercialized as a product for insect control. And we still don't get benefits as a country. So, and partly is because some of these treaties and conventions, when they come into practice, they may not go backwards to say that it is covering what has already been taken out and has already been operations. So if you had a material transfer agreement, and depending on what you had already agreed on those material transfer agreements and other agreements that were necessary, you may be able to get some of those benefits, but the convention may not necessarily be going backwards to some of those places. And I'm sure there are other people who might be better off in discussing this more than I do. But what we are now doing is to say that when we talk about the multilateral fund, if you cannot be able to trace the benefits that are coming from the use of those DSI, and but the companies are using that information. The decision 16.2, as we are calling it, that came from that established the Cali Fund, it has the different modalities of how those contributions can be made. And the point is that any company that is generating resources from or is getting benefits from use of DSI should be able to contribute to the Cali Fund. And if they contribute to the Cali Fund, then it goes to a multilateral fund, the Cali Fund, which is multilateral now, not necessarily sharing the benefits directly from where the sequences may have come from, but those benefits will still be going back to helping in the conservation of those genetic resources. So even when it might be difficult, especially to trace backwards, but we are saying is that anyone who is using digital sequence information should contribute to the fund. And if it goes to the fund, then somehow it will still help us in terms of being used for conservation. Thank you. That's what I want to say for now.
Yeah, thank you. As we look for whether there are any questions online, I think that another challenge would be the awareness about the importance of the material transfer agreements and the other legal frameworks, because if I think about the number of field trips I've done, we knew that we needed to get ethical consent. Maybe the focus might have been on that, and even the legal drafting of the MTA, we never had a lawyer look at it. We probably just, customized the form that we found online and applied it. And then you realize now that you actually excluded yourself from benefit sharing. So what can we do to address this gap?
Yes, thank you. And I see that there's also a comment online that most of the benefits are generally associated with the physical genetic resource. So there is a lot of awareness to be created, that is for sure, starting also from our own government, appreciating what it is that the benefits that are coming out of a genetic resource. And the problem even goes further because partly we have also not monetized our genetic resources. If you look at our natural resources, that we don't have a monetary value that we have attached to it. So awareness needs to be created. And we have, as a country, I think that's one of the things that is being done now to make sure that we can have a way of valuing every resource that we have. And then after that, apart from awareness creation, and where the people are still using genetic resources, even when people are not aware of the value of it. For example, if anyone is using sequences to come up with, let's say, a perfume, and you may not necessarily be able to say where those resources have come from, that is what the multilateral fund was trying to cure. So that then you are saying that as long as you're using a genetic resource, and as long as you are getting benefits, you should share those benefits with the people who utilize, who are conserving those resources. And Priscilla talked about the 50% that of the Cali fund that should be going to the indigenous people and rural communities who are involved in the conservation. And it's also still a big discussion because how the funds are being distributed and going, the 50% going to the indigenous people and the local communities is also not straightforward. And there's a lot of awareness to be created, even from the systems that you are creating through the CARI fund and the discussions that you are going on. Because at the moment, we are still having the issue of looking at the genetic resource as a physical material without realizing that most of the benefits are not coming out of the sequences that are being generated. So it's a question on which parastatals are enforcing the MTAs and PICs. I think this is a big challenge for now, because as you heard the previous speaker talking about the institutions which we are managing. If you look, for example, at the Nagoya Protocol, the focal point, and the people who are dealing with those resources are all different. There is a Ministry of Environment, there is NEMA, there is WildLife, there is NACOSTI, and none of them are having a say in terms of where the licenses are coming from. It's still, I wouldn't want to call it, there's still some confusion for us as a country, which needs to be clarified so that you can also have a proper licensing system. And because you'd have to get the research permit, for example, from NACOSTI, you'd have to get access of materials through if you are looking at the WildLife, through the WildLife services and all that. So that confusion still is there, but I'm sure with time it's going to be clarified, even as we make sure that those benefits are coming back to us as a people of Kenya.
Prof, we're very, very grateful and thankful for that very insightful conversation. In fact, it's not easy what you're doing, because you see the Cali Fund, if they say that 50% goes to the indigenous people of Africa, then, I mean, Africa is so expansive. You know, we have indigenous people everywhere. So who do you give and who do you leave? And if you decide to share it between all the indigenous African peoples, then it's close to giving us nothing, because, you know, we are populous, we are many, and therefore the benefit will not even be there. So we understand how difficult these conversations you're having are, and we appreciate that you're ready to put yourself out there and negotiate on our behalf. And we as African BioGenome Project have started this conversation, and we would be very, very glad to support you in this process, and also sit down with you and have a sharing of ideas on what we can do, not only for our individual nations, but also collectively for the continent in Africa. Thank you, Professor, and we wish you a very good day, and thank you for your presentation.
Thank you, everyone, and it was a pleasure.
Let's give Professor a round of applause as he exits. That was a very, very insightful session. Do you agree with me? Talking about Africa, access and benefit sharing, the Cali Fund, indigenous communities, MTAs. It's really, you know, and the definition of what digital sequence information is, and the next term that will actually come out of that. And I think it's important that this conversation is actually happening and beginning. And we are happy as African BioGenome Project to even start this conversation and begin to understand and also see how we can also bring you as the scientists who are with us in order to bring these conversations closer to home. So now we're changing gear, and you've seen in the presentation that the access and benefit sharing and the CBDs also have other conversations beside DSI, besides the access and benefit sharing. We have how the KMGF framework is going to be implemented. And therefore, this next session is going to talk about indigenous knowledge, gender and participation, and what it means, especially now in this genomic era. Our next presenter is Marie Salvatrice, who is an environmentalist and a sustainable development expert with more than 17 years experience in biodiversity conservation, climate change, gender justice, and sustainable development. She's an active member of the CBD Women's Caucus, where she negotiates for gender responsive implementation of the KMGBDF framework and the Rio Convention. She is based in Rwanda, and her work in Rwanda and the greater Vuruga landscape has focused on sustainable mountain gorilla conservation, habitat protection, restoration, sustainable livelihoods for communities living around this critical ecosystem. She's a founder and managing director of Go Peace Restore Africa Action, who's advancing sustainable biodiversity conservation, community engagement, climate action, gender, and a transition towards inclusive and sustainable development. Marie will also moderate this session, and I'll be here as her very able assistant. So Marie, please go ahead, and we look forward to your presentation.
Thank you so much, and thank you for having me. I'm happy to be part of this discussion. It's really exciting to see the discussions from yesterday up to now. I hope you can hear me very well, and we are learning from each other. I'm not coming to teach, I'm coming to learn. I've been learning from yesterday and until today. And today what I want to take you back. I can't see you. I want to see people in the room. Are you excited today? How are you today? I've seen young people there, and it's really nice to see those young people eager to learn, and they are the future leader in biodiversity conservation. And I look forward to what we are going to do. We are discussing about negotiation in traditional knowledge. And today I want to take you back to the village where you are coming from. I want to take you back to the work of your mom, your grandma, the beekeepers in the village, the agriculture in the village. What is there? When we talk about biodiversity conservation, when we talk about traditional knowledge. When I talk about the communities, I am always excited because sometimes they call me community person. And I always want people to consider what the life we live in everyday life. And sometimes we forget. From yesterday, we were discussing about research and research recommendations from the research, and how those research and recommendations are simplified to reach local communities and to reach youth, and to integrate them. Because sometimes you find that people are doing research, sometimes you find women are not included. The village people are not included. And what are we doing then? So I can maybe share my presentation a bit so that I can continue. Can I?
Please do that. Thank you.
See if I can share. I think this technology is
Take your time, no worries.
So Marie, are you okay?
I
There's that
actually,
arrow at the bottom.
Yes, I am there, but you can't find how to. I always share screens, but today I don't know what is happening because it shows share settings.
Happens sometimes.
Eh?
So you're the co-host, so you should be able to upload.
Yeah, I am the co-host, but I don't see. Anyway, let's continue because I don't see where I can.
Or you can quickly email it to me, and as you're having the conversation, we can.
Okay, I think it's better because I tried. I don't see PowerPoint. Let me see PowerPoint. Yeah. Open system Zoom. Request me to open system Zoom. No. Anyway, let me send to you, and you can share.
Yes, please.
As we continue our discussion here. Even I think it's not necessary to make a presentation. What I'm going I can send it via your email.
Yes, just send to
or in the chat, the host in the chat.
In the chat, it's okay? Yes, it's okay.
I can share in a private message.
Message, yes.
I think I sent you.
Please do that.
Sorry. Okay. Fixie Technology.
Yes.
There is something.
Marie, we've sent you a chat. Just respond to it.
Okay. Thank you. The issue is that it requires me to share through Google Drive, and I have not saved it on the Google Drive. Anyway, let me continue. I think there is no problem. Maybe it's not necessary to share my screen. As I was discussing.
Yes.
As we can continue discussion. What I want to say today is about Kuming Montreal Global Biodiversity Framework. What is it? And why we are discussing this today? As I was telling you, from yesterday, we were discussing about technology, about digital sequences, information, and we are discussing so many things. But when it comes to all those discussion, we go back to biodiversity conservation. Biodiversity is life. When you are tired, what do? You go to the village, you go somewhere, you go in a park to relax and to breathe to have a good breath. And when you are you have cough, what do you do? You just think about traditional medicine. So this is biodiversity. Biodiversity is, we are talking about Kuming Montreal Global Biodiversity Framework. It's the framework that's engage people, that guide people on how to conserve species, animals, plants, and land, water, and life underwater. All those ecosystems, I said I to take this as the whole ecosystem, but all around the central of this is people. People are living within biodiversity. They are benefiting from biodiversity, and how can this can be sustainable? How can this can be done in a way of sustainability? We remember we have like wildlife conservation, where we have most of the time when we talk about biodiversity, some people, they think about wildlife. It's not that, only that. It's the whole ecosystem. It's the living species and also the microorganisms we don't see. I am talking about this so that we can know what is the role of all of us. It's not about those high-level policies. It's also, for me, when I talk about this, I talk about how to link global policy to the grassroots solutions. That's why I'm taking you to the village. Biodiversity conservation. In the coming Montreal Global Biodiversity, by 2030, they went to conserve 30% and restore 30% of land, of water, of all resources. But what can this be done? This is the role of everyone. If I come back to what my previous colleagues presenter, they talked about coming to Montreal. They talk about somehow they linked it to the different governance and also different technical working group that implemented the coming Montreal Global Biodiversity Framework. What is the Global Biodiversity Framework? It has I can say 4 goals: to protect and restore ecosystem integrity and species survival. Second, to prosper nature, sustainability of use of biodiversity. Most of the time, people think about money. And money, we forget that we are using resources which are limited. How can we make sure that people make money, use the natural resources, but also conserve those resources for future generations? That's why I was saying I'm happy that there are many young people here. They have to think about how to use the resources, natural resources, and without compromising the benefit of future generations. And when we were talking about the DSI and how those resources, modification of those resources, and also how the traditional knowledge, sometimes they are used to generate new knowledge. And when those knowledge are used, how do they consider those traditional knowledge they have been used? How local community they can benefit? How those who own the original information benefit from that? How are they integrated? This is inclusive. Inclusive is participation, but efficient participation, not just going to the meeting and sitting. It's efficient participation, but also how do those people are supported, how to add values on those natural resources, on those traditional knowledge owned by our grandmother, our grandfather, our people in the village, so that they can be integrated in the global scientific knowledge. It's complementarity. I'm talking here about complementarity. Also, invest and collaborate. When you invest, we see a lot of things happening with the private sector. How the private sector is engaging local community. How do they consider they are concerned? The human rights. We are talking about human rights. Then together we are talking about. Now I am talking about one society approach. This approach, it integrates everyone in the process so that the conservation of biodiversity can be inclusive, but also can benefit everyone. Within those 4 goals, there are also targets, 23 targets. I will share my presentation, then they will share with the participants. But what I want to tell you, we can't say everything today. you have to go and read. You go to the CBD Secretariat website, you read, you understand, and also you take actions. Don't look at the big actions. We are looking into the grassroots solutions, small actions, which lead to big actions. From that, I am moving to another, the governance of coming by Montreal Global Biodiversity Framework. What is the governance? The previous presenter focused on different the COPs, the SABSTA, SPI. SPI is about implementation. SABSTA is about technical, the scientific committee that advises the implementation. Come with the knowledge, and those knowledge are implemented following the SB8G. SB8G was adopted because they find out that the local communities have knowledge. Local communities are contributing a lot. They have to have a space, a clear space in COPs, in those meetings, so that they can contribute to biodiversity conservation. And when it comes to implementation to the global biodiversity framework, we come back to One Health. After COVID, we have seen COVID, have seen how many species were affected, the people were affected when it comes to transmission of diseases within human being, and also livestock, but also wildlife, especially when it comes to mountain gorillas, as I am in mountain gorillas. The 90% DNA, 97% DNA as human being. Imagine when the disease transmission is very close. We are talking about a One Health approach where everything we have in conservation, biodiversity conservation, we have to look at people, animals, living species, but also micro animals, micro species, so that we can make the whole ecosystem healthy. As the whole ecosystem is healthy, people also will be healthy. When I talk about this, sometimes I come back to the food we consume. And when I talk to the role of women, I come back, I say, who doesn't say, I want to eat traditional chicken? So, and when they go to buy to buy food, they say, this is traditional. And now it's more expensive. So we have to think of those traditional knowledge, which are really, I take this, they are original. I am not denying that the Gemon or those technology is wrong. It's good but we have to combine all those knowledge so that they can complement each other. So from there, I'm coming back to one society approach. Also, I talk about human rights approach, gender responsive approach. You can't forget the role played by women in soil biodiversity, subsistence agriculture. When it comes to big organizations or companies doing agriculture, this is okay. But go to the village again, look at the traditional, how people are eating. They are the majority, and they are using their traditional knowledge. Most of the time, you find that during those negotiations, when it comes to agriculture, there are many issues. They don't agree on different things. This is where we have.
Marie, maybe you can start to wind up so that you can also have other.
Thank you. Thank you so much. And then inclusive, it's about stakeholder engagement, research institution, private sector, local community, indigenous people, and UN agencies. All they have to work together so that they can achieve those goals. Coming back on EPRS and local communities, we have that body. We have also implementation by countries. And also those countries, the commitment of countries should consider engaging local community and women. And to sum up, when I was referring to gender responsive approach, where we have to recognize the role of women, and also when I talk about the role of women, look at the energy, firewood, water, food, soil biodiversity, all of those, they integrate traditional knowledge, but also they are linked to climate change. They do play a role in climate change adaptation. So I think the key messages from here, this is it's a global commitment that deliver 2023 target, which you can find on their website, and everyone is involved, and everyone should play a role. And coherent governance, it's inclusive. No one should be left behind. This is the motto of SDGs. I always link SDGs with the document on Montreal Global Biodiversity. And share the responsibility. Everyone should have responsibility and be accountable. And also, there is no one who can be left behind. Women are agents of change. Youth are agents of change. We don't have to forget about this. I invite those people here. As I said, I am a member of CBD Women's Caucus. We do advocacy for the recognition of traditional knowledge and the role of women in biodiversity conservation. Please visit the website, and when you want, you can join and be biodiversity gender champion. Thank you so much. I am happy to be part of this conversation. I hope another time we have time to come together to discuss. Thank you so much.
We will. Thank you. Thank you so much, Marie.
So Marie, please don't leave us. You can remain online, because we are waiting for that very interesting conversation that we will have, especially with the comments that are coming from the audience here, the ones that I have seen online. And we can have that after the next two presenters. Our next presenter is Mr. Daniel Mukubi, who is an international renowned expert from the Democratic Republic of Congo. He is deeply committed to issues related to biodiversity conservation, climate change, and sustainable development, engaging with indigenous peoples and local communities. In this capacity, he is DRC's negotiator for the CBD and the UNFCCC, working for the Ministry of Environment, Sustainable Development, and New Climate Economy of the Democratic Republic of Congo. Mr. Mukubi is going to give us a presentation on empowering the guardians, bridging indigenous knowledge and genomics in DRC and beyond. And you have the floor, and you can also share your slides. And you're very, very welcome.
Okay, thank you very much. And thank you for inviting me. Can you ensure me that you are catching me well?
Yes, we can see you. We can also hear you.
Thank you very much. As we wanted this session to be interactive, I will not share some slides, but I will share my thoughts about this topic that you have already announced, how to empower the guardians and to bridge indigenous and the local knowledge and genomics in DRC and beyond. So, for example, you will know that for decades, Western scientific paradigms have viewed genomics and indigenous ethnobotanical knowledge as separate, if not parallel disciplines. While biogenomics often treats genetic resources as raw data, and while the ancestral stewardship that preserved these resources goes unacknowledged. So in the Democratic Republic of Congo, as you know, it is home to over 60% of the Congo Basin forests. Now, indigenous peoples and local communities have served as the ultimate guardians of biodiversity. And this presentation explores how the historic outcomes of the 16th Conference of the Parties to the Convention on Biological Diversity provide a concrete legal and ethical blueprint to bridge biogenomics with indigenous peoples and local communities knowledge, ensuring that scientific advancement no longer comes at the cost of exploitation. So you will recall that the new global biodiversity framework is also for genetic data. So a critical frontier in modern science is digital sequence information on genetic resources. Genomic research increasingly relies on open access databases, and we understood it from Dr. Priscilla this morning, and containing sequences uploaded from across the globe. However, this has historically created a loophole where the physical country of origin and its communities are excluded from the commercial benefits. So Decision 16/2 changes this dynamic by establishing the Kali Fund. And I hope also that Professor Douglas came up with these ideas on the Kali Fund. And under this multilateral mechanism, large commercial entities benefiting from DSI must contribute a portion of their revenue or profits, indicative rate of 0.1% of revenue or 1% of profit to a global fund. So, crucially, for the scientific community, page 4 of this decision outlines strict standards for data governance. Database operators must ensure that open access while respecting data integrity. So they are now required to request information on the country of origin and metadata, including traditional knowledge indicators. So crucially, researchers must integrate the findable, accessible, interoperable, and reusable principles with the care or collective benefit, authority to control, responsibility, and ethics. And also trust principles. So open science can no longer be decoupled from ethical accountability. That is why we need to co-produce knowledge and institutional power. And to successfully bridge these words, science must move away from an extractive relationship towards the co-production of knowledge. And Decision 16/4 establishes a program of work to 2030 that mandates traditional knowledge be given the same respect and consideration as modern science data. And task 4.7 of this program of work explicitly calls on scientists and indigenous communities to co-produce knowledge necessary for climate resilience and also for conservation under rapid environmental changes. And this means genomic projects in regions like the Africa and DRC should be designed with communities from inception, utilizing biocultural protocols. Furthermore, Decision 16/5 elevates this from a voluntary gesture or to an institutional mandate by establishing a permanent subsidiary body on Article 8J. And IPLCs now have a formal permanent seat at the United Nations decision-making and table and to advise on all matters regarding biodiversity and genetic resources. And this contextual focus, the GRC and beyond, we want to say that what does this mean practically for biogenomics in the DRC? The DRC holds unparalleled genetic diversity that could unlock solutions for medicine, agriculture, and climate adaptation. But under the new framework, accessing this data requires strict adherence to free, prior, and informed consent. So according to Decision 16/2 on DSI, at least half of global Cali Fund disbursement must directly support the self-identified needs of indigenous peoples and local communities. And if a pharmaceutical or agricultural company develop a product using DSI derived from a Congolese plant, for example, those funds flow back into local community-driven conservation and development projects. So for international and local scientists working in the DRC, this necessitates a shift in methodology. Funding applications and research designs must explicitly include local partnerships, respect traditional land tenure, and ensure that digital uploads preserve the ethical metadata and resource communities. That is why we want to tell scientists that our responsibility is twofold. First, we must update our laboratory and data sharing protocols. When we upload genetic sequences, genomic sequences to public database, we must proactively document and direct the country of origin and traditional knowledge linkages. Secondly, public research institutions and academic entities are explicitly exempted from making monetary contributions to the Cali Fund to protect basic science. However, we bear an immense ethical obligation to ensure our commercial spin-offs and also industry partners comply fully with these benefit sharing structures. And we must actively and collectively champion the development of funding to local institutions, such as national biodiversity funds or direct community allocations to build local sequencing and also to bioinformatics capacity within biodiverse nations like the DRC, Kenya, and so on. So equity means enabling the global south to sequence its own biodiversity, and this is currently a challenge. So true scientific progress is not just measured by the depth of our sequencing database, but also by the integrity of our relationships. In conclusion, I will say that by bridging the molecular precision and biogenomics with the holistic generational wisdom of indigenous peoples and local communities and also traditional knowledge, we do not restrict science, but we enrich it. So we must empower the true guardians of our planet, ensuring that the genomic discoveries of tomorrow are built on a fundamental of equity and also respect and shared perspective. So we need to know that we need to bridge indigenous peoples and the local community knowledge with scientific knowledge and science and based methods so that we can come up with a common understanding of our results and to also allow us to empower these guardians and also to advance with our local community knowledge. Thank you very much.
Mr. Daniel, I hope you've had the applause here. Very, very profound statements that you have made, that we must enrich science, not restrict it, and that equity means enabling Global South, enabling the Global South to sequence its own biodiversity. That resonates with African BioGenome, because that's what we're trying to do. That's what we are actually doing, building infrastructure on the continent so that sequencing can happen on the continent. Thank you very much. Stay on course. We have one more presentation. Yes. Marie, you can guide us on the next steps. So the next speaker is Mr. Temesgen Tigab from Ethiopian Biodiversity Institute, who specializes in aquatic ecosystem management as well as ecological systematic zoology. His core work focuses on documenting traditional indigenous knowledge and advancing genetic resource access and benefit sharing research, operating at the intersection of biodiversity conservation, traditional rights, and governance. Temesgen works to ensure that the local communities and indigenous knowledge holders are equitably recognized and benefited in biodiversity conservation and policy implementation. Mr. Tigab will be giving us a presentation on indigenous knowledge associated with genetic resources, the implications for access and benefit sharing. Mr. Tigab, we can see you. Please unmute yourself. And if you have a presentation, you can upload it. And we're ready and waiting for you. Thank you.
Okay, thank you. I'll share my slide.
So we are seeing your screen, a Zoom page. Yes, now make them into presentation mode and then proceed. Thank you.
Okay, good morning, distinguished guests, researchers, policymakers, and participants. I would like to thank the organizers of the African BioGenome Project for facilitating such kind of biodiversity workshop and giving me the opportunity to present. My title, Indigenous Knowledge Associated with Genetic Resource, that implication for access and benefit sharing.
So Tigab, we can see your slides. Are you able to make them into presentation mode?
Now,
perfect.
see? Okay.
Yes. Yes.
Okay. The central message for this presentation is, you know, that African biological resource and indigenous knowledge are closely connected. As genomics and digital sequence information create new opportunities for research innovation, and we also need appropriate mechanisms to recognize knowledge holders and ensure the fair and equitable benefit sharing mechanisms. You see, the knowledge and genetic resource, you know, genetic resource is a biological resource that contains functional unit of heredity that has an actual or potential value. This genetic resource is increasingly important for medicine, agriculture, conservation, food security, and also biotechnology and other sectors. However, these genetic resources are not exist independently from peoples and also their knowledges. So the local communities have an interactions with biodiversity for many generations. This interaction have developed knowledge regarding for biodiversity, for plants, for microorganisms, and also for animals and for ecosystem. So this knowledge can help researchers to identify the biological resource with potential scientific or commercial value. Therefore, we discussed two genetic resources. We should also consider the knowledge associated with the genetic resource. So indigenous knowledge and genetic resource are now a biocultural relationship. So the local communities have the knowledge for medicinal plants, the knowledge for ethnomaternal knowledge, also ecological and ecosystem knowledges. The important question from here is when a researcher or commercial benefits arise from a resource and associated knowledge, how can those benefits to be shared fairly? This is a central issue of access and benefit sharing. Why this indigenous knowledge matters? Indigenous knowledge very important. Which species or biological sources will may be useful? Which parts, when, how, and for what purpose will be used? Which leads to deserve the scientific attention. So the communities or the indigenous knowledge should not be treated as a source of information, but it is a knowledge holder and a partner. So when it comes to access and benefit sharing, access is a mechanism to ensure that benefits arising from the utilization of genetic resources are shared fairly and equitably. ABS is one of the three pillars of under appropriate conditions, and the benefits arising from the utilization of genetic resources should be shared fairly. So these benefits are not financial compensation. It may be research collaboration, it may be training, it may be employment opportunity, it may be conservation and community development, training, capacity building. Therefore, ABS should not be viewed simply as a financial compensation mechanism. It is also about fairness, participation, recognition, and sustainable development. So indigenous knowledge associated with the genetic resource is the interest and the rights of the knowledge holders need to be considered. So under the Nagoya Protocol and traditional knowledge, the Nagoya Protocol provides an important framework or international framework for fair and equitable benefits arising from the utilization of genetic resource. Importantly, addresses traditional knowledge with the genetic resource. This is very important because traditional knowledge often collectively held and transmitted across generations. Therefore, when a researcher or other want to access such knowledge, several questions need to be considered. Who are the knowledge holders? Who has the authority to provide the consent? What conditions govern access? What benefits should be returned to the community? What customary rules apply? This question demonstrates that ABS is not simply a technical or legal process. It also a process of involving people, communities, culture, rights, and relationships with biodiversity. When it comes to ABS under Ethiopian conditions, the close relationship between biological diversity and indigenous knowledge. the country considerable biological diversity and diverse indigenous knowledge systems. Communities have developed knowledge related to medicinal plants, wild foods, livestock healthy, agriculture, wildlife and ecosystem management. And also they have veterinary knowledge. So they have a relationship of high relationship in biological and indigenous knowledge. However, documentation and research should be undertaken ethically. Communities should not simply be treated as a source of information. They should be recognized as a knowledge holder and partners. This is where my institution is Ethiopian Biodiversity Institute can play an important role in biodiversity conservation, documentation, and ABS implementation. When come to these genomic changes, the ABS landscapes now, I would move one of the most important emerging issue. The modern genomic technologies allow us to generate enormous amount of DNA sequence information. Once the genetic information or the biological sample was sequenced and stored, analyzed, and shared electronically across countries and institutions. This creates opportunities for research, biodiversity conservation, agriculture, livestock improvement, healthy, and biotechnology, but creates new ABS questions about traceability, about attribution, and beneficiaring. Therefore, Africa enters the genomic era. ABS frameworks need to be considered both the physical genetic resource and increasingly important role of the digital genetic information. Where are the pressure points or the key challenges of connected with the genetic resource and indigenous knowledge? The one is loss of knowledge. That means loss of indigenous knowledge, because indigenous knowledge are transmitted orally, and when the elders pass away or younger generations no longer practice traditional knowledge systems. So the second challenge is inadequate documentation. Documentation must be undertaken carefully, because poorly protected documentation can create risk of unauthorized access and use. Another challenge is identifying the appropriate knowledge holders and representatives. There are also challenges to limited legal, technical, financial capacity to negotiate ABS agreements. These challenges demonstrate need for a strong institutional community participation, appropriate governance mechanisms. The pathway from knowledge to equitable beneficiaring. Beneficiaring should be considered from the beginning, not added at the end. Think a pathway, because there is a community knowledge, the ethical documentation, then you must get the PIC, pre-informed consent, and mutually agreed terms together with the appropriate national requirements. Finally, benefits should be fairly and should be contributed to community for conservation and sustainable utilization. The important point here is benefit sharing should not be considered only as the end of the research process. It should be from the beginning.
As you wind up, take up.
Yes. Okay, let's make this practical. Imagine a community helps identify a high-value genetic resource who participate, the knowledge holder, institutions and researchers, what is agreed, access, the consent, the conditions and the benefit pathway, what is tracked. So which of these currently is the biggest gap in our context? The message to take home, indigenous knowledge is not just a data, it is a part of biocultural system, linking biodiversity, people, and ecosystems. My presentation or conclusion, our success should not be measured only by how much genetic data we generate or how many scientific publications we produce. We should also ask, are communities participating meaningfully? Is their knowledge respected and protected? Are benefits shared fairly? And are biodiversity indigenous knowledge conserved for future generations? Therefore, our goal should be very simple. From knowledge, from biodiversity to knowledge, from knowledge to innovation, and from innovation to equitable benefits. Thank you for your attention.
Thank you very, very much. I hope you can hear the applause we have given you. And indeed, a resounding message that has been given by almost all the presenters this morning is that the contributions of the local communities should not be considered at the end, but should also be incorporated in the beginning. I'd like to now turn over the session to Marie Salvatrice. Please moderate this session. We have about 15 minutes.
Thank you, Anne. Thank you for this opportunity. I think as we have heard Daniel and also Timesa, I would like to ask you a few questions, and then the audience will ask also questions. For Daniel, we have seen that there are many people in the room, or young people in the room. What can you tell your young self when it comes to this conversation about the DSI and the traditional knowledge? And which message can you give to them? Thank you.
Daniel, what can you tell the young people in the room?
Thank you very
And
much.
your young self.
And
And
thank
your
you
young
for
self.
this interesting question. Yes, of course, we must know that we are young researchers, and also we are interested in these new areas of the research, and also we need to understand how to value our own natural resources, and also biological resources and genetic resources. We need to find common understanding and to define path that will allow community methods and also scientific methods to come up together with common knowledge, common understanding, to bridge the gap between science and local knowledge, and to enrich our knowledge, and also to develop ourselves. They will perhaps show us that our indigenous and local knowledge are not up to the task, but we must value ourselves, our knowledge, our culture, our natural resources, and also our behavior, so that Africa can be strong and Africa can define its own way of development. Thank you.
Thank you so much. I think here you are talking about collaboration, about value what we have. We can't build on nothing. You have to start by appreciating and value what we have, especially our traditional knowledge. And coming back to the professor who presented on traditional knowledge, I would ask you a question from the discussion with Daniel. How can you motivate people, motivate young people? I'm coming back to the young people because they are researchers here, they are future generation here. What can you do to motivate motivate young people, young researchers, so that they can bridge the gap showed in identifying traditional knowledge, but also prioritize what is there, what is good, but also making something sustainable using our resources, including our culture, sustainable. Thank you.
Is that for Daniel?
No, it's for the Tega.
Yes.
I am failing your name, sorry.
Yes. Maybe Professor Hewan can answer that. I see you've raised your hand, and
it's
Prof.
Hewan. Yeah.
Ethiopia, just go ahead and answer the question. What can we do for the young people? There are very many young people online. Here in the room, we have about 100 of them. What can we do for the young people who are, coming up in this particular area? And maybe you can now then transit into your comment or question. Please unmute Hewan.
Okay. Thank you very much for this thought-provoking presentation, and it's really interesting, definitely. So just to comment what Professor Anne asked me to comment about, how about this indigenous knowledge, how we can make it more useful to the youth. We have to make it commercial. Any science, any publication, any theory, or any knowledge that we have, if we are just talking about indigenous knowledge, but it's not changed into economic value, it's not going to be promoted or being functional, and specifically for our youth. I just wanted to give one example. I learned in Japan. I did my master's and PhD in Japan, and I stayed more than 7 years in that. And still I have a lot of connection with them. And always, I really admire Japanese system. Why? Because the Japanese system is created by the Japanese, and it is really benefiting them. And now they are harvesting millions of dollars every year because of their indigenous knowledge. So as you know, the Japanese culture is fermentation-based food. Their food system is fermentation-based. The famous miso, the plant-based from soybean, and the koji and those fungi, they have isolated it in their own country. They isolated it, patented it, and every year they are harvesting $1 million because of that. So they are not outsourcing that knowledge. There is indigenous institution which is based to do innovation on their indigenous knowledge. So sometimes as an Ethiopian, when I think about my Ethiopian culture, we are indigenous, but we prefer to outsource our knowledge outside. No, we didn't want to create that knowledge here. For example, now I am in the southern part of Ethiopia, and always I eat indigenous food. Instead, one of the false banana, which is green, it's so for our eyes, it is really a pleasure to see, and it makes our garden beautiful. And it is a false banana, and it is like a hunger food. So when the other part of Ethiopia has been in hunger, but in the southern part, it is really green and there is food. But we overlook that. We overlook that knowledge. The insect grows, propagation, and everything, it is indigenous. I will always get surprised. It's much better than the tissue culture technology that we can develop sucker from one insect and we multiply it to thousands. And the fermentation process by itself.
Yes, sum up so that we can receive other questions.
Yes. Yeah, I will finalize it. So then the fermentation by itself, there is a unique type of bacteria which is really involving. and we don't have a starter culture or something. So then can you imagine if the youth are really involved in such kind of technology development and building the infrastructure for insect fermentation and also isolation of the bacteria or the fungi, and then making it a business into the country system. If it's just for the southern part of Ethiopia, we have like more than 20 million people. So that is a huge revenue, but we are overlooking that. So that
Thank you
is
so much.
my point.
Yeah.
Make it business, and it will be
Thank
changing.
you so much. I think she has raised a good issue. When there is no money, people don't hear. But when there is money, people can hear. You want to hear from young people. We are talking about young people. One woman and also one young person. How do you value, build, strengthen our traditional system in Africa so that we can make it economically viable?
So we have a young man here, and please, ladies, don't let us down. Ask a question.
Yeah, I quite resonate with this panel because one of the or part of my PhD project was focusing on the wild crops, the sweet potato wild crops, and I realized that there is we are missing one thing that I used to consider as one of the indigenous knowledge, and nowadays it's called taxonomy. So you find that currently the young generation, when they go to school, they don't see taxonomy as that good course anymore. But taxonomy is one of the key indigenous courses, I will say, that is there, that has been passed from generation to generation. From the way people used to live back in the years, centuries back, passing that generation with specific plant species, how they are being used, how they are important to what we have right now. My question to the panel, and specifically to Silver Trees, what policies do you think we can put to encourage the young scholars that bioinformatics is not just the only scientific, field to go, or molecular biology or genetics? Which policies can we put to encourage the young scholars, the young scientists, that indeed taxonomy, basic taxonomy, botany is still valuable to us, especially here in Africa, because we cry we don't have food, yet there is food in the wild and people are consuming this food? That's my question.
So we'll have a young lady and another young lady.
Good morning, everyone. Thank you for this opportunity and thank you for the amazing presentations that we've had. So my question is so general. What is the call to action to us as young scientists and young scientists, both men and women? I'm an undergraduate looking forward to further into master's and PhD. What is the call to action to us as young scientists in Africa, in Kenya, and everywhere? Thank you.
Thank you. And then hand over the mic.
Thank you so much to all our panelists. I'm a natural products scientist and I really resonate with all that you've shared with us. Kindly, I quite struggle when it comes to the fair and care principles when it comes to the control of sensitive cultural information. What level of access should we actually advocate for? Should sensitive cultural information be declared open to all, or what exactly should we have as the levels of control?
Okay, over to the...
I think, Prof, I can just add one more question so that we can have two. Yeah, and this is for Hewan. Do you think there is misinformation or myth regarding some of this traditional food that we have. Because a case example, I heard somebody say nowadays she can't eat avocado because growing up she used to eat avocado when they were broke. You know, they can't eat sweet potato because growing up that food was meant, you know, during dry season when they don't have money. But that's what we have as our indigenous food and natural food. Do you think there was that misconception about this quote unquote hunger food that is still affecting us even today when people are dying of hunger? Yet when you walk outside their compound, there is sweet potato growing there, there is wild yam growing there, there is other indigenous vegetables that are going there. And even in Ethiopia, you're saying up north, people are dying of hunger, but down south, Ensete is growing and it's like a weed and it's food, but people are not utilizing them. So please address that also. Thank you.
So over to the panel, Marie. Direct the questions to.
I think some of the questions when it comes to negotiation, I will give the hands to Daniel. What I can say, one of the things the young person asked about what policy can work. I can't say what policy can work. I can say how to influence policy. Because you can make a policy, the policy is there, you can make a research, and also the policy, maybe you advocate for the policy, but make sure that you make a space in these international dialogues. Those young people, there is they are a network of young people, but you can make your African network and make it work. And also for scientists, I can say it's the same. Have that commitment. Have that commitment as young people. Don't think that you have seen, now people, they want to do what Anne is doing. She has been doing a process. I always tell young people, respect the process, start now and make your own way. This is where you are going to find the time to sit in those high-level seats. You are going to be negotiator like Daniel. And for the fair and sensitive information, I think we, as CBD Women's Caucus, I am a member of CBD Women's Caucus, we represent the local community who has been advocating for that. How to ensure that local community are integrated, are involved, but also they sign the free consent on the publication, also the knowledge they share, so that they can be aware of what they are sharing and ownership by the community. And I can hand it to Daniel also to add negotiation about policy and negotiation. Thank you, Daniel, up to you.
Okay, thank you very much. Yes, I think that there are interesting and relevant questions, but I will focus on call for action. And because I think that this is what is very important for young scientists, especially. And I think that to harness the full power of biogenomics in Africa, young scientists must bridge the gap between cutting-edge laboratory science and generations of indigenous knowledge. And this is how, for example, young scientists must own the African genome. So we must, first of all, dehumanize colonial data dependency. That you will recall, for example, that Africa contains the highest genetic diversity on Earth, yet represents less than 3% of global genomic data. And that is why we must also, as young scientists, try our best to sequence locally. It means that we must lead the effort to sequence local human, plant, and also animal genomes of African soil rather than shipping samples abroad, because our knowledge is going very far from us and it does not benefit to us, and we must go against that. So a young scientist must also document and validate local knowledge, as I said before. We must also keep partnership with elders, mentorship. It means that we must treat traditional healers and community elders as co-researchers who hold the blueprints of biodiversity. And also we must validate ethnomedicine in Africa, use biogenomics to isolate, test, and also scientifically validate the active compounds in traditional African medicine. This is a wealth we have, and we must exploit it and explore it in deep so that we can be able to value our resources and also to keep them. So we must be interested in this knowledge and in this process. I started, for example, negotiating while I was very young, and I was learning from elders, from other negotiators from Africa and or from elsewhere. And currently, I can be proud of the progress I am making. So young people and young scientists must not be afraid to be part of the dialogue and the process, to ask questions and to be interested and to document and also to innovate. Don't be afraid of new things, of new technology, of new thematics, of new era of research. You must be courage enough so that you can be able to be part of these changes. Thank you.
Hewan, do you have a comment, a quick comment, and then maybe Tigab, a quick comment?
Thank you very much, Prof. Yeah, my comment is negotiation. Let's be Africa. Our soul is here. We are born. It's like there is really a link. Even now, the science is telling us a lot. The epigenetic factor. We inherited all that from our great ancestors. So we are from Africa, and then let's be a real Africa, and let's try to understand our continent. How many of us really we are interested? One day I have been asked if you have been given a chance to be the president of African Union, and they asked me, what will you do? And I said, I'll build train. Every continent, if they are connected by train, then we can share and learn from each other. I think it is easier. So let's really connect it. Let's read from our continent, and let's have our database, not only for genome, but also for our publication. I don't want to export it to Scopus Indexed or Web of Science Indexed. Let's be here, African Indexed publication, African database, that I can access it and it can benefit the continent, and the knowledge will be generated and it can be shared easily. That's my message. Thank you.
Thank you very much. Tigab, your final comment? Yeah, it looks like Tigab is offline. Maria, hand it over to close the session. I'm sorry we are not able to take any more questions. Please type your question in the chat. We will make sure that the relevant person is able to see it, and we can be able to get back to you with the answer. So Marie, over to you, your final comments on this session.
Thank you so much. I think this is really a good session. I think one of the things I can say togetherness, how Africans can work together. This is very important, because sometimes we find we are looking for solutions, but sometimes we fragmented our effort and we don't come together as Africans. I can say that this has really affected us. Let's work together. Let's combine effort, and also let's value our resources, especially our culture. Thank you.
Thank you very, very much. A big round of applause for this panel. It was truly an East African panel, and thank you so much. We had representation from Rwanda, we had Daniel from DRC, we had Professor Hewan from Ethiopia, and also Mr. Tigab from Ethiopia, and all of us here in the room and all over as we are presenting online. So thank you. Another round of applause for this panel. We thank you so much, and we thank you for your contribution. And we could have gone on because the interest here in the room, and I'm sure virtually, is really grasped. And thank you for making it very, very interesting and very, very down to earth. Thank you once again. So we are taking a 30 minutes tea break. So for the online participants, let us meet here at 10 minutes to the hour. 10 minutes to the hour, and we will start the next virtual panel session. Thank you, and enjoy your little break as we proceed. Thank you.
Welcome to AfricaBP East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged one-stop molecular biology shop, and currently we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipments. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped, and work is progressing, as you can see. So we are looking forward to having our clients approve our services, and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples, and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Wilda. We have very quality gel documentation systems. The main core service, we provide this distributorship and sales services. We sell our products across the extent of the Eastern Africa region. all the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our cells are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, amplification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment, we supply equipment, including but not limited to freezers, to fridges, to the safety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans. So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students, our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
PacBio Hi-Fi sequencing technology gives researchers a comprehensive, multi-omic view of the genome, including GC-rich regions and long repetitive sequences that other technologies can't fully resolve. Here's how Hi-Fi works. Hi-Fi sequencing is built on single-molecule, real-time technology. Its accuracy comes from repeatedly observing the same native DNA molecule as the polymerase replicates it. First, DNA from nearly any organism is converted into a circular template by ligating adapters to both ends without PCR amplification. This avoids bias in highly repetitive or GC-rich regions and allows for the simultaneous detection of genetic variants and methylation status in a single assay. Next, primer and polymerase are added to the library, and the sample is ready for sequencing. Sequencing happens on a SMRT cell, which contains millions of tiny wells. Inside each well, the polymerase gets to work, building a new DNA strand base by base from the native template. As each base, A, C, G, and T, is added, a unique pulse of light is emitted and recorded in real time. These pulses create a continuous record of the molecule as it's read again and again. Hi-Fi also captures epigenetic information automatically. When a base is modified or methylated, subtle shifts in the kinetic signature of the polymerase's incorporation rate reveal rich epigenetic insights like 5-MC and 6-MA methylation. This happens automatically on every sequencing run without additional library prep. Hi-Fi reads are generated by combining information from multiple passes around a single DNA molecule. The system combines these repeated observations to produce long, highly precise reads, exceeding 99.9% accuracy. Powerful on-instrument algorithms and analysis tools turn this raw data into rich, multi-omic insights, revealing the genome, including complex structural variants and dark regions, the transcriptome with full-length isoforms, fusions, and splicing, and the epigenome with base-level methylation patterns captured natively. All in one run, all from the same molecule. With Hi-Fi long reads, researchers can do everything from interrogating the hardest regions in inherited disorders to powering population-scale precision medicine with complete, accurate genomic and epigenomic data. Hi-Fi sequencing removes blind spots and delivers the accuracy needed to understand biology at every level.
science is not just advancing, it's accelerating. From the front lines of public health to the frontier of innovation, a silent revolution is unfolding, and at its heart is a new way of seeing life itself. Next generation sequencing. Disease doesn't wait, it doesn't discriminate. But what if you could see it coming before it strikes? In Kenya, the burden of infectious diseases, antimicrobial resistance, undiagnosed conditions, and now a sharp rise in cancers, tuberculosis, and reproductive and genetic health disorders calls for sharper, faster, more precise tools. Beyond hospitals and research institutions, the same technology has the power to transform how we grow food and protect our environment, improving agricultural productivity while preserving and leveraging our rich ecological biodiversity.
The MGI-Tech 100 is a pretty simple sequencer to work with because we did not have a lot of hands-on time.
And so the uptake is really good and the future is looking bright.
With the new MGI-Tech 100, there are some steps that have been optimized or removed so that those steps take place inside the instrument. So as a user, you don't need to do them manually.
MGI-Tech 100, we're able to get the results within 15 hours.
At the Aga Khan University Hospital, we are currently using the Illumina next generation sequencing for a number of key clinical applications. Specific examples are for patients with breast cancer, ovarian cancer, prostate cancer, and pancreatic cancers. The MGI-Tech I-100, which is a new instrument, is promising a lot. Compared to the MGI-Tech 100, which I use, the MGI-Tech I-100 is promising, for example, a jump from 1.2 gigabases of data to 30 gigabases of data. From 4 million reads to 100 million reads. From 150 base pair read lengths to 300 base pair read lengths. Among other advantages, I can still use the MGI-Tech I-100 using a smaller flow cell or a bigger flow cell. And so depending on the number of samples that I have, I don't have to worry about efficiency of utilizing my sequencing cartridge. And that's a key point.
The introduction of the MGI-Tech, the I-100 Plus, is really a game changer in terms of the work that we are doing. It's very clear that this MGI-Tech will reduce the amount of time we are using to do our sequencing from the old one that we used to do about 58 hours to now 15 hours maximum. You can get your data to as soon as 4 hours. So it means our productivity will definitely go high, and the dangers and encumbrances that were associated with the storage of cartridges, the reagents, have been eliminated because now we don't have to do cold chain storage, we can do over-the-shelf storage. So this would really impact on our productivity and the value of science that we're going to be demonstrating.
MGI-Tech I-100 is really at the heart of that transformation.
Our live trials with the MGI-Tech I-100 series had a very positive feedback. Most customers requested us to leave the system behind. This demonstrates the positive impact it has already made. And I believe this is unfolding a new era for our scientists in Kenya.
Illumina's latest innovation, the MGI-Tech I-100 series, couldn't come at a better time. The faster turnaround time, the ability to sequence more on this compact platform, and by leveraging the same X-Leap chemistry as the bigger platforms, it's definitely offering a number of game-changing features that our markets, our labs will definitely benefit from.
us to a new era in science. In a world where time, accuracy, and precision define outcomes, the future of genomic research has landed in Kenya. The MiSeq i100 Plus, compact, fast, and powerful, designed for labs that demand precision, yet operate on tighter scales. Whether you're exploring infectious diseases, cancer mutations, or the human microbiome, next generation sequencing simplified from sample to insight, faster, deeper, and clearer than ever before. And now it's here.
Hi, my name is Peter Miner, co-founder of Project Mocha. Now, in the last 10 years, I've worked in Kenya's agri-tech space, and I've had many conversations with farmers. And 4 things have always stood out. They lack access to finance, access to markets, access to quality inputs, and access to qualified inputs. At Project Mocha, we are turning coffee consumers to become coffee investors. Now, think about it this way. Instead of putting your money in a money market fund, and it's growing at 10%, inflation is growing at 10%, you can put that money into a commodity like coffee, which is ideally deflationary, and you're actually investing into a coffee farm. Now, on the farmer side, what you do is we realize their assets on chain, and the assets they're looking at are their coffee trees. As opposed to traditional finance, we always look at the land as collateral. By bringing these assets on chain, the farmers have a balance sheet on a global ledger, and they can get investment through.
Hi, I'm Paul Gashora, founder, CEO of Project Mocha. To me, I see Project Mocha as a vehicle that helps smallholder coffee farmers unlock capital to reinvest in their farms and improve their productivity. We are at the revolution, we are at the beginning of a new way to finance production. And so we as the retailers, we as coffee consumers, we get to participate in the coffee production, and we also get to participate in the returns from the sales of our coffee. This is nothing new. We are looking at putting money in investable opportunities to make a return. What we've done is made this process simple, transparent, and you can do this on your phone. That's what I love most, is we're simplifying the investment process and making it accessible to retail consumers, to retail investors.
My name is Ombogu, and happy to be here. So my association with Project Mocha goes back around last year, September, over there, when I met the core team, Peter and Paul. And they showed me about what they were doing. For the first time, you know, they brought this concept that you can tokenize literally any single asset that you can say isn't like a piece of land, right? It's like you can consider anything an asset, and coffee trees is, you know, like it's a very interesting asset because it's always productive. A single coffee tree is like a source of income, and you know the fact that that was being neglected by traditional finance was a very, very big gap in the market. The other thing is the team, the dedication, the passion they have, that they carry, just the overall dedication, like from the team, from their partners, the overall support from the community that is just sort of there. I mean, it's just amazing.
My most memorable experience, how would I say it? I will give it the first time I knew about the project. It was so unique from the angle of how they were being able to just bridge the Web3 aspect and the Web2 aspect, and also getting to work together because of their unique aspect. That I will say that is the most memorable experience with me.
The most memorable experience with Project Mocha is during last year's e-Safari pilot, which was the very first pilot that Swiped and Project Mocha did together. So, and really saw a direct, rather a very direct way in which you can actually impact the industry by adding blockchain as a layer to making it work for our farmers. So it's really made the whole industry see the vision behind Project Mocha. And this collaboration with Swiped, I think really make a difference, not just in Kenya, but also globally, especially as now we kick off with coffee. And I believe in this particular project because it touches back home as a Kenyan and also as a person coming from the farming space.
This stuff is really good, I mean, obviously.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged your one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipments. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed
to our extraction room. This is where we do all our extractions.
Good morning once again, and welcome back from the break. We are here on day 2 of the East and Central Africa Regional Workshop. Today, the conversation is about access and benefit sharing, DSI, traditional knowledge, local communities, as we are holding the first African Congress on DSI and ABS, and all these conversations we'll be having. So welcome back after the break. We are now moving into our third session, which is a virtual panel that will be talking about biobanking in Africa towards ethical stewardship of genetic resources. So this is a panel session, and the panel members that will be speaking to us include Dr. Laura Betola, who is a scientist at the National Center for Biological Sciences in Bangalore, India, streaming all the way from there. Dr. Laura has been a panel member, a speaker, a moderator in the East and Central Africa BioGenome Project since its inception. And we're really, really grateful to see you for the 4th year running, and you're most welcome to join us next year too. Dr. Laura is interested in how the rapidly developing field of genomics can contribute towards the conservation of biodiversity. During her PhD at Leiden University in the Netherlands, she applied various genetic techniques to gain insights into the distribution of genetic diversity in lions. Afterwards, through her affiliation with City College, New York, and Copenhagen University, Denmark, she has worked on fundamental science, scientific questions, as well as applied research for a range of species, primarily African species, Asian species, and South American species. She is particularly interested in how she can leverage genomic data for informing the management and policy governance, for tools for research, monitoring, and forensics. Through various NGOs such as Leo Foundation, she supports the implementation of such tools, capacity building, and local decision making. And indeed, she has been a member of African BioGenome Project, and therefore capacity building has been something she has helped us to do. Our second panel speaker is Dr. Anubab Khan, who completed his PhD from NCBS in Bangalore and conducted a postdoc research at NCBS, Penn State, the USA, and University of Glasgow, and the last postdoc was in University of Copenhagen. Anubab has worked on a wide range of topics in evolutionary biology, ranging from evolution of Indian elephant populations to the evolution of cooking behavior in humans. He's mainly interested in understanding small, isolated populations exemplified by endangered species. He has done research on tigers, elephants, rhinos, leopards, jungle cats, does, and horses. He has extensive experience in using next-generation sequencing data to understand threats to endangered populations and how such populations evolved, and has developed and optimized methods for sequencing whole genomes of elephants, from tsetse flies to sequencing whole genomes of cats from feces, and generated resources like chromosome assemblies of tigers, ancestry informative marker panels for resource forensics and species conservation. Dr. Anubab has been actively involved in capacity building for genomics in India, Indonesia, Kenya, Rwanda, and many other countries. And he plans to organize many workshops and generate online resources to develop expertise using big data for wildlife research and management. Welcome, Dr. Anubab, and we are very, very grateful. Dr. Anubab has traveled all the way from India. He's currently on his way to Pwani, Kilifi, which is on the 5 hours drive from Nairobi, to support the practical that will be conducted for the next 3 days. And we value your presence at this this workshop, and we know that this is your second African BioGenome Project presence, and we thank you so much. Our next speaker is Dr. Francis Gakuya, who's a senior principal scientist at the Wildlife Research Training Institute in Kenya. He's a wildlife veterinarian, epidemiologist, and a research scientist over 30 years experience in research, clinical practice, capture, translocations, and captive wildlife management. He was formerly the chief vet officer at the Kenya Wildlife Service. He holds a Bachelor of Veterinary Medicine, a Master of Science PhD in Veterinary Epidemiology from the University of Nairobi. His research area of interest is zoonotic, animal diseases, human-wildlife conflict, disease ecology, One Health, and wildlife genetics. He has contributed to knowledge, science, technology through publication of 84 peer-reviewed journal papers and 5 book chapters. So thank you very much, Dr. Gakuya. It's also not your first time being with us. This is your second time supporting us in the capacity building at the African BioGenome Project, having been a key participant and panel member in our workshop in 2024, I think it was, the one at Jomo Kenyatta. So, dear panel members, dear participants present virtually and online, this is a panel session, and we've spent the whole morning discussing key questions of access and benefit sharing. We've heard about storage of the importance of storage of DSI in local institutions. We've heard about the importance of making sure that our sequences are tagged so that we can be able to get back. But now we're talking about biobanking, which is one of the issues that has been discussed in access and benefit sharing. And my first question will go to Dr. Francis. Kenya is developing the infrastructure for establishing a wildlife biobank. And Dr. Mumo, your colleague, was the first speaker in our morning session on Africa on ABS, and she did mention that WRTI, the Wildlife Research and Training Institute, has gone very, very far in its preparations towards the establishment of a local biobank, which is specifically, and she mentioned that this one is a biobank for wildlife species. Could you tell us what is a wildlife biobank? And why is Kenya investing millions of dollars in this establishment? And is it really worth it? Is it necessary? Especially when we think about institutions like the Kenya Medical Research Institute, that is probably developing a biobank for pathogens and other disease microorganisms and organisms. Unmute yourself, Dr. Gakuya.
Okay, can you hear me?
We can hear you, but the volume is low. Maybe you can add a bit.
Okay, now am I audible?
Yes, it's very loud and very clear. Thank you. Proceed.
Okay, thank you, Professor Anne, for that. Yes, Kenya, we are in the process of establishing our wildlife biobank. As most of you are aware, genetic loss or loss of wildlife species is quite rampant, and even in our country, it's not left behind. There are several wildlife species where the loss of biodiversity is quite high. So in actual fact, why we want to do this is as an insurance, specifically for the genetic loss that we have had. If you look across the country, several species are either critically endangered or endangered, and some of them are rare and endemic. And so a good example is Hilola antelope, the most endangered antelope species in the world. It's only endemic in Kenya, less than 500 individuals left. So if we don't biobank samples that can help us, especially when it comes to issues of reproductive biology, as well as other important areas of research, then we can lose that population. Another population is Grevy's zimbra, which are only again found in northern Kenya, southern Ethiopia, less than 3,000 left in the wild. Again, we have to insure them. And even the northern white rhinos that were in Central Africa, we have only 2 left. They are in Kenya. So again, we require some of these genetic resources to be banked for the future. If you look at the setup in the country, we really don't have any wildlife biobank. I know we have several for livestock, but for wildlife, we don't have. Wildlife Research and Training Institute is mandated to undertake research and also coordinate research in the country. And one of the important tools for this research is a biobank to assist in various issues. So that is there is a deliberate effort for us to establish that important biobank for the future of biodiversity, for research, and also the access benefits that we talked about. Yeah, I'll stop there, Prof, for now.
Yeah, true. And I have seen the plans for the biobank, having interacted with you and the leadership of your institution. And it's not just a biobank, it's a whole building with a relevant infrastructure. You know, you have cold rooms, freezer rooms, preparation rooms, and the whole chain. And it goes with it that there's a lot of capacity building, yes. But I'm still asking the question, why do we need to establish this biobank, especially knowing that it's going to be mainly funded using donor funding? And we have seen, this is not the first biobank in Africa. There are several biobanks in very, very key institutions across Africa. And with the recent budget cuts that we have seen worldwide, the recent reduction of donor funding, what is the sustainability plan? Because you see, if this is going to be funded from donor funding, and we don't have the money maybe in our national budget to maintain it, how will we be able to sustain it? Or maybe should I ask the question a bit differently? What is the WRTI going to do differently to ensure sustainability? Please unmute here.
Okay, thank you again. Yes, we are working collaboratively with the various partners to deliver this important project, which really is a milestone for the Institute, which is about 5 years old. And for us, the way we look at it is that the partners, and the major one that we are working with is San Diego Zoo Wildlife Alliance, will really assist us in establishing the facility and also training also component of training. But thereafter, as an institute, we'll come up with a budget. There will be a budget line specifically for the biobank, which will make it sustainable. Because one of the things that we have done is that there will be funds to ensure that that biobank is sustained. And that is the way we are looking at it, in that it will become one of the components of our yearly budgets from the EPIACA, from our internal resources, to ensure that it is sustained. So that is deliberately there. And we have worked on a strategic action plan, and it clearly spells the future sustainability of this important facility in the Institute. Thank you.
Indeed, that's one of the ways of ensuring sustainability, that we don't use only external funding, that we have to use our own funds. After all, this biobank is a resource for us as Kenyans and for the region. And therefore, to ensure sustainability, that the government of Kenya has to invest. And I'm happy to hear that following the seed money, the seed grant that will be given, there are issues and plans for sustainability. But let me turn to Dr. Laura now. And we're talking about biobanking. And we're talking about how this biobank can be used as a resource for the country. When we do that, how can communities meaningfully benefit from this, especially when you consider that these benefits may not come for many years? The biobank that Dr. Kakuya and his colleagues are establishing will continue being established for the next maybe several years. And the communities that have donated their species, their samples there, will see nothing for those number of years. What are your thoughts around this question, and how can we ensure that the communities who are the owners and the custodian of these genetic resources benefit in the long term, yes, but also in the immediate term? We're going to make you a co-host so you can unmute yourself. Try now. Yes.
Thank you very much, and thanks so much for inviting me again. I always look forward to this event. It's very inspiring to hear all these talks and see how AfricaBP is developing. I think the question is a very, very important one. Biobanks by nature are not prescriptive in what can be done with it. The flexibility of having these kind of samples and having these resources for a wide variety of projects, I think, is really, really crucial. So it's difficult to say. There might, of course, be certain priorities that are set by government or by other stakeholders that will be high on the list to use the resources in a biobank for. But biobanks inherently have this flexibility that you can do a lot of different things with them. And I think that's the power and the value of biobanks. But of course, it makes it sometimes more difficult to communicate very clearly what you will do and what the timelines are for seeing some kind of return. One example that I would like to highlight, I've been working with colleagues at KWS and WRTI for a long time. We had several PhD students doing their PhD projects on topics which were of high priority and relevance to WRTI. And so one of the projects we did was on lions in Kenya, and understanding the distribution of genetic variation and the loss of genetic variation and the effects of past conservation interventions like translocations to mitigate conflict. How is that impacting current day lion populations in Kenya? Well, for that, obviously, you would need to have a sampling from all these different populations, right? And it's difficult to set out doing that once you have this idea, and you still have to start collecting samples. If you have a biobank where routinely samples are being submitted to, for instance, from routine interventions, veterinary interventions, or for instance, from seizures, from confiscated material, then you build up this huge wealth which will make follow-up projects much easier, right? So I think that is a case which really kind of illustrated the value of having collections. We used a lot of samples for that particular project, not so much from a biobank, but from KWS headquarters where they stored material. So even though there was not formally a biobank, the fact that this material is collected and documented and stored, I think is really, really important, and then can contribute to all these kinds of projects. In particularly, I would highlight for forensic work. We're now working on a couple of SNP panels, which are hopefully can contribute to forensic sciences and forensic applications. Also here, having these broad baseline data sets and understanding which diversities were where for different species is a prerequisite to have a powerful tool with potential forensic applications, right? It's difficult if you want to trace back a source population. You need to have these baseline data. So I think for informing management decisions when it comes to fencing, when it comes to conflict mitigation through translocations, when it comes to forensics and detecting poaching hotspots or trade routes, all of these kind of topics benefit enormously from having baseline data and having a documented overview of samples from different populations. So I think a biobank, in particular for these kind of topics, can be really, really valuable.
And thank you very much. We'll come back to you, especially on the topic of forensics. But I'd like to turn to Dr. Anubab now. You've had the opportunity to work in the global north, and now you're working in the Institute of Science, Bangalore, India. What are the key differences that you have seen with regard to genomics, bioinformatics, and the biobanking infrastructure in low and middle income countries? Because now you visit Kenya quite often. And what you have experienced as a postdoc, as a scientist working in the global north, especially with regard towards the investments in infrastructure. And also, I'm curious about how come the north is able to rapidly move with the bioinformatic progress and changes in genomics, and we are having challenges keeping up to the speeds of that.
Thanks for that question. I'll go a little slow. I'll break that down into parts, and if you might have to remind me the question. So again, so the first part, the major differences that I observed between the global north and what I observe in the global south, which Kenya and India both are part of, are this. One of the thing is the intentionality and the mentality. So in global north, what has happened, or what I believe has happened, is I have observed this tendency that the problems of this global north are the global problems, is the tendency that is portrayed. So for example, Dr. Francis here presented all of these interesting cases of what are the priorities that are being biobanked, but what we often ignore are things like insects, things like reptiles, which are priority for our countries, right? So for example, biobanking of snakes can be very important. We have a lot of deaths due to snake bites, which we know are recognized by all the wildlife institutions in all our countries, but they are almost always ignored for some reason. But on the other, because they seem fancier and gather more media attention, UNESCO often does a great job of recognizing what are endangered species, but what are the important wildlife species often gets ignored because they might be least concerned. For example, an extremely venomous snake might not be endangered, is the least concerned species, and we may not give it enough attention unless it falls in the medical. This has happened in India as well. This is a major difference that I have been observing. And this needs to be addressed at a local point of genomics and bioinformatics infrastructure. Yes, my Western colleagues feel it is extremely important to do whole.
So Dr. Anubab, maybe if you switch off your video, we will be able to hear you continuously, because you're breaking a bit. Just switch off your camera so that we can maximize on the bandwidth. Sorry for interrupting.
Traveling, so I don't know where I got broke up, but I'll just continue. So one of the major infrastructural challenges has been bioinformatics, right? I'm going to give the example of India, where we have 54 endemic species of mammals. Now, I give the example of mammals because mammals generally receive more attention than invertebrates, reptiles, and other species. 54 mammal species, out of which 19 of them do not even have a regular cytochrome B or COX-1 gene data. So out of these 19 species, you confiscate anything in the customs department, anything for a wildlife crime, they cannot be prosecuted. And this is just cytochrome B or COX-1 data, right? It's supposed to be garden variety data in Europe. You will get it almost everywhere. And still in the West, we will be talking about generating whole genome data sets. Now that's a major challenge. While we are talking about the challenges in developing our servers, our expensive bioinformatics infrastructure, we need to get at the ground level. We need to look at what are the priorities for my country. Where are the current challenges? I need to survey that really well. Second thing, the challenge in developing bioinformatic infrastructure is what is the awareness that I have? How much capacity do I have? In India, at least, and what I observe in Kenya also is like there are hotspots. There are like pockets where some university researchers are able to access and understand this kind of data set. Majority of my community, majority of my peers are not able to make sense of what is being talked about in genomics. So first, I need to build this capacity also, where an appetite is built for such data set to be utilized. And then I can go ahead and say, okay, now I need an expensive server so that all my peers and colleagues can keep up with the data sets that are being discussed. So I see a great potential in what Professor Dr. Francis has been talking about. It definitely is a very important step. For example, the questions you asked Laura, and Laura rightly pointed out, the uses to the community. I think the biggest benefit is visible when we look at the endangered species like gorillas and chimps. Billions and trillions of dollars that the pharmaceutical industry has made is by studying these endangered species, right? And a lot of it the cell lines that we are talking about will come from biobanking majority of these kind of endangered species, and we have only touched the tip of the iceberg. We have not even traversed the first few meters of it, right? And there's a huge potential. Our wildlife hold the key. We are wildlife rich countries, right? Global North does not have the wildlife, but they somehow hold all the expertise somehow. And we are the wildlife rich country, and we have not been studying our wildlife enough. So I would like to reiterate, it's very important to get to the ground level, reassess what the priorities for our country are. Of course, we are all signatory on UNESCO and a lot of other species. Some of the species are global priority, but again, we always need to think about how much my community is going to benefit from this species versus the glorious other species that we host, and start with that. Build capacity slowly, maybe if necessary, and build all the resources that are necessary, and then look at the fancy stuff like whole genome sequencing, genome assemblies, which of course I am also biased towards, to be very fair. But when I look at things like, okay, these 19 species I can't even detect in a wildlife trade, I need to do something, right? And if there was something I missed, please do remind me.
No, I think you've really addressed it, and you've touched on a very, very key point, which I want Dr. Francis to pick up. This whole issue about funding from locally generated resources like tourism. People travel all the way from, the corner of the world to go to Rwanda to, see a gorilla. And in Kenya, we have a very vibrant tourism industry. How can that be able to build into some of the sustainability plans that we're talking about? Because it's an important asset. It's a national asset, not only for Kenya, but also for the region. But I also want Dr. Francis to touch on something about how can Kenya stop losing its samples. I know WRTI is able to give permits for samples to be shipped outside the country. And now that we're building these resources and capacity in Kenya, is it still necessary to do that? Or should we start thinking of how we can, better collaborate and better finance the building of infrastructure locally so we don't continue losing samples to scientists out there? Thank you.
Thank you. I think in terms of resources, definitely tourism has a role to play in terms of sustainability of some of these projects. Some conservation fees could be one of the ways to go. There was establishment of wildlife conservation trust that will be able to manage such collections, and then that kit can be used for such a project. So it's something that is there. Definitely, tourism will go a long way in sustaining some of these important projects that are for conservation benefits. In terms of sample export and import, we always encourage that most of the tests or most of the work be done locally, because it's only through that we can be able to improve our infrastructure. If we say every other sample that has to go out of the country, then we'll not be able to manage or go out of our countries. We'll not be able to develop the infrastructure. So as much as possible, it's important that virtually all the processes or all the procedures be done in country so that we can improve our infrastructure. And if something has to go out, it can only go for to a reference laboratory to ensure that whatever was done here is acceptable across board. But I think I believe the way to go is to ensure that all of it is done locally. And whenever it goes out, then the material transfer agreements and other important documents, prior informed consents, mutually agreed terms must be well negotiated to ensure that the benefits are accrued from the resource provider. Yeah.
Thank you very much. So those of us who are here in person and also online, this is not a conversation we're having in exclusion of you. Please note down your question. We will be able to ask the panelists of a question that you have asked or something that you're seeking clarification. Remember, we're talking about biobanking and custody of samples and also questions of data sovereignty. But talking about data sovereignty, Dr. Laura, and you mentioned forensic applications, which also involve cross-border. You know, somebody leaves Kenya with a wildlife sample and goes all the way to the corner of the world somewhere in India, and you're talking about forensic sampling, and you need material. You need this data bank. You need a material to be able to compare your samples with. Could you make a comment about these forensic applications and how restrictive or unrestrictive? How can we make it work better? In other words, and then just very, very quickly, perhaps also talk about sovereignty. Because maybe that's how we're shipping samples out to to Anubab there in Bangalore to be able to see whether somebody walked out with an elephant hide or something like that. Your comments on this? Thank you.
Yeah, thank you. I think this is a really, really important topic. And I think one way of moving forward is making sure that countries have their own capacity to do this kind of analysis, right? Maybe not every country. Maybe we start with kind of regional hubs. There's an East Africa hub, there's a Central Africa hub. But it's really important, I think, that there's not a overly reliance on a lab somewhere in Europe or somewhere, in a different continent to generate the data that is needed, especially if it's maybe data that is needed to support in court cases, right? One of the problems that we had in the past, because we did quite a lot of the genotyping and the sequencing we have done in Europe, we're now we've been working for the past year and a half on what would be the roadmap to make sure that everything can be done locally. For instance, I worked with some Kenyan colleagues on this. But when we were exporting samples to Europe, I have to fill out on a CITES form what the species is, right? We don't know what the species is. That's why we want to do genetics. So what do I fill out on the CITES form? right? Or if I don't know the source country because it was confiscated in an airport or in harbor somewhere. So it just makes everything very complicated if there is not local capacity to do these kind of things. And actually, I think the local capacity is already there, but maybe it's not accessible as of yet, right? We know there's sequencing capacity in Kenya. We know that there's very bright researchers who already know bioinformatics or would be able to pick it up very quickly. So I think we're just a little step away from it, and the capacity might be already there, but it might not be fully accessible yet, especially not for wildlife. Probably for human genomics or cattle or livestock, it's already there completely. But to make it accessible also for wildlife, I think that's the little step that we have to make. And actually, I would like to connect to something that Dr. Anubhav just said. I fully agree that we shouldn't stare ourselves blind on these fancy whole genome data sets, which are amazing and can give us all this kind of information. But for a lot of questions, we can actually do perfectly well with, for instance, smaller SNP panels that just target these 100 SNPs and which allow us cost-effective genotyping, so they can be used for routine monitoring, like population estimates through capture-recapture. You can do that every year or every few years. And the other thing is, it also makes it much easier to analyze the data. You don't need a whole lot of computational infrastructure. You don't need to have all this intricate knowledge of how to filter your data, because genotype data are much more simpler in a way than when you start with whole genome data, which might be, some might be high depth, some might be low depth, some might have quality issues. So I think we need to think very pragmatically, like what kind of answers do we need, or what kind of data give us sufficiently high resolution information to give us the answers that we need, for instance, for forensic applications. And how do we can that actually applied and feasible to be done on the ground. And I think moving forward with building capacity, and I think AfricaBP is one of the great vehicles which is moving in that direction, building capacity locally to address these kind of issues.
And that is why we at Africa BioGenome Project are focusing on doing the reference genomes for the scientists, so that you as scientists can, do short read sequencing, can do your SNPs. You don't have to invest in the whole genome sequencing. We're saying, let's partner together. Let's sequence your organism once. We do it really, really well. And then you can, continuously use that for your, population diversity studies, your, monitoring, surveillance of numbers, and or eDNA analysis, whatever it is that you're going to apply this information, it'll make it much cheaper for you and much, much easier for you. But I want to turn to Anubhav, and Dr. Laura mentioned about building capacity. I've been building capacity for the last 25 years, but I think I can count on my hand the number of scientists who are still left around me. Most of them have left for, other jobs in other institutions. Why do we lose them? Why are we not able to retain them? Is it an issue of being unable to give them positions in local institutions? Is it the salaries? Is it the lack of the career growth? Help us understand this brain drain, because we have very many young minds here. We have an institution like Mount Kenya. They have, sequencing infrastructure, bioinformatics infrastructure. They're doing robotics. So the infrastructure is here, but the young scientists are leaving us to retrain and retool over and over again. What is your comment on this?
I think the challenges are manifold, and thank you for pointing it out, Professor. So I'll start with the first one, is salaries. So salaries in industry are way more lucrative, of course, and the bioinformatics skills themselves are a bit rare, and they're broadly applicable. This is also something I say to my students who often come to me or my department and is like, why should I choose a wildlife project instead of a neuroscience project or a cell biology project? Because they're thinking in terms of I'll immediately get a job in a biotech company if I do a neuroscience project. On the other hand, I tell them, see, the skills are transferable. You learn statistics in my lab, you learn genomics, you learn bioinformatics. And this is how students get placed in biotech in industry. The same thing happens to scientists. So a biotech a biotech industry is definitely interested in the bioinformatics skills. They are able to offer more salaries, and that takes away from academic salaries, which are always generally low. That is one. Second, dollar by dollar, I get much lesser in Africa and India or other global south countries than I get in the Europe. The sequencing cost in Africa, for example, for 1 GB of data, I am paying 3 times the money I would pay in Europe, or maybe 5 times the money I would pay in Europe. So the research grants are lower in my country and here, but I have to spend more to do the same work that a researcher in Europe would do. So I would rather shift my lab there, get more recognition, publish nice papers with brilliant data sets for much cheaper. And the research grants there, I don't have to spend as much efforts as I would to get the same amount of data, right? So if I'm generating, for example, $100,000 here and $100,000 there, I get to do much more because costs are lower. And here the costs are higher. The costs are high because we don't manufacture. Is the problem one. None of the consumables get manufactured here. Two, even the importing the sequencing reagent, we have to pay a customs duty on it, which is actually very prohibitive and very high. Second, even if I have all infrastructure, getting all that infrastructure into the country is also expensive because the infrastructure is also not manufactured here. Right? Everything, and we are placing, fourth thing I forgot is the journals. I published something to a journal, I sent them, they're like, oh, this is a very specific interest, maybe go to a specialized journal. This is not of general interest to people. There are 1.4 billion people in India. We are 1/8 of the world population. If something is of interest to India, it is of global interest, right? But a Western researcher who is a reviewer or an editor, and all the journals are somehow based in the West, won't view it like that because it is not of interest to Europe, right? That is a fourth problem. If I base myself out of these countries, I'm not going to be able to publish to these high impact journals easily. Fifth, my university is still going to judge me based on whether I have published a paper in Nature or Science or not. Nature or Science is going to charge me $12,000 per paper. My government, my university, my research organization does not provide me that fund. How do I publish in Nature or Science? And you're going to compare me with my Western counterpart. So this is all the many reasons, and I'm sure there are a lot more if we put our minds to it, which make it difficult to retain talent in our own countries, in our own academic organizations. I would recommend actually getting out of this space and thinking very inclusively and thinking about, we should not judge the work based on the publications we make, but rather the ground impact I have. How much outreach does my work do within my own country? Does my work have impact within my own country rather than what we call a global impact, because my country definitely has more population than any other country in anywhere else in the world, right? So we need to start thinking about what we are doing. Plus, if there are some ways to facilitate research by reducing import costs or start manufacturing within country. Kenya has a huge coastline. You literally have the algae needed for making the agarose we use on an everyday basis. We don't even manufacture these agarose, which we definitely can do this in Kenya. I mean, the costs are incredibly high and we should start manufacturing and we can't, or we don't. And these are the things that we need to start thinking about. It needs a very systemic change to retain our brains in our own countries.
Well, I'm so happy to see you're back home in India and you're not, in Copenhagen, and I'm sure you're building a lot of capacity. So there's always hope, despite some of the challenges that you face. I think if you look at the coin on the other end, you do your bit and you make the impact where you are, and I'm sure it will resonate. I'd like to now turn to the audience here. Do you have any questions that you would like to field? Is there a question online that we can be able to discuss? And as you're getting organized, I'd like to hear Dr. Francis's thoughts on how this biobank will impact in Kenya and in the region. And I'm sure it's not only a resource that's going to be available for Kenya, but maybe I think it's the first wildlife biobank that is going to be established in the region. And what are your thoughts about that? And how can we bring out its visibility? And more importantly, as we're backing up our samples in biobanks elsewhere, because we have to do that, how can we maintain visibility on our samples?
Okay, thanks, Prof. Yeah. As you have said, as we come up with this biobank or this facility, we also focus it as a regional hub, really, where a lot of science work goes on. And as I stated earlier, the biobank will be very important for progressive research, especially in areas of genomics and molecular biology. You talked about eDNA and so on. It will also be very, very important when it comes to health, especially wildlife health, whereby it will be very important for disease surveillance, disease diagnostics, especially for retrospective studies, and more so where you link the epidemiology of diseases and when probably it started, so that you can have a good mapping of diseases. Reproductive biology, I talked about that, very, very important, assisted reproduction, especially for the species that are either extinct or are critically endangered, or they are rare and endemic, to ensure that whenever we have problems, at least you have a fallback. Also in terms of histopathology, diagnostics, and so on. So it will be really a very, very important facility for us to be able to move research, especially applied research, to the next level. In terms of accessibility, the Institute, again, is in the process of establishing a wildlife database that will be able to bring most of the information that is there. Is a requirement of the Wildlife Conservation and Management Act, the legal document, or the legal the legal act that manages the wildlife in the country. So again, whatever we get through this biobanking facility will be able to input into the wildlife database. So therefore, is a big milestone in terms of advancing research, conservation and management of wildlife resources in the country and in the region, and also will be very important, especially to inform policy, because policy is dependent on data, is dependent on information for future management of the resource that is very critical for our countries. Yeah. Thank you.
Thank you for that. And I know you have a very strong education arm that is attached to the WRTI, which is doing incredible outreach to schools, so that the children who are learning around the institution can be a bit more informed about the wildlife, and they will be the next custodians for this particular resource, because they live in some of these national parks, and therefore they have interacted with the wildlife, but ensuring that they are aware of the specific role they play, especially as custodians of wildlife. Do you have a comment about that, about the next generation, rather than having myself and my students coming all the way from Nairobi to guard wildlife that's in the middle of the park? What about the people who are living with them? How can they benefit more?
Wildlife Research and Training Institute has a training wing that mostly focuses on diploma and certificate courses in wildlife management and wildlife management related courses. And it trains middle level professionals who really work with wildlife conservationists, with communities in the conservation areas, and then they become very good ambassadors of wildlife conservation. So they will be able to be exposed to the importance of biobanking, the importance of biological resources, and especially for the importance of biological resources and the importance of this facility, so that they can also be able to create awareness among the communities and among those who live very close to the wildlife areas. And we also have postgraduate students who come to us, attached to the various research projects from various universities in the country and outside. Again, these are other very, very important ambassadors of genetic resource conservation for future, as a future insurance for wildlife conservation. So generally, that link between research, training, and awareness creation among communities and those that live around conservation areas is very, very important for us. And these facilities will be able to enhance that role of the Institute and other conservation agencies. Yeah.
Thank you very much. So we have a few questions here. Please raise your hand. Also look at the questions that we have online. Put your hand up and we'll unmute your mic and you'll be able to ask the question. A question for the speaker? Yes, there's one there. And there's another here.
Thank you so much for the valuable conversation.
Your name and institution?
Okay, my name is Fathi Jacob Jaquat Bawesti. I'm doing my master's. My question is, are we can as African, from my experience with biobanks in Egypt, the main challenges is to support local research, not just to store the samples. And how can AfricaBP initiatives can protect our genetic data and ensure that local African researcher truly benefits from their samples without export from outside or took them to outside. Yeah, thank you.
Okay, so I don't know whether you got that. It's how can we support local researchers? It's not only about storing biosamples, but how can we have other initiatives like African BioGenome Project to support? Yeah, if I understood your question? Yeah. Yes, excellent. We'll ask the second question. Go ahead.
Hey everyone.
You can stand.
So my name is Stephen Mburu. I'm a student of biochemistry and molecular bio at JKUAT. So my question is, in this whole discussion, where would an undergraduate play? How will he contribute or she contribute to this conversation? And is it that, because most of the things I hear out is once you are done with your undergraduate, pursue something outside Kenya. And when you're outside Kenya, the samples you are doing, you do them from here and then you take them back there. So the question is it that we as Kenyans or Africans in general lack the infrastructure to actually do some of these things? And also, how would we as undergraduates contribute to the discussion you are having today? Thank you.
I can assure you've contributed to it by asking that question. You already are, but I'll hand it over to the panelists. Anyone who wants to take on the master student and the undergraduate student's question.
Yeah, I can maybe say something. I mean, I don't think I'm in a position to actually answer these questions because these are the big questions, right? But I can share some thoughts. So I mentioned before that we've been working with KWS and WRTI for a long time because there is a system in place where people can go on a study leave, pursue a PhD, which can be also outside of Kenya, and then come back. So I think having these kind of structures where you have an opportunity for your own people to pursue, whether it's a master's degree or a PhD, but also make it attractive to come back and disseminate the knowledge within their own organization. I think that is a very good way of doing it, and also ensuring that whatever they do during their PhD is actually something addressing a question that your own organization has, right? Of course, it's great if you do a PhD and can pursue whatever, but it's even better if it actually addresses a question that you have. How to benefit? I think there is it's really important to think about if you have a biobank and you make samples available for the scientific community, which is great, right? Because that's one of the reasons why you have a biobank. What are the conditions? You know, how can you set conditions in a way that you say, sure, people abroad can maybe utilize the biobank, but there's certain conditions, for instance, involvement of our own researchers, active involvement, not an honorary co-authorship, but a real involvement of our own researchers in these projects. And I just want to emphasize something which Dr. Francis already said, and I think yesterday Dr. Mishuki also mentioned. It's the traceability of material, traceability of resources, right? If samples are shared with another institution, then it's extremely important that there's some kind of documentation in country that these samples were shared. Because I've been in situations where one of our PhD students was working on a set of samples, and the same set of samples was being sent out to another party somewhere else. Of course, it's not our decision who to collaborate with, right? So there's free choice on who to collaborate with, but it's good to have this information documented somewhere just to avoid that your own people are being scooped or resource funds are being wasted because essentially two different groups who don't talk to each other are doing the same thing. So this documentation and setting conditions under which you can share samples, I think are extremely important components on making sure that your own colleagues, your own research, your own students in your own institutions benefit from these initiatives.
Dr. Anubab? Unmute Dr. Anubab. Yeah.
Okay. I agree with everything Laura said, and I would like to add a couple of more points. So one of the questions was, I am an undergrad, what can I do? As an undergrad, you're already quite privileged compared to a lot of other people. Not everybody is undergrad. Not everybody even has that level of education. So already quite in a privileged position to start raising awareness and spreading awareness about what is being discussed and what are the challenges in wildlife. That is one very important contribution. Second, I don't think our researchers or our institutions lack facilities that are available outside. So like Professor Ann just said, that the university hosting us already has a lot of infrastructure. You can see the brilliant work being done by WRTI, right? These are all world-class infrastructures, and these are already amazing outlook and thinking that is leading to all of this. If you just follow actually what Laura said is you get trained and you make sure you come back because you have trained on something that is relevant on ground here. I think that solves a lot of issues. Making sure this happens is very important, and raising awareness and being aware of your own rights, not for a lack of better word, constantly compare ourselves to what is being achieved outside, because also think about what's already being done outside is very different from what we have here, right? Our countries face different kinds of issues. So we have to work in a social fabric in which everything needs to fit together. So it needs to be important that we come back, spread awareness, and even if we don't go out, there is a lot of things to be done at home. And it is important that we do it and not just think about, oh, if I was outside, I would have done more. No, whatever contributions you're making at home is also very important, and you need to do it.
Dr. Francis, anything to add?
Yeah. I think the first thing that I would like to add is WHI is a public institution, so this platform will be a public facility, really. So to encourage young researchers or even experienced researchers to be able to access it, because the public, the country is investing is investing for the public good. So it will be accessible for students. It will be accessible for young researchers and even experienced researchers, definitely on condition. The other important thing is that for those who probably will study abroad, you can still register there and do part of the work there, the sandwich programs that are there, and then you can do your research or your project within the country using or accessing some of those samples in that facility. So why we are doing this really is as much as possible to encourage and enhance our research capacity, both within and without the borders, but more so for the Kenyan public. So this will be accessible facility for all of us. Yeah, I think that's.
All right, we have a hand up online. Salvatrice, please unmute yourself and go ahead.
Thank you so much. Thank you so much for this discussion. I like the discussion about how to engage with the local communities in this biobanking. My question goes to Francis. How do you work with, do you hear me very well?
Loud and clear.
Okay. How do you work with these local CBD regional centers, which are really strengthening this traditional knowledge in the local communities, and also the work of CBD to strengthen at regional level? The one in Africa, I think, is based in the RMD. I don't know if there is somebody from here who was really in this conference. I think it can be something good to connect with them. And also another thing is, I was wondering, there are already existing knowledge of local communities, especially in wildlife management, like wildlife conflict management. Animals and the local communities are in contact with wildlife. How this biobanking can be integrated in this wildlife management of local communities and community-based wildlife monitoring and the protected areas management. Another question was about tourism. Tourism is generating a lot of money. How to link this genomic project with local communities, traditional knowledge, but also tourism actors who have money, who are using money, who want to collaborate with local communities, and also the researchers. I think it's something to think how to bring all those actors together. Thank you.
All right, we'll take two more questions, one here and one online.
Thank you. My name is Protas. I'm a postdoc at MKU. So I have two questions. Maybe I will also link them to Dr. Francis. The first one is about traceability of these samples once we bank them. So I wanted to know, do we have like a system that even after 20 years, we will be able to track this sample? Maybe the location where it is located, the collector or the provider, so that we can be able to still make the information useful. Then the second one will be, I think you've talked about it. We have some young researchers here, but their main challenge sometimes is funding. So they get a funding from maybe a collaborator from outside. So they will use the samples, like you've said, to generate the information. So do we have like an oversight to ensure that we don't just store the samples, but the information is being used like outside there? Do we have a way that this information will still come and benefit us here in Kenya? Thank you very much.
All right, we have Jeff online. Go ahead. I hope you're noting down the questions that are coming. So Jeff, unmute yourself and go ahead.
Hello, everyone, and thank you to all the presenters for the wonderful presentation. I'm a student doing ecology, master's, and I did my data collection in a park. But since there were insects, I also worked with the museum. So my question is, as the WRTI is, that's the Wildlife Research and Training Institute, is thinking of establishing their biobank, what are the plans to ensure that they use the capacity that already exists in the national museums? For example, we know that the national museums have experts in entomology, which mostly lacks in the WRTI. So what are the plans in place to ensure that the already existing capacity in national museums and the samples that are there, some are as old as more than 100 years, are there plans to ensure that we do not do each other doing the same thing?
All right, thank you for that. One more question.
The second question is,
okay,
you realize that as you study,
Go ahead.
Oh, I think his network has dropped him out. So while we wait for him to get back, Rael Mary, you can ask your question.
Hello.
Hello.
Hi.
We can hear you.
Can you hear me?
Yes. Go ahead.
This is Rael Mary from National Biosafety Authority. Now, my concern is on the biobanked information. Do we have some strategic frameworks where we, despite the information that has been stored, the samples that have been stored, do we have a strategy where we align to the person, the founder of that information, whoever worked on it, apart from just the organization, so that by the individual names, if we can make a follow-up on them? Maybe if we go back to the previous topic where we were talking of compensation to them, so that we make a follow-up on, can they be compensated, like despite less talk of the wildlife, if at all the funding comes to the wildlife because they're the ones who provided, can it downstream to the original founder, like the information owner? Then if second question, we also have this information that has been availed since the old ages, before the coming of these, before we start this economic strategy. Are we going to consider those people who ever did that work that has never been recognized? Will we make a follow-up on them and maybe recognize them, compensate them? Because after all, their information is what we're still using at this moment for reference to our new and current research. And number three, and the last one, if we are putting this information in the biobank and then someone wants to access it, what are the figures that we are going to put in place so that we ensure that this information being found out is not so expensive to the researchers, so that research work is now constrained with the amount of money that has to be paid for someone to get the information? That's all.
Thank you. Thank you very much, Marie. Dr. Gakuya, lots of questions to you, but after you've made your comments, Anubab and Laura, you're free to make any additions or clarifications. Thank you.
So thank you. I think I'll start off with the CBD one. Yes, as I said, service or resource providers, community are some of the research providers that are there. So every time that that resource is being, that resource, somebody is accessing that resource, there must be that negotiation with the community through the PIC, prior informed consent, and mutually agreed terms to ensure that the benefits are accrued, that will be accrued, referred back. So in terms of Nagoya Protocol, we are very strict on that. If any of that resource is in community RAD, then it has to go there, and that has to be signed. And in terms of management, really, we are moving towards community conservancies. I think for those who follow the Kenyan conservation and management policy, community conservancies have been coming up in very many areas where now the community benefits directly from wildlife as part of sustainability of wildlife conservation. The second one is on traceability. Yeah, that will be very important. We have to, we foresee having a very robust system to ensure that traceability is there. Laboratory information management system that will really capture all that information that is required, such that even if you go many years back, you'll be able to identify the species, whoever collected that, and all that information, all that metadata will be very, very important. So I think that will be taken care of, because there's no way you can have a biobanking facility without metadata, because those samples or that information that is there might not assist in terms of research. Funding oversights, definitely the oversight will be done by the scientists who will be involved, and also they'll be able to guide on exactly how to access those samples. In terms of museums, we are working very closely with our museums are one of our stakeholders. They are part of our partners. We're working closely with them in developing the strategic action plan and even going forward how we go about it. And it's not only the National Museums of Kenya, but all the research institutions in the country that have a stake in wildlife conservation, because conservation cuts across board, as you have stated, all the way from mammals to avians to reptiles and so on. So everyone will be part of this process. It's a process that involves diverse stakeholders across the country and even outside, especially in terms of the technical backup. About the compensation, I think there are ways of doing that for the resource providers. We've talked about that. Nothing will be able to come out without prior informed consent, which will definitely state how the benefit accrued will be able to be shared. The same with the coordination, probably to come out very clearly, or it comes out very clearly in the PIC. In terms of access, as I said, this is a public institution. Access will be there. Obviously, there's some cost, and the cost is really for sustainability, not for profit. Remember, as I said, being a public institution, profit is not what drives that. Is for public good, but definitely there will be fee that will be negotiated as a whole that will come up for access, but really for sustainability, not for profits. Professor, I think I've tried to answer as much as possible all the questions.
Thank you. Any contributions from anyone?
I don't know how feasible it is. One of the questions raised was the research money is limited, and how do we make sure that the biobank samples, if they are being accessed if they're needed by multiple people, if there's a way to reduce the burden of the research fee. One of the ways this potentially could be done is if it is possible for the maintenance of the biobank or WRTI to have a system where we can say, okay, these are the samples that are available. This is a person or this is a institution that is currently generating this kind of data. And you make it a condition that if there are other requests for use of that kind of data set, it becomes shareable across institutions, provided the research questions are not completely overlapping, then that will lead to conflict. But if there are two different questions to be asked with the same kind of data set, maybe the organizations can share it. That saves precious biobanking material and also the limited research money. I think that might be a good strategy. I also think one of the ways that some of the other concerns were raised could be addressed if there is a research proposal that is evaluated in terms of there is a specific box which says what is the benefits to Kenya, or what are the benefits to wildlife, or what are the benefits to capacity building or local researchers in this particular research question that is being addressed with these biobank samples, so that the question of sustainability is addressed for sure, but also how important this biobank becomes for the country it only improves with time, rather than it becoming another pot where a lot of parachute science happens, where these are just treated as a one-stop shop for all African biodiversity, and most funded researchers get access to it, rather than the researchers with the most impactful work on ground get access to it. I think these are some of the safeguards that might be thought about and planned for the future. I don't know how feasible it is, but the same.
Yes, thank you. There's a question from Prof there. You can stand so we can see you from here.
Thank you for
Make
this
it,
opportunity.
Brief and.
Oh, yeah, thank you. I'm Professor Sifuna Sudi, working with Center for Malaria Elimination, Gitaka Lab. So mine is I don't know if I can put it as a question, a comment, or just an addition, because from the discussion that has been ongoing, I get the idea that research information data is coming from Africa, being sent abroad and being commercialized, and the African researchers, or the source of the information, they're asking, how do we benefit from this? And the question that has been rising within me and burning me is, are the inventors of this knowledge not benefiting basically because the users of the knowledge out there are not sending back the benefits to the people, or they are sending back but the benefit is not reaching the people because of some factors? So I've lived in Japan for 8 years, and one of the factors, or the thing that I saw drive their economy, is trust and integrity. So is trust and integrity affecting the way the inventors of this knowledge in Africa are benefiting from this knowledge that they are generating? Is it a hindrance? Is money coming back and not being shared equitably? Or someone somewhere is hiding the money? So that the reason why I said I don't know if it's a question or a comment is because I want to urge us, as we set up this Center for Biogenome in Africa. I'm not a specialist in genomics, that's why I'm not really into my question, or my comment is not really into it. But as we set it up, let's ensure that we are guided by principles of trust, principles of integrity, so that when someone generates something, settles, rest assured that for real, I will benefit from this, not because it has not been brought back, but the benefit have been brought back and they are being held up somewhere. So I'm very happy to see Professor Francis Gakwe as a person who sits in policy making, who sits in the tables up there, who can make serious decisions and make serious comments in wherever he is. So please, I urge you and I urge all of us that we put integrity forward, so that we don't take up the benefit of what does not belong to us. That will motivate all of us to work very hard towards making Kenya and Africa a very nice place like other countries out there. Thank you so much.
Well, that's a high note to end it on, actually. Are there any comments on this?
I can make a very brief comment. I fully agree with you, and it's not a problem that is unique to Kenya or to African countries, even though I can see that it plays out differently. But we are involved in a large study across European museums, natural history collections, and they have a huge problem that they send out loans and people sequence them or do interesting stuff with them, and they don't always get the data back. Or if the data come back, then maybe there's no unique identifier anymore. So there's a big problem across the board when it comes to the direction and the communication and the direction of information. So part of it, I think, like yesterday, Vice Chancellor Professor Yagani said, you go from field to freezer to flow cell to findings. Maybe, but part of it is also bookkeeping, making sure, and I think that's where biobanks come in. You collect your resources, you collect the metadata, you do thorough bookkeeping to know what you have, where it's being sent to, under what conditions, and how you involve the different stakeholders and the different people in these kind of projects. So I think I completely agree with what was just said.
Thank you very much. Dr. Anubab?
I just wanted to highlight, it was a very noble point to be raised, and I completely agree with you. It's just that the level of complications actually go a little further. We need to ensure that we have the capacity to utilize all of the data being generated and used. So you work in malaria, for example. A lot of researchers actually work on malaria, and there's an active effort to build malaria vaccine. Malaria impacts a lot of countries, Africa especially. But do we have the infrastructure to manufacture the malaria vaccine if we actually come across one, right? The vaccine manufacturing capacity lays outside of the continent most often. And this is the case in a lot of countries and a lot of the tropical countries and tropical continents, actually. Things like that. So snake venom is another example. We are all affected by snake bites, but where are the antivenoms manufactured? Majority of the manufacturing actually happens outside of the country. And then we get the benefit in terms of the finished product, which we sell for a very high price in our own home countries. Even though the problem was ours, the solution laid with us. We provided the raw materials for generating the solutions, but we got a finished material for which someone else is making the profit, and all we get is some benefit out of it. So it's a very complicated network, and it needs to be addressed at a more fundamental level. I think it is very important that we motivate and sensitize our students to get into startups, to generate or mechanisms of utilizing their own data set to commercialize some of these knowledge that is being put out there, and to be more industrious with our own bioresources. It's very important. Otherwise, this problem will just persist, and we will not be able to solve it at a fundamental level. It is very also important, I think, to add a part where we share IP. So if our sample or data has to go outside, I think there needs to be a condition that IP has to be shared with local partners. For example, if WRTI is doing it, WRTI needs to have a condition that if any IP is getting generated, WRTI is a partner in that IP. I think it's extremely important to do that. Otherwise, whatever the concerns were raised by the professor will persist for a very long period of time.
Thank you. And final remarks, Dr. Gakuya Francis.
Yeah, thank you. I think that was powerful and it was good and the way to go. As he has stated, we have the resource. So it's for us to ensure that as we negotiate for access and benefit sharing, we have to ensure that the resources really come back to us. And especially when it comes to IP, the intellectual property, right? We have to ensure that in the document it's very clear that this resource belongs to us, and any benefit that is being accrued has to come back to us. Again, it's also important to create these facilities and infrastructure so that we can also develop the local capacities to do some of these things. Because I know you have quite advanced very well in basic research, but applied research, we are still lagging behind, as you have stated, in terms of production of vaccines, pharmaceuticals, and so on. Is still in most of it is still in the global north. And I'm sure with the more we expand such facilities, the more we bring the resource availability closer home, probably the more we advance in those areas. So we begin somewhere and move and grow slowly until you reach there. Yeah, I think I'll stop there, Prof.
Thank you, and that's a summary that we have talked about. And I must thank Professor Sifuna for asking that question. That has allowed us to sort of like wrap up and summarize. At the end of the day, integrity in sample collection, research, and also applied research, and plowing back the benefits to the communities. This has been a very, very, very stimulating panel. Every time I come and say, okay, the next panel is, I'm going to speak about this. And I'm like, oh yeah, this was very, very stimulating. I'm here to say it. It's a conversation that needed to happen. It has happened. It has just begun. And we want to continue having these discussions and these tense. Sometimes you have to have a tense discussion in order for the best result to come out. You don't have to always agree with each other, but we need to push our agenda until we agree that we have reached a middle point. And that is the point that we want to emphasize. So a round of applause for our panel. Thank you very much, Dr. Anubab, Dr. Francis, and Dr. Laura, for spending an hour, more than an hour, actually an hour, 15 minutes, in this very, very enlightening discussion. And stay with us. We're going to take a lunch break, but we'll be back. We'll definitely hear from Dr. Anubab again as he explains to us the practical that they'll be doing in Kilifi from tomorrow. So online panel, we're taking our 50 minutes lunch break. So let's convene in the next 50 minutes. And that, according to my time, will be 10 minutes past the hour. Thank you. Thank you very much, and look forward to you enjoying your lunch break. Thank you.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipments. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Vilba. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC we have Rwanda, we have Uganda, we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our cells are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, amplification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
PacBio Hi-Fi sequencing technology gives researchers a comprehensive, multi-omic view of the genome, including GC-rich regions and long repetitive sequences that other technologies can't fully resolve. Here's how Hi-Fi works. Hi-Fi sequencing is built on single-molecule, real-time technology. Its accuracy comes from repeatedly observing the same native DNA molecule as the polymerase replicates it. First, DNA from nearly any organism is converted into a circular template by ligating adapters to both ends without PCR amplification. This avoids bias in highly repetitive or GC-rich regions and allows for the simultaneous detection of genetic variants and methylation status in a single assay. Next, primer and polymerase are added to the library, and the sample is ready for sequencing. Sequencing happens on a SMRT cell, which contains millions of tiny wells. Inside each well, the polymerase gets to work, building a new DNA strand base by base from the native template. As each base, A, C, G, and T, is added, a unique pulse of light is emitted and recorded in real time. These pulses create a continuous record of the molecule as it's read again and again. Hi-Fi also captures epigenetic information automatically. When a base is modified or methylated, subtle shifts in the kinetic signature of the polymerase's incorporation rate reveal rich epigenetic insights like 5-MC and 6-MA methylation. This happens automatically on every sequencing run without additional library prep. Hi-Fi reads are generated by combining information from multiple passes around a single DNA molecule. The system combines these repeated observations to produce long, highly precise reads, exceeding 99.9% accuracy. Powerful on-instrument algorithms and analysis tools turn this raw data into rich, multi-omic insights, revealing the genome, including complex structural variants and dark regions, the transcriptome with full-length isoforms, fusions, and splicing, and the epigenome with base-level methylation patterns captured natively. All in one run, all from the same molecule. With Hi-Fi long reads, researchers can do everything from interrogating the hardest regions in inherited disorders to powering population-scale precision medicine with complete, accurate genomic and epigenomic data. Hi-Fi sequencing removes blind spots and delivers the accuracy needed to understand biology at every level.
In Kenya, science is not just advancing,
It's accelerating from the front lines of public health to the frontier of innovation. A silent revolution is unfolding, and at its heart is a new way of seeing life itself. Next generation sequencing. Disease doesn't wait. It doesn't discriminate. But what if we could see it coming before it strikes? In Kenya, the burden of infectious diseases, antimicrobial resistance, undiagnosed conditions, and now a sharp rise in cancers, tuberculosis, and reproductive and genetic health disorders calls for sharper, faster, more precise tools. Beyond hospitals and research institutions, the same technology has the power to transform how we grow food and protect our environment, improving agricultural productivity while preserving and leveraging our rich ecological biodiversity.
The MGI-Tech I100 is a pretty simple sequencer to work with because we do not have a lot of hands-on time.
And so the uptake is really good and the future is looking bright.
With the new MGI-Tech I100, there are some steps that have been optimized or removed so that those steps take place inside the instrument. So as a user, you don't need to do them manually.
MGI-Tech 100, we're able to get the results within 15 hours.
At the Aga Khan University Hospital, we are currently using the Illumina next generation sequencing for a number of key clinical applications. Specific examples are for patients with breast cancer, ovarian cancer, prostate cancer, and pancreatic cancers. The MGI-Tech I100, which is a new instrument, is promising a lot. Compared to the MGI-Tech 100, which I use, the MGI-Tech I100 is promising, for example, a jump from 1.2 gigabases of data to 30 gigabases of data, from 4 million reads to 100 million reads, from 150 base pair read lengths to 300 base pair read lengths, among other advantages. I can still use the MGI-Tech I100 using a smaller flow cell or a bigger flow cell. And so depending on the number of samples that I have, I don't have to worry about efficiency of utilizing my sequencing cartridge. And that's a key point.
The introduction of the MGI-Tech, the I100 Plus, is really a game changer in terms of the work that we are doing. It's very clear that this MGI-Tech will reduce the amount of time we are using to do our sequencing from the old one that we used to do about 58 hours to now 15 hours maximum. You can get your data to as soon as 4 hours. So it means our productivity will definitely go high, and the dangers and encumbrances that were associated with the storage of cartridges, the reagents, have been eliminated because now we don't have to do cold chain storage, we can do over-the-shelf storage. So this would really impact on our productivity and the value of science that we're going to be demonstrating.
MGI-Tech I100 is really at the heart of that transformation.
Our live trials with the MGI-Tech I100 series had a very positive feedback. Most customers requested us to leave the system behind. This demonstrates the positive impact it has already made. And I believe this is unfolding a new era for our scientists in Kenya.
Illumina's latest innovation, the MGI-Tech I100 series, couldn't come at a better time. The faster turnaround time, the ability to sequence more on this compact platform, and by leveraging the same X-Leap chemistry as the bigger platforms, it's definitely offering a number of game-changing features that our markets, our labs, will definitely benefit from.
into a new era in science. In a world where time, accuracy, and precision define outcomes, the future of genomic research has landed in Kenya. The MiSeq i100 Plus, compact, fast and powerful, designed for labs that demand precision, yet operate on tighter scales. Whether you're exploring infectious diseases, cancer mutations, or the human microbiome, next-generation sequencing simplified from sample to insight, faster, deeper, and clearer than ever before. And now, it's here.
Ready for your new mission? The EcoPen.
Hi, my name is Peter Miner, co-founder at Project Mocha. Now, in the last 10 years, I've worked in Kenya's agri-tech space, and I've had many conversations with farmers. And 4 things have always stood out. They lack access to finance, access to markets, access to quality inputs, and access to qualified inputs. At Project Mocha, we are turning coffee consumers to become coffee investors. Now, think about it this way. Instead of putting your money in a money market fund, and it's growing at 10%, inflation is growing at 10%, you can put that money into a commodity like coffee, which is ideally deflationary, and you're actually investing into a coffee farm. Now, on the farmer side, what you do is we realize their assets on chain, and the assets they're looking at are their coffee trees. As opposed to traditional finance, we always look at the land as collateral. By bringing these assets on chain, the farmers have a balance sheet on a global ledger, and they can get investment through.
Hi, I'm Paul Gashora, founder, CEO, Project Mocha. To me, I see Project Mocha as a vehicle that helps smallholder coffee farmers unlock capital to reinvest in their farms and improve their productivity. We are at the revolution, we are at the beginning of a new way to finance production. And so we as the retailers, we as coffee consumers, we get to participate in the coffee production, and we also get to participate in the returns from the sales of our coffee. This is nothing new. We are looking at putting money in investable opportunities to make a return. What we've done is made this process simple, transparent, and you can do this on your phone. That's what I love most, is we're simplifying the investment process and making it accessible to retail consumers, to retail investors.
My name is Wambugu, and happy to be here. So my association with Project Mocha goes back around last year, September, over there, when I met the core team, Peter and Paul. And they showed me about what they were doing. For the first time, you know, they brought this concept that you can tokenize literally any single asset that you can say isn't like a piece of land, right? It's like you can consider anything an asset, and coffee trees is, you know, like it's a very interesting asset because it's always productive. A single coffee tree produces is like a source of income, and you know the fact that that was being neglected by traditional finance was a very, very big gap in the market. The other thing is the team, the dedication, the passion they have, that they carry, just the overall dedication, like from the team, from their partners, the overall support from the community that is just sort of there. I mean, it's just amazing.
My most memorable experience, how would I say it? I will give it the first time I knew about the project. It was so unique from the angle of how they were being able to just bridge the Web3 aspect and the Web2 aspect, and also getting to work together because of the unique aspect. That I will say that is the most memorable experience with me.
The most memorable experience with Project Mocha is during last year's e-Safari pilot, which was the very first pilot that Swiped and Project Mocha did together. So, and really saw a direct, or rather a very direct way in which we can actually impact the industry by adding blockchain as a layer to making it work for our farmers. So it's really made the whole industry see the vision behind Project Mocha. And this collaboration with Swiped, I think really make a difference, not just in Kenya, but also globally, especially as now we kick off with coffee. And I believe in this particular project because it touches back home as a Kenyan and also as a person coming from the farming space.
This stuff is really good. I mean, obviously.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged your one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments that we can carry from the client site to our offices. Basically, these are small equipments. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Vilba. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our sales are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, amplification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So
thank you.
We like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
PacBio Hi-Fi sequencing technology gives researchers a comprehensive, multi-omic view of the genome, including GC-rich regions and long repetitive sequences that other technologies can't fully resolve. Here's how Hi-Fi works. Hi-Fi sequencing is built on single-molecule, real-time technology. Its accuracy comes from repeatedly observing the same native DNA molecule as the polymerase replicates it. First, DNA from nearly any organism is converted into a circular template by ligating adapters to both ends without PCR amplification. This avoids bias in highly repetitive or GC-rich regions and allows for the simultaneous detection of genetic variants and methylation status in a single assay. Next, primer and polymerase are added to the library, and the sample is ready for sequencing. Sequencing happens on a SMRT cell, which contains millions of tiny wells. Inside each well, the polymerase gets to work, building a new DNA strand base by base from the native template. As each base, A, C, G, and T, is added, a unique pulse of light is emitted and recorded in real time. These pulses create a continuous record of the molecule as it's read again and again. Hi-Fi also captures epigenetic information automatically. When a base is modified or methylated, subtle shifts in the kinetic signature of the polymerase's incorporation rate reveal rich epigenetic insights like 5-MC and 6-MA methylation. This happens automatically on every sequencing run without additional library prep. Hi-Fi reads are generated by combining information from multiple passes around a single DNA molecule. The system combines these repeated observations to produce long, highly precise reads, exceeding 99.9% accuracy. Powerful on-instrument algorithms and analysis tools turn this raw data into rich, multi-omic insights, revealing the genome, including complex structural variants and dark regions, the transcriptome with full-length isoforms, fusions, and splicing, and the epigenome with base-level methylation patterns captured natively. All in one run, all from the same molecule. With Hi-Fi long reads, researchers can do everything from interrogating the hardest regions in inherited disorders to powering population-scale precision medicine with complete, accurate genomic and epigenomic data. Hi-Fi sequencing removes blind spots and delivers the accuracy needed to understand biology at every level.
In Kenya, science is not just advancing, it's accelerating. From the front lines of public health to the frontier of innovation, a silent revolution is unfolding, and at its heart is a new way of seeing life itself. Next generation sequencing. Disease doesn't wait. It doesn't discriminate. But what if we could see it coming before it strikes? In Kenya, the burden of infectious diseases, antimicrobial resistance, undiagnosed conditions, and now a sharp rise in cancers, tuberculosis, and reproductive and genetic health disorders calls for sharper, faster, more precise tools. Beyond hospitals and research institutions, the same technology has the power to transform how we grow food and protect our environment, improving agricultural productivity while preserving and leveraging our rich ecological biodiversity.
The MGI-Tech 100 is a pretty simple sequencer to work with because we do not have a lot of hands-on time.
And so the uptake is really good and the future is looking bright.
With the new MGI-Tech 100, there are some steps that have been optimized or removed so that those steps take take place inside the instrument. So as a user, you don't need to do them manually.
ISEC 100, we're able to get the results within 15 hours.
At the Aga Khan University Hospital, we are currently using the Illumina next generation sequencing for a number of key clinical applications. Specific examples are for patients with breast cancer, ovarian cancer, prostate cancer, and pancreatic cancers. The MiSeq I100, which is a new instrument, is promising a lot. Compared to the ISEC 100, which I use, the MiSeq I100 is promising, for example, a jump from 1.2 gigabases of data to 30 gigabases of data, from 4 million reads to 100 million reads, from 150 base pair read lengths to 300 base pair read lengths, among other advantages. I can still use the MiSeq I100 using a smaller flow cell or a bigger flow cell. And so depending on the number of samples that I have, I don't have to worry about efficiency of utilizing my sequencing cartridge. And that's a key point.
Introduction of the MiSeq, the I100 Plus, is really a game changer in terms of the work that we are doing. It's very clear that this MiSeq will reduce the amount of time we are using to do our sequencing from the old one that we used to do about 58 hours to now 15 hours maximum. You can get your data to as soon as 4 hours. So it means our productivity will definitely go high, and the dangers and encumbrances that were associated with the storage of cartridges, the reagents, have been eliminated, because now we don't have to do cold chain storage, we can do over-the-shelf storage. So this would really impact on our productivity and the value of science that we're going to be demonstrating.
MiSeq I100 is really at the heart of that transformation. Our live trials with the MiSeq I100 series had a very positive feedback. Most customers requested us to leave the system behind. This demonstrates the positive impact it has already made. And I believe this is unfolding a new era for our scientists in Kenya.
Illumina's latest innovation, the MiSeq I100 series, couldn't come at a better time. The faster turnaround time, the ability to sequence more on this compact platform, and by leveraging the same XLEAP chemistry as the bigger platforms, it's definitely offering a number of game-changing features that our markets, our labs, will definitely benefit from.
Seconds to a new era in science. In a world where time, accuracy, and precision define outcomes, the future of genomic research has landed in Kenya. The MiSeq I100 Plus, compact, fast, and powerful, designed for labs that demand precision, yet operate on tighter scales. Whether you're exploring infectious diseases, cancer mutations, or the human microbiome, next-generation sequencing simplified from sample to insight, faster, deeper, and clearer than ever before. And now, it's here.
Ready for your new mission? The ICA Pen.
Yeah, so welcome back to our last session, which will be dealing with talking about the practical sessions that we are going to have during this fourth East and Central Africa BP workshop. So one of the things, or one of the objectives of Africa BP is to enhance on capacity building across the continent, and especially to young scholars, young researchers who are still doing their masters, they are doing their PhD, they are doing their postdoc, and even some who are doing their undergraduate. So one of the, I would say, objective of the workshops that we have in different zones, in different nodes like East Africa, West Africa, North Africa, and Southern Africa, is for us to equip ourselves and our young scholars with the skills to deal with the digital sequences that we have, with the skills to deal with, taxonomy, understanding the biodiversity that we have. Remember, we have the highest genetic biodiversity actually in the whole world, and we need hands-on skills, expertise to handle this data. So we are privileged to some of our partners and our tech partners who also not only sponsor such kind of event that we've been having for the past 2 days, today and yesterday. But also they take part in organizing practical sessions where they invite young scholars from different institutions to go and, teach them on skills, basic skills. And the skills can be on basic taxonomy, skills can be on basic, molecular skills or techniques, from pipetting all the way to extracting DNA. And even last year we had session where some of the participants took part in sequencing of this molecular data that we are having. So we are privileged to have, I think, how many? This is 9 practical sessions this year. And this session will be handed to some of the practical organizers that we have to take us through the outline, what they are preparing for us, for the students who will be joining them, and also the output, what they are envisaged at the end of the 3-day coursework or training the students will be able to achieve. So we are going to start with Pwani University, which they are going in collaboration with Indian Institute of Science in India, and they are going to do a practical on a hands-on introduction of African wildlife genomics. And we are privileged to have Anubab Bob, who is going to take us through what he is preparing to give his student. I was privileged to be there 2 years ago with Anubab, and I can tell you for a fact he's a very good instructor. So Anubab, the floor is yours. Kindly take us through what you have in store for us. Thank you. You can start.
Thanks, Paul. I'll just quickly share my screen.
Yeah, sure, no problem.
I don't know.
Just
am I visible? Is the screen visible? So, yeah, what I'm basically sharing is the website we have for the workshop. This year we have modified the workshop a little bit. It's from 19 to 21. And we have added a new module to it. This workshop has been running with ABP for about 3 years now. And we have a new instructor we have added, Dr. Pratna Goswami. And then the workshop is going to be conducted by me and Dr. Sami Wambua, who are based at Pwani University. So day 1 of the workshop, we are going to introduce you to some of the lab techniques and the field techniques we use in wildlife genomics. So this will involve hands-on DNA extraction, hands-on QC, and hands-on library preparation for Illumina sequencing. Day 2 will have a module on population genomics, where we will look at some of the interesting things, which is VCF file and how to look at some of the important population genomics questions, like how many populations are there, is there gene flow, connectivity between populations, how can we measure genetic diversity, how do we measure inbreeding. And then on day 3, we have a metagenomics and an eDNA pipeline where we will try to quantify how much diversity is there in an environment and how much we can do with environmentally collected samples in terms of, for example, measuring diversity of fish and microbiomes. Apart from this, the workshop is publicly available on these websites. So for example, there are questions and solutions available for basic Linux. So what we try to do is generally, like for example, we have asked these questions and we are also providing solutions for some of this. And a lot of this material will be available with a server, so that if you are a professor or a postdoc or somebody who wants to offer this course in your university, it becomes accessible there. So all of these materials will be online. So if you're not able to come in person, the material will be online. The course is completely in person. Okay, the course is not online. Just the materials like the scripts and some of the servers will be online, so that you can access it from anywhere in the world and run it and offer it in your own universities. And of course, our contact information is here on this website. If you want to take a note of this, I don't know if it is visible. It's Wildlife Genomics 4 at Pawnee University, it should be more accessible. And yeah, I think that's almost all I have to say. What you will learn is this year, you will also learn hands-on practical skills on how to handle wildlife samples. And you will learn how to ask questions, what kind of questions can be addressed with what kind of data, and some basic population genetics. And second, if you're doing metagenomics or eDNA kind of work, you will learn some of those skills as well. Thanks so much for the opportunity. I hope some of you can actually show up and join in the workshop. I'm open to questions if you have any about the workshop that we are offering.
Thank you, Bob. Thank you for that wonderful introduction to your course of this year. Do we have a quick question or clarification that any of you might want to know about this course? Online, maybe in person? Anyone? Anyone here who will be joining Bob? None? Okay. Oh, that's true. They're on the train to Mombasa and Kilifi. All right, that's true. That's true. Online? None? All right. Thank you. So I think then next we go to INCABA East Africa. This year I see they have 2 workshops. One on bioinformatics without tears, introduction to phylogenetics. And I have a quick question for them. Where were they when I was starting my master's, Prof? Because I really suffered. Yeah, I didn't even know how to download sequences from NCBI. So you guys are lucky. So we're going to start with the bioinformatics without tears. That is introduction to phylogenetics. And secondly, understanding biodiversity through Sanger sequencing. So from INCABA, who do we have? Dennis is around. Ah, nice. Thank you so much.
Okay, so my name is Brian Andika. I'm an application specialist at INCABA Biotech. So this year we've organized 2 workshops. The first workshop will be on phylogenetics. Then the second workshop will be on Sanger sequencing. So to answer your question, we started operations, the lab operations 2 years ago. So we will invite you definitely to come and get into the sessions and learn a bit more about the data analytics. Okay, so on the phylogenetics, basically what we'll do as an outline, we'll try to infer relationships, evolutionary relationships in different data, NGS data. We'll also have some Sanger sequencing data that we normally do in our daily basis. So basically what the learners will learn, or the delegates will have to learn, is how to do sequence alignments. The next thing they'll do is, of course, learn these, how to infer the phylogenetic models. And finally, how to interpret these results. So we're talking about bootstraps, we're talking about how to root a phylogenetic tree. Because not all trees can be rooted. So we'll also be able to cover that in the training. So the sessions will basically take between 3 to 5 hours. That's for 3 days. Morning hours, typically we'll have our lectures. So we'll go through the basics of phylogenetics. Then during the lectures, we'll also have some exercises we're doing as part of the lectures. So the main aim of this workshop is to build the hands-on experience on some of these analysis techniques, especially the phylogenetic analysis. But we'll also cover parts of sequence alignments and interpretation of the trees as a part of the training. Okay, so for those who are going to be attending the workshop, so I think what you'll need is a laptop. Just bring your laptop. I believe I've shared the softwares that we'll be using for this analysis through the emails for those who are attending. But we still have space for around three more students. So you can send in your applications and we get to invite you to the workshop. So we'll be doing some, of course, we'll be installing softwares in the laptops for these sessions. So kindly bring your laptops. Yeah, so I've also shared the sample data sets for these sessions that will take place in 3 days. Finally, who should attend this workshop? So basically, we are looking at new researchers who probably don't have enough experience with data analytics, that is especially phylogenetic analysis, because we'll be introducing you people to some of the softwares that we use for our normal services at INCABA, that is phylogenetic analysis, if you've ever ordered such a service from us. So we're also looking at experienced researchers. And if you also have your data set, we can accommodate you. We go through your data set and we show you how to make these inferences in the trees. So I think that's basically it for the phylogenetic analysis. We'll be going through all these softwares. We have some of these Bayesian methods. We'll also be going through the normal GUI methods for analyzing the phylogenetic trees. So I think at that point, let me introduce, unless you have questions, let me introduce Alan, who is my colleague at INCABA. So he'll be taking you through the Sanger sequencing workshop. I'll be taking you through the phylogenetic analysis workshop. Any questions? Or we'll take questions after the.
Are there any questions? We can just take the questions around that now. Online? All right. I think we can proceed, Alan.
Okay. So Alan can proceed.
Sorry, there's a question online.
Yes.
From Mercy. Mercy, you can go ahead and unmute yourself and ask the question. Maybe she wants to take up one of the slots. It's good to be quick.
Yeah, sure.
All right.
Hi. Thank you so much. My name is Mercy. I'm a PhD student at the Pan-African University. I'm just asking for the link where we can apply.
Okay, so for the application, I think I can share with you.
All right,
Thank
you
you.
can send an email to the East Africa BioGenome Program at gmail.com and we will forward your details to INCABA. So one slot gone, guys.
Okay.
So Alan, carry on.
All right, thank you. Good afternoon, everyone. I am Alan, as you have heard, from INCABA Biotech. I am delighted to be the one who will be taking you through the Sanger sequencing workshop at our lab in Riverside. And basically the title, Understanding Biodiversity Through Sanger Sequencing. You should be able to at least learn one or two things from Sanger sequencing that will help you through your projects, maybe as a master's student, as a PhD student. So for all the participants that have been shortlisted for that workshop, congratulations in advance. We hope to see you tomorrow. So okay, so just a brief introduction of the services that we offer at the lab. We are an one-stop shop as a lab. We offer all services from molecular biology, ranging from DNA extractions, so your DNA extractions up to PCRs, Sanger sequencing, up to NGS sequencing. So we are quite holistic all around. We offer those services, and if you will be coming tomorrow, and also if you will be coming tomorrow, so you'll be able to at least have a hands-on experience of all these services, right from DNA extraction. So why should you attend this workshop? Why is it important to you as a student or a researcher? So you should be able to learn the fundamentals of Sanger sequencing. We understand that Sanger sequencing is the oldest technology, right? Yet the very gold standard sequencing technology that exists, and it's very helpful in understanding the biodiversity that we have in the continent. And what you also be able to learn is your sample preparation, the best practices on handling your samples, right from how you collect your samples from the field up to transporting them, bringing them to the facility at the lab where we handle them. Third, you'll also have a hands-on practical experience. We will be able to give you that platform where you're able to work with the kit, the DNA extraction kit, the PCR kit itself, up to loading your samples on the instrument, the sequencer itself. And finally, you'll also be able to have a look and get an experience on the data analysis part, which is quite interesting. You should be able to, I mean, it's interesting for me. You should be able to find it interesting for yourself. So I really welcome you to come and have an experience in that workshop tomorrow. So this is just the lab setup that we have. We have several labs from the DNA extraction labs, post-extraction labs, then the sequencing labs. So you should be able to see all this tomorrow. And then this is just a highlight of how it was last year. It was quite interesting. We had 10 participants selected for that particular workshop. And I am quite aware that they were able to get something from the workshop itself. So, I mean, I can't exhaust how much interesting it will be tomorrow and how much exciting it will be. So really, really welcome to all the participants who will be joining us tomorrow at INCABA. And yes, so questions before I ask you my question. Any questions? None. Thank you so much. So I have a question that we can just think about. So the question goes, if Sanger sequencing can only realistically process one sample at a time, and Africa is home to an estimated 100,000 plus undescribed species, what does that mean for how we prioritize which species to get sequenced first and who gets to decide? That is my question to you. Thank you. So if you want to reach out to us, there's the email there at jumbo@incababiotech.africa, or if you have any queries about the lab services that we also offer, you should be able to reach us through laboratory.ibea@africabiotech. Thank you.
Thank you so much for that great presentation. And I think to answer your question, the scope of African BioGenome already answers that question, because to address that problem, we need to collaborate. We need to empower young scholars, young researchers with the skills that you're teaching them. And this is also where the DSI also comes in, because we have the skills, we have the raw materials, the resources, the biodiversity, but now where can we store our data? So I think by the time we are achieving the scope or the goals of AfricaBP, we will answer your question well. So thank you so much. And I highly encourage students to attend your practical sessions because it's basic phylogenetics, and you need the basic for you to go to advanced level. So thank you so much. Next, we are going to FNS Scientific. They will be carrying a practical session on breeding pipelines with the Illumina powered genotyping by sequencing. And also on top of that, I remember yesterday we didn't have their introductory presentation, so I believe they will also be doing both to introduce Illumina, the FNS Scientific, as well as their practical session. So thank you so much, and I think Dr. Anthiga will be the one taking us. So the floor is yours. Thank you.
Thank you, Paul. Good afternoon, everyone. Yeah, my name is Peter Ndhiga, as I've been introduced. I work for a company called FNS Scientific. I'll introduce the company briefly, and also I'll highlight the practical workshop that we are planning from tomorrow. So the theme of this practical workshop, we've named it Accelerating Your Breeding Pipelines with Illumina-powered genotyping by sequencing. So when we talk about accelerating breeding, one fundamental thing that we need is data, right? And we need to generate this sequence data from our crops, from our animals, from our population. And the key thing that when you talk about accelerating or rather sequencing this data is ensuring that we can't analyze what we don't have. That's why we need to start fundamentally with the data itself. And that's why highlighting the infrastructure that we have as a country is very important to understand the gaps as to why our African data is underrepresented. Okay? So to be able to generate this data, definitely we need a supportive ecosystem. And this is where FNS Scientific comes in. So FNS Scientific is a company that has been in operation for over 40 years. It is in Quartande, Nairobi, and it has a regional presence across East Africa, where it provides, or rather it serves different sectors from food to energy to water to industries. And we do all this by collaborating or by partnering with innovative global brands, Illumina being one of them. So what do we actually do on the ground? So behind every company is a team. We have a collecting team of engineers. We have a collecting team of application specialists like myself and Dr. Cecilia. We have a collective team of the logistics people who ensure that you get your supplies and reagents in time. And all these activities, whether it's sales and support, whether it's trainings, whether it is attending events and forums such as this one that we're attending today, we do all this with one major goal. That is to ensure that we provide, or rather we journey with you throughout from sample all the way to data analysis. So one of the key challenges that we have in Africa is we find that piecing together the whole aspect of generating data becomes a challenge, especially to a researcher. So this is what the company has done. It has pieced together all these solutions, and it enables you to formulate whether it's your proposal or whether it's your project in a way that will give you a whole snapshot of what you require to do that project. So essentially, we journey with you throughout, whether it's storing your samples, whether it's performing extractions. We ensure we provide you with all the technology to make that a success. Illumina is one of our core partners, and one of the privileges that Illumina provides us with is this massive portfolio of the sequencing platforms that they have. We have low throughput platforms, middle throughput platforms, all the way to high-end throughput platforms. So what this basically means is, depending on what your research goal is or what your scientific goal is, you are spoiled for choice. And there's an instrument, there's a platform for you to achieve your specific goal. I will highlight the MiSeq I-100, which is featured on our exhibition booth today. This is the latest, it's the 4th one from the right. So this is the newest instrument from Illumina. And what we say internally in our circles is that this instrument was designed specifically for Africa, because it has addressed so many pain points that we tend to have in Africa. Number one, the sequencing consumables that accompany this instrument, they come at room temperature. So what this means, you don't have to invest in a freezer or a fridge to store your sequencing consumables. And also it means if your DNA library is ready, you can sequence on demand without having to do your cartridge or having to wait a long time before you perform your run. The other thing is speed. So what happened after or during the COVID pandemic, we saw the need to generate data quickly to address situations, okay? So with this particular platform, you can get your data in as low as 4 hours and in as much as 15 hours. So we have really accelerated the time it takes for you to get your sequence data, which is very important. The other key advantage of this platform is portability. As you can see on our exhibition booth, this is a platform that can be translocated from one bench to another easily without needing any technical calibration, as opposed to our previous platform. So that is a very big plus, especially in Africa, where a platform might be required in a certain lab quickly and urgently. Finally, another key aspect that has been addressed with this instrument is data analysis is on board the instrument. That is very important, because at the end of the day, generating data is what is key and is what is key. Talking about data analysis, we have a comprehensive way in which we provide you our data analysis options. So we have layered them into key 4 options, if I may. We have what we call primary data analysis, we have secondary data analysis, tertiary data analysis, and all these are AI powered. So just to quickly highlight, when you talk about primary data analysis, this is the data that is generated by the instrument, that is from the images that are processed during base calling to your sequence reams. Beyond your sequence reams, when you are doing your alignments and reference mapping, we call that secondary analysis. So we have a whole portfolio of curated applications that you can choose from. Our base space app, which I highly encourage you to subscribe and check through, because some of the applications that are on that platform are free to use. Others are subscription based. We have another layer of analysis we call tertiary analysis. This is where you generate now your biological meaning out of the genotype data that you generate after your secondary analysis. And just to quickly highlight the different, you just mentioned the softwares that are powering all these analyses. When it comes to primary analysis, we have what we call real-time analysis software. Again, this is used to do your base calling processes and all that. We have secondary analysis layer where we are using a Dragon-powered server. So what this essentially means is, once you generate your FASTQ files, the raw format it does from the instrument, you can leverage this particular server to do your analysis very quickly. So it accelerates your processing capacity. Because Africa, we are limited in terms of resources and we can't use our laptops to analyze some of this data. So you can leverage on these platforms to do that. When you talk about the tertiary analysis layer, this is essentially important, where you want not to move, again, when you want to get those insightful information out of your data. We have what we call Illumina Connected Insights, Illumina Connected Multiomics. So we'll talk about this during our practical workshop and highlight how you can leverage most of these applications. just to mention, all these applications are powered by a platform we call BioInsight, Illumina BioInsight platform. So the reason why this is important is because we need to accelerate in this era of AI. We need to accelerate decision making in a fast manner, and also we need to work with real-time databases to get accurate data information or accurate information from our data. So this is the platform that we are leveraging that is coupled with AI to do this kind of analysis. Now, let me quickly highlight what we've been doing. So essentially, just to mention, take you back a little bit. So all these portfolio of Illumina sequencing platforms, interestingly enough, we have all these platforms in the country. We have an iSeq, we have a MiniSeq, we have a MySeq, we have NextSeq 550. Interestingly enough, we have all these platforms in the country. So one of the key things that we are noticing from the industry side of things, access to genomic technologies, especially if I may speak for Kenya, is not much of a challenge. The challenge that we are having is getting, or rather the capacity to utilize these instruments. So we have the instruments, we have also the data analysis solutions, and as I've highlighted, some are even free to use. So that's why we're having this practical workshop, so that we can provide this awareness. And we take this opportunity to thank AfricaBP to give us this platform to be able to highlight this and what can be leveraged. And it is in this regard why we are having this particular workshop tomorrow. So this particular workshop, essentially what we'll be doing, we'll be leveraging the platform that you've seen on the exhibition, the MySeq Hi-100, to perform targeted sequencing on maize. Okay, we are working, I think, with about 20 maize. Maize is quite big in terms of genome size. That's why we are not working with such a big data size. But essentially what the theme, or rather the focus, is to transition to high throughput sequencing. Day one will be a mix of fundamentals and also some hands-on. So we'll start with some background or context, why we want to do genotyping by sequencing on the Illumina, on the MySeq Hi-100 platform. And then we'll do QC, library QC, and proceed to do some partial library preparation tomorrow. We'll see how far we go. And then, so there'll be theory and hands-on. Second day, we'll perform library QC for the library that we have generated the previous day. We quantify it. So we use the normal fluorometric assay method. And also we'll do a fragment analysis to check whether we have achieved our fragment size threshold. And then we'll proceed to do sequencing. As you can see, it's really not as hectic as sequencing is perceived to be. So we plan to do this within 2 days. And of course, there'll be a mix of theory sessions to highlight what is happening, and also to explain the metrics that we'll be observing on the instrument as the data analysis is ongoing. So for this particular workshop, we'll be using a 300 cycle kit. So this means within 15 hours, we should have our data generated. And hence, on the third day, we will retrieve our data and walk you through how you can do analysis on that data, and how the data that we generate from the instrument can be leveraged for impactful decision making when it comes to breeding decisions. So it's such a privilege to the participants who will be attending tomorrow. So just to sum up, as I summarize, again, I repeat, I think Africa does not only need access to genomic technologies, but essentially Africa needs the capacity, and we are grateful for such forums such as these, where we are able to take our participants through these technologies to enable the adoption, and also to see how insightful the data we get out of this can be of impact moving forward. Thank you.
Any questions in the room or online? No questions online. One question in the room. All right.
Thank you, Dr. for such a nice presentation. Now, my question is on that machine where you are uploading the samples, does it have a limit of the samples you are uploading? And does the size of the samples uploaded affect the quality of the outcome of this analysis, I mean sequencing? And again, on the analysis, please, if you can expound, what kind of the analysis does that does? Thank you.
Thank you for that question. So we'll be using a platform we are calling BioSearch Caller to do the analysis. So for this particular workshop, we'll be more interested with the genotyping data that will be generated. There's no limit size because the amount of data that we generate, the amount of data you generate is dictated by the flow cell that you're using. So for this particular workflow, the way they have optimized it, the data that is generated, there's no limit in terms of the data that can be uploaded into that platform. Yeah.
Are there any slots remaining?
Very interesting question. Maybe Dr. Cecilia, you can highlight whether there are any slots remaining.
Wow, okay. So unfortunately for this year, we don't have any slots remaining. So maybe next year? Yeah? Will it be late? All right. So for the hands-on this year, we don't have some slots remaining. Maybe as FNS and Illumina, we will consider to partner with the AfricaBP at some point to do an in-between. But for now, unfortunately, it's full. Yeah.
Where is it held?
So this year, the practical will be held at the Kenya Agricultural Research and Livestock Organization, KALRO. And we are partnering with a team there that does a lot of breeding work for maize. You guys know Kenya, we are very good at breeding maize. You know, Katumani variety and all that, yeah? And they do a lot of work to create varieties that are resilient, either to drought or pests. And they do a lot of marker-assisted selection. And they're now looking to leverage on NGS to be able to do even deeper analysis of the varieties that they generate. But so we are working together to deliver this practical to the AfricaBP community. Peter, I see there's a question online for you from Muhammad. Okay. Maybe you can unmute yourself, Muhammad, and go ahead.
Thank you very much for this wonderful presentation. It's really a wonderful one. So my question are two. Number one, someone asks about the capacity of the material that will be uploaded on that particular machine. I didn't hear what he said. And the other question is, I'm asking about the meaning of bioinsight. What does that mean? And does it has any parameter to detect some sequential material on the machine?
Okay, thank you for that question. So let me start with the BioInsight platform from Illumina. So essentially, you can consider this as the engine that is running most of the analysis on the Illumina applications. So that is what I essentially meant by BioInsights. Now, in terms of this workshop, we'll be using an application called BioSearch Caller. So it is more of a collection of open source applications that we've bundled together to create this analysis suite. So it is more open source in a way. That's why there's not that much limitations in terms of data that can be uploaded into the platform.
Thank you very much. Thank you.
We have another question in the room.
Okay.
Yeah. Hi. I was asking if we have a criteria on the people that are being selected to go for these workshops, so that maybe next year we don't miss out.
I think that's more a question for the organizing committee. What was the criteria?
Yeah, so when we put out the call for the workshops, there is a questionnaire that you fill in. And in the questionnaire, you also are asked to state which practical. We give you the choice of choosing the practical that you would like to. And this is just for everybody. When you're selecting the practical, it is better that you have a reason why you have chosen that practical. Maybe you're doing some studies in that area. We also ask whether you have some experience. For instance, if you are very familiar with the Illumina platform and then you want to go to the Inkaba platform, we give you that ability because we would like you to have an experience with more than one of the technologies. So when we have all the applications, we just download that Excel for the people who have applied for a particular workshop and we send it to the company, because they are the ones who will be running the company or the university. They are the ones who will be running the practical, and therefore they do the selection. So for us, we know that the people who are selected are the ones who look like they'll benefit the most. So your application really matters. So this is just for you out there. Be deliberate. I saw somebody who would put, somebody has asked, which practical would you like? You say you want to do this one. Why? You leave the place blank. Then, that will not really help you, because like for each of these practicals, they have a capacity of a maximum of 15. Some of them, like Inkaba will tell us, for the lab, our lab is small, it fits 5 comfortably, but we can squeeze in 10, So if we have like for Inkaba, I think we had 169 applicants for 10 slots. So scanning through that application, it's the ones that stand out that will get an opportunity. And we are happy that we are not the ones who do it, because it was an overwhelming task, and the companies are the ones that do it. I hope that has explained to you. Thank you.
We have another question online. The question from Ngeno is the data that is uploaded on BioInsight secure?
It is secure. Yeah. So these are all, what do we call them? Like regulatory requirements that are there when it comes to using Illumina like data analysis applications. So something I always mention whenever I'm asked this question is Illumina is a New York Stock Exchange traded company, and if there is any report of data breach and stuff, that can really impact the share price and stock price of the company. So one of the things they highly make sure is confidentiality of the data. So the way they have set it up is when you log into it, we don't have visibility of what has been uploaded. So it's your own account to do your data analysis. So there's no whatever like access, like than party or indirect access to that data. So it's very secure.
All right, thank you. Over to you, Paul.
Thank you so much, Peter, for that wonderful presentation. Next, we are going to University of Copenhagen in collaboration with Pala Research Center in Laikipia here in Kenya. They'll be doing a practicals on hands-on wildlife conservation genomics. And the team will be led by Professor Rasmus Heller. I don't know if we have Rasmus online to give us to walk us through the outline of the project. Rasmus. And just to reiterate on what Prof has just said regarding the application of this workshop, you have to remember that we are not being charged to attend these workshops. So when it comes to screening of the participants, the tech companies, the universities, they are usually very strict. Of course, you've had this high volume of application and the slots are very limited. So your application indeed has to be very good and you have to be very convincing. And sometimes also you have to show that indeed what you're going to learn, you're going to apply in whatever you are doing. And if not, maybe even try to educate others on what you've just learned. So come next year when the calls will be announced and they will be opened, please do submit your application, but be very good with the application. Yes. Uganda. So Rasmus is not around at the moment. So let's go to the next practical session, which will be organized by the National Animal Genetic Resources Center and Data Bank, Entebbe, Uganda. And they'll be working on genomic prediction of adaptation to future environmental conditions. The person in charge of the workshop will be Dr. Joel Ogwang, and he'll be taking us through what he's planning. He has on the menu for his students in Uganda. Karibu, Joel.
Oh, thank you so much, Paul, for the introduction. Can I share my screen?
Yes, please.
Yeah. Yeah, please let me know if you can see clearly.
Yes, you can put it on presentation mode. Yes,
Yes.
thank you.
Thank
Carry
you.
on.
It's good. Carry on.
Okay, thank you. The practical workshop in Nagrik and Entebbe will be on genomic prediction of adaptation to future environmental conditions, and I will be using the program called Gradient Forest. So I am a PhD student at the University of Wyoming in the US, but I work as a molecular geneticist at the National Animal Genetic Resource Center and Data Bank in Entebbe. So just to outline the motivation behind this practical workshop, we are all aware that worldwide biodiversity is declining, and you can see how wildlife has declined over time. But the area of my interest, well, is aquatic resources, aquatic biodiversity, and you can already see that over time we have lost more than 100 freshwater fish species. And of course, the drivers of these are habitat loss, pollution, invasive species, and normal use like harvesting, and of course, climate change. Interestingly, worldwide, nearly a half of all freshwater species are data deficient, and that is a gap that a lot of countries around the world is trying to address. If you look at East Africa, for example, there is a lot of publication highlighting the opportunities that exist to kind of get information that is important for biodiversity conservation. So all these publications point to the need for scientific information to better make freshwater biodiversity conservation decisions. And we can ask the question, what is the future risk of extinction? And that's where machine learning comes in. You can ask the question, what's the extinction outlook today? So here you relate the population today, for example, allele frequency or genetic diversity, with environmental variables. You can talk of temperature, precipitation, or greenhouse gas emissions. But how will this picture be some 50 to 100 years from now? So what you'll need are the projected values of environmental variables, temperature, precipitation, greenhouse gas emissions, etc., and scale these with population level parameters like allele frequency or genetic diversity. So what we get is, is to what we see today, is it a match? Is the environment and the population parameters matching? And in 100 years, how does it look? Will there be a mismatch? So this is what this practical workshop is going to try to do. And I will be using a group of programs, which is based on the principle of random forest. Random forest is a machine learning approach based on decision trees to make a prediction. So it's based on a bunch of yes and no, responses, and a combination of all that gives us an average of what is the most appropriate decision. So this is useful for handling large, high-dimensional data, like genomic data and so forth. So the application is in genomics, proteomics, and even clinical predictions. And this random forest handles only one phenotype or environmental response at a time, and that is where the gradient forest comes in. Sorry, my screen. Yeah. So in principle, we will go through the normal process of data analysis, pre-processing of data that has been largely done, the data training, evaluating the model, tuning the model, and interpreting the results. And the data that we will need are genomic data, environmental variables, and some GPS data, mainly the shapefiles, which are useful for drawing maps. For this workshop, I'll be using publicly available data on an endangered migratory bird, that is the southwestern willow flycatcher. And we will be asking the question of how adapted will this bird be to future scenarios of climate change. Specifically, we will assess which environmental variables are important drivers of population structure, investigate how these variables are correlated with genetic variation, predict genomic vulnerability, or what we call genomic offset, and then visualize these results on the geographic range map of this bird. So at the end of the workshop, I expect learners to be able to install R and R Studio and all the packages we need for this workshop. Of course, this is more important to a basic user of R as opposed to people who have been using R for some time. And then for the rest of the learners, you should be able to read the data types that we'll be using and check their basic properties, and then identify which environmental variables are significantly associated with the genetic structure of the willow flycatcher. And then you should be able to define what genomic variability or genomic offset is, and then predict it using gradient forest and visualize these results on a map. And I want to acknowledge my sponsors. Specifically, I want to acknowledge the Wagner Lab at the University of Wyoming. The team at the lab were very useful in helping me to decide on which program that incorporates machine learning and which fits, very well with the theme of this year's workshops. Thank you very much. And if you have any questions, I can receive them now.
Thank you, Joel. Thank you for that wonderful presentation. Do you have any questions in person? Yeah, we have one.
Yes, we have one question on the floor.
Thank you for such a wonderful presentation. Now, my concern is, I don't know, is this model, have you used it or you are about to use it? And if you have used it, what is the level of the accuracy in terms of the prediction? please elaborate on that.
Thank you. Go ahead, Joel.
So there are very many programs for doing predictions. I've tried a few, including SLIM, which is also like a forward prediction model. But I felt this model is fairly, well used, and publicly available data for its implementation is available. I could have used my own data, the fish data, which is what I research on, but I could not access, the environmental variables that I needed accurately. So I decided on this. So much as I've not used it extensively, I have a very good idea of how it works, and I've run it a few times. So yeah, and I would say it is the best because it can handle large data sets which are multidimensional. You can talk about categorical variables or, continuous variables in large quantities.
Okay, thank you. And do we have any remaining slots open for your workshop, or is fully booked?
Well, the unique thing about the workshop this year is it will be virtual. So I think my Zoom can handle up to 300 participants. So there is still a lot of slots.
All right.
Yeah.
Good. People are happy. So maybe, you can share maybe a link, a form where we can share with participants and register. And yes, Prof.
I have it. I have the link. I have the secret code. I have everything. All of you must have received an email from eastafricaBiogenome.gmail, right? So if you're interested in attending this workshop, write to that email, and then this evening I will liaise with Joel and share his secret code with you. And then those of you who don't have that email, just see me and we'll be able to share. Yeah,
perfect. Thank you. And Joel, on a side note, I have a question, but not regarding to your practical session. I was in Uganda and I saw some of the Ankole cattle, with those big horns, so majestic, beautiful horns. And I wanted to ask, are there any genomic data on those cattle available?
Yes, there is some genomic data on Ankole cattle. I can say public access is limited, and the majority of the data is held securely through a few projects, one of which was coordinated by the Roslin Institute at the University of Edinburgh.
Hey, interesting. I think that's open another door for discussion, because now they are restricted, they are ours as Africans, but now the project was funded somewhere in abroad. But anyway, that's a topic for discussion next time. And then the last question.
Yes.
Would you happen to know if there is any correlation between the horns and the quality of meat and or milk produced by those cattle?
I cannot say there is a correlation between horns and, the productive traits. I don't have a good answer for that at the moment.
I think, Prof, that's a research idea, because you are an animal genetist. Maybe we can write a concept note on that and try to understand. Just out of curiosity. Yeah.
I would be very happy to collaborate on such. So let's keep that discussion going for sure.
So if anyone who has heard this, please don't use this as your concept note. It is mine. The good thing it's on YouTube, so it's recorded. I have evidence. Thank you, Joy.
All right. Thank you so much, and see everyone tomorrow.
Okay, see you. So next we are going to Addis Ababa University. in collaboration with Bio and Emerging Technology Institute in Ethiopia. So they will be taking us through their practical, which is Python-based bioinformatics data analysis, a hands-on approach. And the team will be led by Dr. Helen Negusi, Zevedu Edia, and Professor Kasahun Tesfaye. Do you have them online? Yes, please go ahead.
I'm here. Thank you.
You're
I would
welcome.
like to thank the organizer. I think I shared my slide. Did you share me with that, or shall I share it from here?
I mean, you can share it from your lap.
We are sharing?
Okay, we are sharing it.
Okay. First of all, I would like to thank the organizer committee to give us this opportunity, and the Addis Ababa University and Bio and Emerging Technology to support us to run this practical session at our campus, College of Natural and Computational Science. This is our fourth practical session. We already started with Africa BioGenome Project, East and Central Africa workshop when start their first project, and we are really happy to be here and able to manage the fourth practical session. Our this year practical session will be Python-based bioinformatics data analysis. It is a hands-on approach. Why this is, I will talk briefly on the syllabus of the current practical workshop, the overview of our practical session, the practical session organizers, and the training. Why really use this Python-based bioinformatics data analysis? Because as I told you, this is the fourth year practical session. We have used next generation sequencing using Linux for the first, and the second one, we also used RNA-seq analysis the last year. Today we will try to introduce a new approach because there are strong demand and interest from graduate students and early career scientists across public universities and research institute. So based on the interest, we will a bit divert to Python-based bioinformatics, because nowadays there is a rapid growth of genomics, transcriptomics, proteomics, and other biological data set. So this demand strong need for computational skills. So this Python-based a little bit is open source relatively compared to others, and it is easy to learn and widely used in bioinformatics. So we select this topic. It also facilitates automotive repetitive tasks. It integrates different bioinformatics tools. It helps to also analyze high throughput data set in omics data. It also helps to the development of customized the workflow. So that is why our choice for this is practical session is a Python based. Next. Syllabus, we have 3 days and we divided into three topics. The first day in tomorrow we'll use it for introduction to Python, the basic Python and biological data. We'll have some theoretical background and hands-on training, almost all the hands-on practical approach. In the second day, we'll try to cover the Python for sequence analysis, which is help to how the file handling system, the biological data format, some data analysis, DNA translation, some protein sequence, and so on. In the third day, all are practical interactive session. The trainee has opportunity to interact in the session. The third day will be a little bit advanced in visualization and data analysis. And this part also will try to cover how to visualize the data analysis and how to clean those data, how to visualize in different composition, in different format. So our trainees, for confirmed trainees 34, which is a little bit great number from compared to the previous year, and all are the representative across the country. And the interesting thing in this practical part, most of the participants from far area committed to cover all the costs to attend this practical session. This is really interesting and show us the commitment and the interest of this graduate student or early career scientists to improve their capacity in data and bioinformatics data analysis. One which is our of the overview of past practical session. We at our University in Betin is always collaborator with Africa Genome Project because both institute is a national node representative. Next. This was our first workshop, which was have been held in at Betin, and it was only on the next generation sequencing. We were around 20 plus participants in different public institute and research center. At the next, also the same in 2024. Again, in the Betin NGS data analysis from genome mapping to visualization, we had also interested trainers and have impact in graduate students. The third workshop have been held here in Natural and Computational Science at Addis Ababa University on RNA-seq analysis. In this workshop also there are representatives from public universities and research center. And this year, the same we host at the College of Natural and Computational Science at Addis Ababa University. We are really proud to host this workshop at our college. The college management is really supportive in collaboration with Bioemerging and Technology Institute. This is the organizer team, the local team of Ethiopia. Professor Kasaun, Dr. Zaudu Ida from Bio and Emerging Technology, myself representing our university. The trainees both from Addis Ababa University and Betin. Dr. Abi is Python specialist, Dr. Mesfin from computational data science program. Mr. Getachew and Mr. Addis also representing Python, their expertise in Python programming and bioinformatics analysis. I hope will contribute for the knowledge and the practicality of Python-based bioinformatics for our trainers. So this is, I hope we will have a very good enjoy, and I would like to acknowledge African BioGenome Project at Saba University and Betin for the supporting all the financial issue and hosting, facilitating all the facility which help to run this workshop at our college. So this is all I have, and we are excited to host all the participants tomorrow at our campus. Asante sana. Thank you. Amasagdalo galatoma wal mahasantai gidai. Thank you so much.
Thank you so much.
This
Thank
is
you.
all the representative of our language. We have 18 different languages in Ethiopia. Thank you so much.
Interesting. See, we are rich in language diversity as well. So that's
Yeah, ecology, so many
yeah.
diversity we have.
Exactly. Maybe we can try and come up with a phylogenetic tree of languages. Try to understand how
The
they evolve.
ecology we have Dalol that is 500 below sea level up to 4,500 above sea level. Imagine how we are diverse, even ecology, the language, nationality. We have so many amazing diversity in Ethiopia.
I can
We
imagine.
invite you to enjoy this diversity as well.
Yeah, that gradient diversity is massive. That's good. That's good. Do you have any slots remaining there? Maybe there are some people here who can fly to Addis in the evening and arrive there in the morning.
We'll try our best if they are really interested to join us within a day.
Okay, thank you. Are there any questions online, in person? Any questions?
There is one question
Okay,
in the room.
please go ahead.
Thank you for the nice presentation. Now, my question will be on proteomics analysis. We know that in a given pathway, upstream and downstream, maybe the expression of a given protein can vary in terms of the expression. Now, during the analysis, and you're trying to determine the functionality of this protein, how do you go about it? Please expound on that. Thank you.
Okay, the Python programming is just a programming language which has interaction like we did this expression analysis using R programming to identify which is up regulated, which gene is up regulated and which is down regulated. The same analysis can be done using the Python program. Only we can show the participant how to use this especially the BioPython, which is dedicated for bioinformatics, to show them how to use this language to generate those maps and those visualization. Unless otherwise the principle is the same, we can use it in RNA-seq analysis, but now we change the language because there are different opportunity. The participant will take the convenient programming language for their future analysis and future use. Thank you.
Thank you. Thank you, Doc. So, still in Ethiopia. Any other question before we proceed online? No? Okay. So, thank you so much, Doc.
Oh, sorry, there's one question
Yes,
online.
please.
Osman Ahmed, you can unmute yourself and go ahead and ask the question. Osman?
Hello?
Hi, we can
Yes.
hear
Thank you,
you.
Doctor.
Yeah.
Hello.
Yes, we can
Thank
hear
you for
you.
nice presentation, Dr. Helen. I would like to ask the question, where is the site for the workshop on the Addis Ababa University?
There is a participant communicated before. Are you the one of the participant in the practical workshop?
Yeah.
So we already
But
communicated via email if you missed in case. That is at Addis
okay.
Ababa University College of Natural and Computational Science at
Okay.
Arat Kilo campus. We
Okay.
already communicated via the Telegram. Did you join the Telegram group we have created for the participant?
Okay. We'll join it and we'll try to ask on the bureau, on the office.
Yeah, it is at Arat Kilo. If you're already there, you will get this information. We already communicated.
Okay. Thank you.
Okay. So, still in Ethiopia, we are going to Hawassa University, where we'll be having a practical titled DATSEC generated GBS data filtering and analysis. So the practical will be facilitated by Professor Hewan Demisi Degu. So do we have Professor Degu online? Okay, please go ahead. Thank you.
Okay, thank you very much. Is it possible that I can share a slide?
Yes, please. Yeah, do share.
Okay. Okay. Our practical aspect is huge data generated by genotype by sequencing using Illumina platform. And I just wanted to highlight what it means, how it is really different from whole genome sequencing and other aspect. So this is really a fast, simple library preparation and affordable and suitable specifically for many crops that we are using, specifically for molecular breeders. They can use it really effectively. And specifically, if you are really interested in breeding on drought or abiotic tolerance or or biotic tolerance or for yield improvement, and to understand the stress resilience location, the genotype by sequencing technology is one of the effective technology worldwide, and many people, many scientists all over the world are really using it. So after getting your, you might use the Illumina platform or send your sample for sequencing. Then after that, you'll get your data. Then how you are going to handle your data, how you are going to handle the missing data, and what type of restriction enzyme you have used it and what its purpose. And then you'll go for the bioinformatic requirement, like in the detailed analysis by filtering the complexity of your data. So the GBS is GBS is genotyping by sequencing. So it has a lot of application, but let me show you what it really means. First of all, for breeding, we need genetic marker. Long time ago, we used to have one or two marker in order to identify this cultivar or this plant from that plant or this race from that race, or from different type of races that we are using. And that is a genetic marker. It is heritable. That means you, from a female to the offsprings, or from parent to the offsprings, it will be inherited. So it is a heritable polymorphism that can be measured in one or more population or individuals. So it is the heart of modern technology. It enables the study of important questions in the population genetics, in ecological genetics, and also evolution. So it is the advent of next generation sequencing, because after the whole genome sequencing of Arabidopsis and also human genome, the sequencing of different plants and also animals is becoming the trend line nowadays. So you can resequence your plants or animal or your samples, and you can discover, and by sequencing, and you genotype thousands of your samples, depending on what is your population you have. So the sequencing is genome-wide. They are really high throughput because they touch from one chromosome to the number of chromosomes that you have. For example, for human being, we might have 23 pairs of chromosome. It can be really covering all the 23 chromosomes. So it is comprehensive, it is wide, and it is really used for associated studies for different organisms, since it is genome-wide, and you can use it for cultivated, also wild population. So the genotype by sequencing, I have already said, it is a high throughput method, discovering and genotyping. It combines the technique is there is a restriction enzyme. By digesting the DNA, by using this restriction enzyme, the next generations, and using next generation sequencing, like the Illumina platform, you generate the data. It is widely used in plant breeding and specifically in genetic researches. So when we talk about genetic restriction enzyme, let me show you with a picture like this. For example, let's say that you have two samples. So both of two samples, they might be different cultivars or different type of accessions or you might have collected from one place and another one from another place, then you really wanted to see what is their difference. So when you are using the restriction enzyme, and at the same time, when we are applying that restriction enzyme in the genome, it might cut it into different places or sizes, as you can see in this picture. So in the top sample one, there is a similarity between sample 2 at 150 base pair, at 350 base pair, or 700 base pair, but you can see that sample 1 has another restriction site at 1,300 base pair, but there is absent in sample 2. So this is what we are going to use as a, okay, why it is really different. So this is a whole genome approach I have already showed you, because all the other different whole genome sequencing, as you can see, they are really intense with different protocols and also steps. But at the last, as you can see here, the genotype by sequencing, it has 3 steps. So I'm not going to go through all the details, because that is the purpose of the sequencing or the sequencing platform, or the company that you are using. But we are more interested to work on after getting the data. After getting the data, what type of data you are getting, and then how you are going to shape it, to present it to any person, or for publication, or for the discovery aspect. So all that detailed information, definitely we might use reference sequence, or we might not use reference sequence. And then after we get the sequenced, or the genotype by sequenced data, we are going to use the R platform, by using different packages. Actually, we are working a lot with one company where they are generating this GBS data, and a number of our samples and breeding samples are sent to us through website exchange of the data after this sequencing, as you can see. So we follow that based on that sequence data, and we extract the SNP information. I'll go fast. So for example, here you have the whole genome, and then you use two type of, or different type of restriction enzyme. Then after that, you ligate that with different type of barcode, that is a kind of primer that you target and you sequence it, and this sequence information that you are going to use to generate the GBS. So it is high throughput, as you can see. Multiplex information is really there. It is intensive, so that's why the different colors, as you can see here, the different adapters, the different sequencing restriction enzymes. So they will give you from step 1 up to step 6, it gives you different level of information. After quality check, you get the clean data, and that clean data will be sent to you. So there are different type of restriction enzymes. I will not go through that, but know what matters in the GBS sequencing by using Illumina sequencing machine is the DNA quality and then DNA quantification. Definitely your sampling and everything has to be really good when you are sending for that sequencing purpose. And then if the DNA quality is bad, or if your sample is really bad, what will happen is that the result will not be exemplary, or will not explain your data. So it is better to be careful with your data, DNA quality and DNA quantification. So as you can see, after during the whole genome sequencing, your DNA has to have a very clear band with high molecular weight, ready for next generation sequencing platform with good quality. So then all these steps, as you can see, this is the bioinformatics part. This bioinformatics part, we might not work from the sequence file up to the other, but no, what we are really interested is after the SNP calling. After the SNP calling, then you'll get your data, then how that data is going to be analyzed. That is our purpose. So then what we'll do is we get your data in Excel form, and then that is like, it depends on the population numbers that you have. It might be 150, or it might be 300, or it might be 1,000 samples, but what you'll do is like, you are going to use different packets in order to give that SNP information a code, 1, 0, or 2. 0 means it is the same, 1 means it is heterozygous, 2 means like it's completely different. So then we give that a code, and we use the RStudio in order to filter all that information. We start from our first cube data and align it, and you'll get such kind of end product, very clean data like this. So all the production pipeline, it might be done with your vendor, the sequencing service that you are providing. And then after that, we get the filter data and imputate it. Might be all the data might not be functional, clean it, and then make it ready for data analysis, phylogenetic analysis, population structure analysis, also all the detailed GWAS or other QTL mapping analysis. And that is what we are going to learn from tomorrow up to Friday if you are coming in. So, for example, one of the data might look like this. This is a HapMap file or a data sequence file. So the HapMap file, it tells you about guanine, cytosine, thymine, or something by the name of the nucleotides. Then it tells you that which part is really different compared to the reference genome. For example, G might be the reference genome, but your sample might be adenine. That is the alternate. It might be like different. So compared to the reference genome, your sample might be different, and that is what we are doing with the population, the diversity analysis, and also the phylogenetic analysis. So we will equip you from the start of filtering your data up to the finish, which will be ready for your publication. So that is the purpose of this GBS based data analysis here in Hawassa University, which our work is a lot on this technology for the past 10 years. And thank you very much. Any question, you are really welcome.
So do you have any questions in-house, online? Questions? Okay, are there any slots? Are there any slots in your practical sessions? Maybe we can fly to Hawassa.
You are welcome. We have a slot. Don't worry. We have a wide laboratory, bioinformatics laboratory. So
that's good.
We'll host
Good.
you. Yeah.
All right. I'll think about that.
Thank you.
Any questions in-house, online? Maybe you can check if you have someone online. So the GBS can as well be used to estimate ploidy levels, Prof?
Pardon? Would you mind to repeat your question?
Can someone still use the GBS technique to estimate ploidy levels of different plants?
I haven't checked that yet, but I think like it's depending on the calling that we can trace it. But I haven't done that, and I am not confident to reply to that.
Yeah, that's good. Because one thing I've also realized is that right now with the technologies that we have, sequencing is not a problem in Africa. We have tech companies to do that for us. But what's also cropping out is the underutilization of the data that we are generating. Because one person might just generate a data to, do the genotyping, but the same data can as well be utilized, to understand diploidy levels of these crops that we are having. So I think that's also one of the things that we should put into consideration moving forward, that we generate data that can be multifunctional. It can be utilized in different other aspects. Otherwise, we generate this data, we upload them in this database website, and then years later, somebody is producing a very good paper with your data, but a novel idea of something that maybe you could have done as well with your own data. So I think for every scientist who is listening here, when you're producing this genomic data, try to utilize them as much as possible. Yes, do you have a question online?
Yes, we do have a question.
Just to clarify,
yes,
I just wanted to clarify this point. First of all, anyone who wanted to work in high throughput sequencing, first they have to understand the ploidy level of what the plants that they are working on. The taxonomic aspect is really important first. For example, most of the time we are working on our agricultural crops, maize or teff or barley or something like that. So these have the defined ploidy level, because that one you have to use a different flow cytometer in order to understand the chromosome and everything, whether it is diploid, tetraploid, and everything. So that information is really basics. But no, for us, GBS is only for diversity analysis. And also, for example, if I have a dwarf barley and a tall barley, I really wanted to understand what is really happening and what gene is really changing. I might breed them, and then rather than working on all the generation, 10 generation or 15 generation, I'll use their genotype sequence by genotype sequencing, and then I can develop a key-tail mapping with the offspring that we have, and then it can pinpoint for me where that location is really there. And also genotyping, GWAS, that is also another aspect that we are really using for GBS. This is more of a diversity analysis, whether there is diversity or something, but everyone has to be really strict about to understand the taxonomy and also the ploidy level of the plant or the animal that they are working on. Thank you.
That's true. Thank you. Thank you so much. Yes.
All right. We have a few questions from Kiplangat Ngeno. Maybe Kiplangat, you can unmute yourself and ask your questions. I think we have about 3. While you do that, Mengistu, are you also in a position to unmute yourself and ask your question?
Yes, yes.
Okay, let's start with who's that? Kiplangat?
Okay, shall I continue or?
Mengistu, please continue. We're losing you a little.
Dr. Yohan, I really appreciate your hard work and really thankful for especially politicians because of the ability to technology. And there are some startups from this plant breeding point of view and initiation. So, trying to this professionally, I'm an analytics and breeding. So, I need to ask one question. You are hearing me?
Sorry, we can't hear you clearly. Maybe you could type your question. I think we're getting a lot of feedback from your end. Sorry, we can't quite get you clearly.
Is that audible now?
Yes, that's better now. Go ahead.
Is that audible? Am I right?
Yes, please go ahead.
So, Dr. Yohan, thank you so much for your nice presentation, and it's really appreciable and nice work. Having said this, I do have one question. Professionally, I'm animal genetics and breeding. So, from that point of view,
I come across to ask one question, which is you are talking about or you have presented about the GBS, genotype by sequencing. So here, in terms of cost effectiveness as well as the reproducibility and genome coverage, which one is best if we are going to compare with the DDR sequencing and this genotype by sequencing for this in terms of animal genetics and breeding? If you have some experience, please share us. Thank you.
Okay, can I reply or?
Yes, please.
Okay, GBS, as I have said, it is genotyping by sequencing. It's RDD technology. It uses like two type of primer, definitely, but not the primer restriction enzyme. But we have to really careful what type of restriction enzymes that we are really selecting for the specific plants that we are using. For example, me, I am crop molecular breeder, or that's what I was using a lot. So if I use the same type of restriction enzyme, which is used for maize, for barley, it might not be working, because that restriction enzyme might be genome specific, and it might be working in one species, but we might not find that on another species. So there is a number of review literature on that aspect, the type of restriction enzymes that we are using. Since my background is not in animal genetics or breeding, but it's much better to consult those who are pioneer, who are really, if they have used the genotype by sequencing technology for breeding. So it's really good to use it for breeding, because it gives you a lot of information, sleep information. It's genome-wide, so it will give you a dense population structure, very clear population structure information, and then you can select your sample or your genotypes that you wanted to work on for further breeding. Okay, that is the advantage of genotype by sequencing. But restriction enzyme, I'll be really very careful to select based on the genotype information or the information of that plant or that specific animal, and literature review is really a shortcut for that. Thank you.
Thank you so much for that. We move on to the next and the last practical session that we'll be having. And this will be at the University of Kinshasa, DRC Congo. And the practical will be on machine learning with R for genomics and bioinformatics. And our facilitator will be Dr. Antoine Mafuila. So Antoine, please take us through your practical, how it will be, and any other information that the participants will need to know. Thank you so much.
Thank you. Can you hear me?
Yes, we can hear you loud and clear.
Thank you. I can share my screen. Let me share my screen. Okay. This year again, we are pleased to be part of the African BioGenome Project to organize practical session in the University of Kinshasa, affording the topic of machine learning, and we will use coding with R, helping students to be able to do machine learning and artificial intelligence using R. So the Laboratory of Molecular Biology in the University of Kinshasa is collaborating for more than 4 years with the African BioGenome Project, and to reinforce capacity of Central Africa scientists on bioinformatics and data science. So during the last session, we have four topics like NGS data analysis using Linux, Galaxy, and other coding programming language. But this year, particularly from tomorrow, we will conduct a practical session on machine learning with R for bioinformatics and genomics. So the objective of this training.
Dr. Antoine, please increase your volume or speak louder.
Okay.
Thank you.
Okay. So participants will practice machine learning, and they will learn how to do data preparation and feature selection, how to handle missing and incomplete data. We will also see with them how to do filtering feature by correlation threshold to remove multi-collinearity and redundant predictor. And they will also learn how to split data in training and testing partition. So for model training, you will go through the regression model, how they can train model using multiple linear regression, and also using classification algorithms like the decision tree. And also we will finish by showing them how to evaluate model using test train model on test data to verify the performance of the model across independent samples. So the content of the training, the practical session will be first the remind on basic R and advanced, how to install R, Studio, and R Markdown. We will go through different data structures, vectors, matrix data frame, how to do operation on data frame, mainly subsetting, because mostly machine learning will use those operations. We will also show them how to build function, the basic principle of building function, and to do looping with for or while, and also some looping function like apply, lapply, and so on. And the second part of the practical session that we will conduct the second day of the training, we will go through machine learning practice using linear regression, multiple linear regression, and decision tree model, reaching different steps, going from data processing, pre-processing to model evaluation. So for the candidate that has been shortlisted, we will organize this training in the Laboratory of Molecular Biology in the University of Kinshasa and through Zoom, so it will be hybrid. And 30 participants has been shortlisted. It will be in French because it's mainly for Francophone country. And if we have one or two people speaking English, we will also combine these two languages. So breakfast will be provided to participants that will attend in presential. Yeah, that's what all that we can say about this training now. Thank you.
Thank you so much, Dr. Antoine. Do you have any questions in person, online, regarding the practical session? Yes, please.
So thank you to all our afternoon speakers. My question is going to be just general, that if in future I want to use two platforms, maybe Python and R, but then for now I'm shortlisted to attend only for R, how is it possible? How can you advise me to also join Python? Because I think it's just unfortunate that all the programs have been scheduled to go for 3 days at the same day. So how are you going to advise me on how I can attend to both the Python and R? Thank you.
Good. That's a good question.
Yeah, it's also a tough question. It's a tough thing to answer because for this practical we had 1,000 applicants. For the workshop itself, we also had 1,000 applicants. For this particular practical, I think I saw a list of over 100. So if you've been selected for one and you're out of the 30 who have been selected, you're so privileged.
Yes, that's true. Yeah.
So this just shows you the demand. The demand is very, very high. We run these practicals every year. And so if you attend our meetings, in 5 years, you'll have learned 5 different skills. So we hope that this year you can do R, next year you can do Sanger sequencing, the next year after that you can do something else. So it's not easy, but we're trying. Yeah. Thank you.
And maybe even to just add on that, I remember when we started, there was a practical session on just basic laboratory techniques. And then from there was a practical session, I think by Dr. Antoine also, on just basic command line techniques. And then from there, we've had introductions to R practical session. We've had introductions to Python practical sessions. And then last year, we also did not only DNA extraction, but also sequencing. So you can imagine a student who has been with us for the past 4 years, having garnered all that skill. By the time we are closing the 5th year, this is a student who can even conduct his own or her own workshop. So these avenues are there. They are there for us, and we have to take advantage of those opportunities that we have. Today, actually, we have R running concurrently with Python. Next year, we'll have R running concurrently with Python, running concurrently with Rust programming language. So it's an avenue. You can just pick one and, make use of that skills that you'll get from there. Good. Any other question? Yes.
Thank you to all the presenters. So my question is not really an independent question, but in connection to what he said, I think he was asking of the possibility of running these practicals on separate days. This will give a student a chance to cover all the 5 practicals in, for example, 1 year, rather than having to take 5 years, like you've said. You've just said that in a span of 5 years, a student will be able to run his own workshop. Now, can we shorten it to a span of 1 year? Because if we can run this practical between this day and this day in January, then this day and this day in February, we run the other one. Yes. So this gives a student a chance to apply this one, fail, then apply for this other one, fail within the same year without having to wait. And I think that's what he was asking for.
Yeah, and that's true. And that's a very good question and a good way of seeing things. But also you will have to understand that also these practicals are dependent on the availability of the instructors. You know, like somebody like Anubab from India, he took this opportunity because it's summer holiday from where he is, just to come and do this workshop. Maybe during the year he's busy with academic work. Same way with Dr. Sami, same way with Dr. Antoine Mafuila. So it's something that we started organizing this workshop 10 months ago. And getting people to free this week just to hold this workshop, we really had to persuade them as well. And that's why, again, we reiterate that take these opportunities seriously, because you never know. Maybe this year we have R, next year we don't have R. This year we have Python, next year we don't have Python. This year we might have all of them, next year we might not have, because it all depends with the availability of these instructors that we have. So that's one of the things that you usually put into consideration as well. But that's a good suggestion. We will see if we can have different workshops across the year for different participants and instructors. Thank you. Another one?
I see question in the chat.
Yeah, thank you for the good present. A quick one.
Yes.
From the expert advisor for the new student who want to run the R. Now, which version of the R Studio will be advised for the installation for maybe tomorrow's practical?
Dr. Antoine, maybe you can address that.
I think our video and our set is updated regularly, and it's better to update your, if you are a data scientist, to update your R and your R Studio as well as you can. So for tomorrow, we don't need the last version of R or R Studio. I think it's the basic version. You can learn and practice what we will present tomorrow.
Thank you for that. So I don't think we have any other question.
I see some question in the chat.
Okay, there is one also in person. Maybe the one in person can ask, and then we go to the online.
Okay, thank you for that presentation. My question is, I know it's deep to learn R in one day for a person who hasn't worked with R before. Is it possible that you have, after you've done the practical, then you can have a follow-up assignment, like it's exercises which the student or participants can actually be doing in their free time and learning the skill, so that at the end of the day, it's not only a program of one day, but a continuous learning, so that we don't end up having it one day, and within some few days, you forget what you've done. Thank you.
Yeah,
thank you.
Yeah, that's exactly the problem we have, mainly when we are learning data science. But what we do, secondly, is to provide the resources as much as possible. Video, coding, script, and also learning material we share with the participants. I think we will create a small community with the participants so that we can share with them some issue that they face and concern that they can raise after the training. I understand that it's not in one day that you can be comfortable with R, but we provide the principle that the participants can use to build the future use of this programming language. Okay.
Thank you. Do we have those two? Maybe we can take two questions online and then we proceed. If
All
they are
right.
still there.
So we have a question from Kiplagat Ngeno. Let me read it. So, given the potentially large size of genomic data sets, will participants have sufficient computing resources to complete their exercises? If not, do you have alternative computational platforms that will be provided?
Sorry, I was a little bit disturbed. Could you repeat the question?
Right. So the question is around the computing resources.
Okay.
Given the potential of having large data sets for the genomics work, will the participants have sufficient computing resources to complete the practical exercises? And if not, do you have alternative platforms which you will provide?
Yes, it's a practical session, so we are able to use real data, or we can use also available data set. We try to make things very easy to use the data set that cannot be complicated when using normal computer, but when evaluating the model, when we have some problem, we can explain to participants that having more large data set, we can improve the model, the performance of the model to predict or to classify something. But we have possibility to use different type of data sets for making the practical session very easy.
All right.
Thank you.
Thank you. And then there's a question that I think this one is to the organizing committee. There are many experts across the continent with expertise in bioinformatics, genomics, AI, and statistics who could potentially contribute to similar training programs. So in cases where additional expertise or capacity is required to address the participants' concerns, are there ways to engage or collaborate with such experts to complement the existing trainings?
Yes, we're very, very much open for collaborations. Indeed, we are slowly building the collaborations on the continent. And please just respond to the email that sent you your invite. It will get to me, and then we will be able to plan for next year's practical sessions. So as you can see, there is a lot of demand for training in different areas of bioinformatics, wet lab, even taxonomy, even sample collection. And therefore, all the scientists who are out there, if you're willing, if you're able, if you can, please reach out and we will be able to plan more the next practical, and we'll be able to give more access to this great group of young scientists. And the practicals don't have to be based in Nairobi. You saw that we have some based in Kilifi, which is about 600 kilometers from here. So as long as you're in East or Central Africa, you could be in DRC, you could be in Rwanda, you could be in Uganda, you could be in Nairobi, you could be in Meru. We are ready to partner with you to bring that practical to the community that is around you.
Thank you. So I think, yeah, I would like to give thanks to the practical organizers for this year's practical workshops that we'll be having. And to all the participants of those practicals, I want to wish you all the best, and please do take advantage of such opportunity that you've been awarded. Also for me, I would like to give thanks to Africa BioGenome Project because this is the 4th year that we are running this workshop. And we started when I was second year, doing my second year of my PhD. And currently I've finished and I'm so happy for that. But I can't give my PhD journey story without mentioning AfricaBP because it has been instrumental, not only on the practicals that I attended with Anubab and Dr. Sami in Kilifi, but also the organization skills that I learned through the process, through the 4 workshops that we've had here in Kenya. And I'm grateful for you as the community and also to the practical organizers and all the tech companies that have been supportive in the journey that we started 4 years ago. So thank you so much, and I leave the stage to Professor Ann to continue. Thank you.
Thank you again. We can't reiterate that any young scientist who follows AfricaBP grows. We throw you in the deep end, you drink a bit of water, but finally you get to swim and you don't drown. And that is our goal, because we're not going to be here forever. We want you to be the leaders of the African BioGenome Project in the next generation. And we want to make sure that you are really, really good at it. Also, please note that we haven't charged you as a student anything. Anybody who's attending this practical, the only thing you'll have to pay for is your transport to the venue. But we will provide you with a meal at the beginning of the day. And I always tell students, don't be so poor. At least contribute something. Do you know how much it costs for, one of these practicals to run? If I give you the figures, you'll be shocked. Some of them run up to $10,000. That's about over 1 million shillings. Just for 10 of you to be able to spend 3 days doing hands-on practical. And this is something technology companies want to do, because you're the consumers of tomorrow, you're the lab scientists of tomorrow, you're the vice chancellor of tomorrow. So when you apply for this practical workshops, you also help us create demand, and then we're able to ask other people to support us in this journey. So thank you very much to the practical organizers who have gone through and even written out a curriculum for what you will do over the 3 days. We cannot thank you enough. And we thank all the technology companies that are sponsoring, providing reagents, providing kits for free. I remember some technology companies, I think it was last year, Inkaba was able to fly in people from South Africa at their own cost to train people here in Nairobi. And that just shows you the level of commitment that all our technology companies have, and we cannot take it for granted. So thank you very much. A round of applause to all our practical organizers and technology companies. So now we're entering our last and surprising session. We didn't put it on the program, so it can be a surprise. So this is our last session, and I can't believe we're at the end of the day, but I'd like all the participants who are in the house to stand up. Those of you who are online, you just wait for the surprise. Please stand up, everybody who's here. Okay, if you're from Mount Kenya University, you can take a seat. Those are our hosts, so I'm not going to hassle them too much. If you're from PAU, Pan-African University, come with your chair in front. If you're from Embu University, put up your hand. Who is from Embu University? Come with your chair. Somebody from Chuka University, please come with your chair. Somebody from Jomo Kenyatta University, come with your chair. I was going to ask, which university have I left out? But I was sure you'd not give me an answer. Technical University of Kenya, I know you're in the house. Please come to the front.
Meru University.
University of Nairobi. Okay. The rest of you, you're saved. Only because I've run out of space here in front. Yeah, please take your seat. And I can see Jomo Kenyatta there hiding. Please come. You cannot be spared. You are the anchor or institution for African BioGenome Project. I have an excellent team of young African scientists here in front of me. They have been with us for 2 days. And I want to ask the question. Who has the mic? You have the mic. Tell us who you are, where you're from. I can see you're learning in Kenya, but you're not a resident of Kenya. What have you learned, and what are you taking home with you?
Good afternoon, everyone. Good afternoon again.
Good afternoon.
We are the last day today. Okay, actually, I'm Ismail Salah, PhD student at Pan-African BioIversity University here in Kenya. I'm doing PhD in molecular biology and biotechnology. Actually, I wanted to say something to start about with it, that in this moment, I feel I'm so lucky to be here, to be part of this workshop as a participant. Because before I come here, I thought a lot. Go to this workshop or not? I have a lot of work as a lab, but the best thing that make me attend with you through those days, the first thing, the marketing of this workshop. Through all of last 2 months, we receive maybe every day a lot of emails. Every day morning, I go to my email to check the new emails. I found AfricaBP workshop, AfricaBP workshop, AfricaBP workshop. Every day. So after those 2 days of this workshop with genomics and phylogenetics and Africa genetic resources, and how we can to analyze the data, sequencing, data analysis, the industry partner, the workshop that will be through the next 3 days, all of that. Oh, how I thought before about the attendance. I'm so lucky to be here through this very valuable workshop, and I wanted to thank the organizing committee about everything to make this workshop as it with all of these benefits. And through all of this, I'm sorry for this long speak, but actually through those 2 days, I felt as everyone here, participant in this room or online, have a feeling, a very good feeling through Africa. Everyone want Africa to be better. Everyone want Africa to be as everyone here want the Africa to be. And I wanted to say Africa will go forward with its people. Really, everyone need to work to work a lot on. We need to work a lot in our themselves to be better. Actually, small note, just the last 30 seconds. A lot of people ask a lot of big question. I remember Prof. Lori today said I can't answer all of those questions because those very big questions. And I'm so happy to hear all of those questions from ourselves, from the people, from the researchers, from the Africans. The last thing I wanted to really appreciate Professor Anne. I don't remember. I saw her sit for continuous 2 minutes through those days. So let her show our appreciation, please. Thank you once again, and Africa will go forward with its people to be the Africa we want. Thank you.
So I'm happy to see that our emails were read after being ignored. They were finally read. But you see, it's a concerted effort. We have an organizing committee. I'll be recognizing them at the end. But you have the mic. What have you taken home? What have you learned? And what will you tell your people out there?
Hi, everyone. As you can look, I am the youngest in this. I'm actually an undergrad taking biochemistry and molecular bio from JKUAT. And what I have learned actually is, before even I start my research, I know that it's a very big no-no to take your samples out of Kenya. And during the lunch break, I had asked some researchers here, when you send your sequences out of Kenya, what do you get back? Do you get the whole data, or do you get just the sequences? And what happens with the other data that has been hidden? And yeah, I found out that we are losing a lot. We are actually contributing to them building powerful AI that actually they will come again. It's like we are giving ChatGPT data so that we can pay for the premium access, and the data is ours. So that one was, it is an eye opener for me. As I move forward with my project as well as my career, I know I have connected with a lot of people. I have made connections. I have learned a lot actually from the presentations that have been made here. And also that since I'll be attending the Inkaba Sanga sequencing workshop, I know I'll learn a lot. And now again, when I go back to JKUAT, since I'm a student leader, there's some other first years who are joining, so they learn too.
Excellent. And you're so privileged. I don't know what you wrote in your application form to get you as an undergraduate into in-person. You know, the algorithm is we have an algorithm that we run.
I can say I'm not your general undergraduate.
Yeah, we can see that. So pass the mic next, and let's hear from you. Tell us where you're from, which university you're studying in, and what are you going to take home? What is your greatest impact, or what do you want to tell the audience about the conversations that you have had, especially?
Well, thank you so much for this opportunity. I'm very appreciative for you and for the whole staff for a magnificent workshop and this 2 sessions for 2 days. And I hope tomorrow will be more practical sessions. My name is Jacob. The first name is kind of different, so let's start with Jacob. I think it's easy. I'm Egyptian. I'm doing my master's in molecular biology and biotechnology in BioST, Pan-African University, hosted by JKUAT, located in Georgia. I currently work on my genome editing, CRISPR-Cas9 in Hiroshima, Japan. Actually, I'm traveling to design my CRISPR-Cas9 using in silico bioinformatics data analysis in Japan. So I have left 2 topics here. One about malaria antigen to development kits, and another one about breast cancer using some plants, local plants like marijuana. So I leave them for now, and going to push myself to masters in CRISPR-Cas9 in Hiroshima. For do this, I have learned many things about bioinformatics and how we are as African. We reach this point in these technologies, and I hope so we can build our data analysis like DDGB, Japan, and NCBI like USA, and as well ENA like Europe. And also I have learned and connections, networking, so useful, so productive, valuable for me. And I hope we can from here can proceed and have connections. So collaboration, it is so much useful, can help us to have good work, good quality data analysis in bioinformatics specialty, because will be useful for upcoming. I'm looking forward to do HIV mRNA HIV vaccines for Kenya. So I hope can see some collaborations, so we can work on data analysis by generation and youth between 15 and 24 years old, male and female. So we can target the sequence and we can data analysis until we reach the main point for experimental here in Kenya. And I hope so we can have collaboration, so we can work on this project together with people from researchers and scientists from Kenya as well. Thank you so much.
Yeah, thank you. Great dreams, great aspirations. You're very young. You still have your PhD to go. So yes, come and let's collaborate. And we're looking forward to those products, the mRNA vaccine. And we're also looking at some of your kits, testing kits. And we just wish you well. And thank you for reading the email and coming. Next, tell us who you are, where you're from, what has been most impactful, and what are you taking away with you?
Good afternoon. My name is Mariam Diaye. I'm from Mali, West Africa. Currently, I'm a PhD student at Pan-African University in molecular biology and biotechnology. Actually, I feel very lucky because even when I was applying for this workshop, I was back home and I was not able to candidate because of maybe the area. So I had to maybe mandate someone, a countrymate here in Kenya, to apply for me. So to be here is I feel very lucky. And what I learned so far is that we are generating data from Africa, but the data is not being as much as possible used for Africa. And yeah, what I'm taking home, I'll also challenge myself before coming to Kenya that I will have a hands-on practicals on sequencing. I had like theoretical knowledge, but I need the practical knowledge. And this is a very good start for me. So thank you.
Good evening, everyone. Okay, I'm Ramos Mulinge. I'm an MSc student in genetics from University of Embu. And I'm currently working on my research project on about soil microbiome diversity at Isipe. I think this is my first time here. I've been attending the workshop but online, and it's a privilege to be here physically for this workshop. And I think from the previous discussion that were there yesterday and today about genomics and biodiversity genomics, it's really been helpful to me because it really resonates well with the work which I am doing. And another thing I took from this workshop is that I think in Africa we have all the resources to do the work which we are doing in our research, and at this point we don't have to, the sequence, the samples that we have, we don't have to take them to ship them abroad for maybe sequencing. We have all the resources here. Actually, for my work, because I've already done the sequencing was done abroad, and I was really impressed to know the fact that we have all the resources in Africa that we need to do on the sequencing. And I think this is an opener for all of us who are working with such kind of samples. We can do the sequencing here in Africa without having to ship them there. And I think that's something we need to focus on. And I think I have learned a lot on genomics and how I can use AI to integrate it in the work that I am doing. And I think this we all need to learn from this. And I appreciate the African BioGenome Project for these workshops. I think they are good for us as young scientists as we aim at in future we are the ones who now we are going to replace prof when they retire. And I think we are learning a lot on how, when that time comes, how are we going to also lead others who are coming, who are just maybe undergraduates, and also the ones who are in high schools. At the one point, they will be at where we are. And at that point, we'll be a step ahead of them and we'll be able to guide them. And I think I really appreciate this opportunity. Thank you.
Thank you. Next. Check if your mic is on.
Hello, I am Jepkoech Masito. I am an MSc student at JKUAT, and I feel so lucky and privileged to have attended this workshop in person. Last year, I managed to attend online, but this year, when I saw the link to apply, I applied very fast, and I thought I should attend it physically this time so that I can also connect to the scientists in person. And I am really grateful for this opportunity because I have learned a lot. I've learned that scientists, we should also connect, as a scientist, I should connect the indigenous knowledge with the genomics. Also, I should not just think about starting from the top. It is a journey. And also something else that has stood out for me, I think most of the presenters has made it clear that it is very possible because, we are doing these things in our continent. I remember sometimes when we are writing the proposals, in the methodology, you include some equipments that you have to do outside Africa, like my fellow scientists have said. And then that can be very discouraging because you see it as a very complex thing, bigger than you, but when it is made like available closer to you, it feels very reachable and attainable. So I am so grateful for this opportunity, and I also request, like you said, Professor Anne, that as African BP, you take up students and you train them to make sure they can swim by themselves and become leaders tomorrow. I am also available as a student if there is an opportunity to join you. I like this opportunity, to use this opportunity to say that I am available for your mentorship so that I know I have the ability to become a leader tomorrow, and I believe in myself. And thank you for making me know that this is possible and I can attain it. Thank you.
Good evening, everyone. My name is Lorino Nyango from University of Nairobi, taking PhD in pharmacology and toxicology. So the first thing that was so interesting to me was about coming up with biobank. Ideally, the research I'm working on is on aflatoxin, and my main aim is really to come up with the unknown biomarkers that I can eventually use them to get at least a kit that can be used for biosurveillance purposes. So you see, in such case, the biobank will be really essential to me as a person and also as a researcher, because I would love to use them and also do other things apart from just getting the kit for biosurveillance. Number two is the discussion on biodiversity. When I was doing my MSc in molecular biology and bioinformatics at JKUAT, I really find interesting prokaryotes in some of the known forests in this country. But then we don't have the gene bank for African. So I was forced to, post them in NCBI. As much as my paper is out there, is really doing well on Nature, but somebody else will use those data to do something interesting, no? But that one could be the advantage for us as Africans. So that is also very important. Then I've really connected a lot within these 2 days. I've developed a protocol and I was getting some challenges. So I reached out to Professor Josephat over there. So we exchanged ideas and I was seeing some leeways, on how to go about them. Because I was forced to ship the plates that I really needed for the protocol all the way from Italy. And it has taken me two weeks. So it means as a scientist, I've wasted 2 weeks, no? Yeah, which is not very interesting. Number 3, we have discussed on how to help young people on how we can retain the brains that we have as African here. But really, I still really feels like it is easier said than done. Why? There is still a big gap on the career position in this country. So me as a young person, if I get the opportunity right there, most of the time we don't want to come back. Because where am I coming back to zero when I've climbed some ladder over there? So that is still a gap. Yeah, but thank you so much. This is my second time. Professor Kuja Dr. Kuja is your student, but Dr. Kuja is my supervisor.
Mm-hmm.
Yeah. Thank you.
So I'm your academic grandmother.
Yes, sure.
Yes. So I have a question before I come to you. I'm going to ask you. Yesterday we spent the day talking about AI. Are you using AI? And you're all students. How has it transformed your academic journey? And I'll start with you.
Okay. Actually, in my PhD project, I have 4 objectives, different objectives. The last one is about to use AI to analyze the data that generated from the previous 3 objectives. So it's necessary to learn through those 2 days and the practical session through the next 3 days about the AI and how we can to use to analyze our data. I want to add a small thing about the Africa Bank of Genome and Gene and Genome. I feel that dream may will happen one day. You believe about that? Yes. Yeah, and may this AfricaBP workshop will be a part of this dream. Thank you once again for the opportunity to be here in this workshop. Thank you for all.
Who else is using AI? Please, pass a mic.
Hello again. Yeah, I use AI a lot. I actually have 2 premium accounts.
Tell, is it Claude or is it Chat?
It's ChatGPT Plus and Gemini Pro. Why? Because for Gemini, it accesses Google Direct. So it has, you can have reliable information. And the Pro version gives you ability to extend. You don't have a limit of when you're using your Pro. For ChatGPT, you all know ChatGPT, right? Now, how do I use it? I believe we are in the era of tech. And we, as different as we are from the people from 19th century or 20th century, huko nyuma, they had to have this big library of books reading. But right now, you can just have an idea, you put it in ChatGPT, you get all that information filtered out for you. Even actually, ChatGPT is good such that it even gives you links, direct links to to the papers. So for me, I feel like I use it a lot when reading. Now, when it comes to AfricaBP and what we have been talking about here, I feel like we as Africans it's time for us to have our own AI, our own ChatGPT. Maybe you can call it AfricaBP AI. Our own way of doing things. And it will help us a lot in when we want to do research. At some point I was imagining what would happen if we were having a data center as big as the one ChatGPT has. How good will we be? How many things will we do? Even when you look in history back, you see that most of these we came from apes. Most of those bones are found in Africa. So it means that we have a lot of natural diversity here. We have a lot of data here, but due to, and I wouldn't call it in a bad way, I'll say we lacked the knowledge, we lacked the access to technology, and that allowed us to actually fall behind and actually give access. Right now, you're all here. You know why you should not send your samples. You know that if you want to do whole genome sequencing, there are platforms here in Kenya. And actually, we are blessed that Kenya is actually known as the capital city of Africa. So it means we have everything we want. So as Kenyans who are here, please, and you have attended this, please, we need those samples, we need those sequences, we need to do crazy research. We want to do things that will put us in front. And so that is what I'll say.
Thank you. Who else is using AI in their academics, apart from typing in your exam questions?
Well, for my case, I'm using AI, but for data analysis and also for CRISPR-Cas9 to design the primers and the Sanger. So I'm using tools bioinformatics AI. Even when I'm doing my proposal, I was doing Conscious AI to looking for the literature review gaps so I can develop my own work. So it just give me the idea or give me the fast time so I can finish early and if can develop my work so fast. Also, I using visual code to analyze the data. I was working on bacteria genomics, so I have data, so I try to finalize and try to filter the FASTA-Q, and the end these tools just give you the development your work, but your work it is still your work. So you whatever you're doing, you just use this tool like something can make your work just have good quality, nothing more. Yeah, thank you.
So let's give them all a round of applause as they take their seats. To all the young scientists who are online, it's only because you are not here physically, but I'm sure you also had an opportunity to say in your heart what this workshop has done for you. What I can tell all the young scientists is that presentation of your results is key. You have to be good communicators and you have to be ready to communicate at any time. So this was spontaneous. But as a young scientist, if an opportunity presents itself to you to stand up and say something about yourself, stand up and say something about your work, don't let it pass. And this opportunity today, I picked you out, but next time, if somebody were to ask you, perhaps you can volunteer yourself, because you might say something that will open a door or a window or an opportunity for you, and it's a way of learning. So thank you to all the scientists that I put in front of here. It was a hot seat, but you were very, very relaxed. And I think you're doing well in your academic journey, and we do wish you well. So ladies and gentlemen, we have come to the end of the African BioGenome Project. I can't, workshop for this last 2 days, the 4th East and Central Africa Regional Workshop. It's been on 2 intense days. Yesterday, we spent the whole day talking about AI and its impact in education, in research. And today, we spent the day talking about DSI, access and benefit sharing. We talked about samples. We talked about biobanking. And really, it has been not only enlightening, but it has brought out that we need to continue having these discussions. And I do hope that when these discussions are being held, you who have participated in this workshop, wherever you are, you'll be sitting at that table and you'll give a view, because that view will have been a view that has come through a consensus that we have all agreed that we should do certain things, like avoid sending samples out, like do local sequencing wherever you are, like the importance of building infrastructure in our national, local institutions, and that will carry the day. So as we come to the end of this workshop, it's really a great pleasure for us to host. Yes, I see a hand over there. You want to say something? A question? Okay. We can hear the question.
So my apologies for interrupting you, but I had a burning question, and yes, I wanted to ask you specifically, and this is my question. As Kenya, is there anything that has been done or that can be done at the level of the government to come up with a policy to favor the work scientists are doing, and also maybe to spread awareness among those people so that they can understand the importance of our work and how it can help in the development of the country in various areas such as biomanufacturing and therapeutics, and also come up with, yes, those diagnostics tools. Yeah, thank you.
Yeah, indeed, the conversation with the policy makers is really important. If we remain in this room and online and we just speak as scientists, we speak the same language, we need to invite the other stakeholders, the common people, the business people, the politicians, the ministers in charge of these dockets, because then they will hear the opinions from our end. It's one of the things that we're doing as African BioGenome Project, but wherever you are, you have access to some of these offices, including your Vice Chancellor, including your Dean. Those are some of the stakeholders who have opportunities to take this conversation forward. And it's really, really important that the correct views are collected and put in front of the table. And we meeting and forming consensus about certain topics ensures that the views that are taken forward are not personal. I might have a personal opinion about something, but when I hear majority are saying that we should not take samples outside, then I will be shy to push my opinion, but I'll be very confident that if I push this other opinion, it will be the opinion that is approved and registered by the majority. So thank you very much for that. It is something that is important, and we definitely will do that. And just to also mention that AfricaBP, in writing policy papers, we publish in order that we can reach out to some of these stakeholders. And we hope that they will be able to read, if not the full paper, but also the short briefs that we do write. So I would like to invite the local organizing committee here present so that we can recognize them. Dr. Kujak Josiah had a work trip outside Nairobi. So I'm the chair of the local organizing committee. We have Dr. Kujak Josiah, who is a member of the partnership committee of the African BioGenome Project. We have Dr. Paul Kagame, who was Mr. Paul when we started this journey of workshops. And this year he got his PhD. And we're very happy about that. He's a member of the local organizing committee. And we also have Dr. Cecilia Rumberia, who is a member of our local organizing committee. So this committee of 4 is the one that put this show together. It took us 10 months. It will take us another maybe 1 month to wrap it up so that you can all get your certificates. And then after that, we'll say, we put a lid, take a break in December, and then start again for next year. So give them a round of applause for the work that they did. So thank you very much. Yeah, now I give you leave for all your Saturdays, and because we meet every Saturday to review progress. I'd like to thank the co-chairs of the African BioGenome Project, the Professor Boabid of North Africa, Northern Africa Node, who's also the chairperson of the African BioGenome Project, Professor Julian Osuji of University of Port Harcourt, who's the chair of the West Africa Node, and Professor Craig Keener of South African Medical Research Council, who's the chair of the South African Regional Node. I'd also like to thank Dr. Thangod Ebenezer, who has kept us on our toes and made sure that this workshop has come together. Thank you very much, Dr. Thangod. And last but never the least, I'd like to thank the all the node participants in Congo, in Rwanda, in Ethiopia. All of you have come together to make this workshop a success. We cannot leave without recognizing the contribution of Mount Kenya University, who have given us the venue, who have provided us with refreshments throughout the last 2 days, and who also opened up their lab for a practical workshop. Let's give a round of applause to Mount Kenya University for being an excellent host for the second year running. And we thank you very much, and we thank the Vice Chancellor and for his leadership. We thank Professor Jesse and his lab and all the participants, members of that lab, for the technical support that they have provided for us who are here. Lastly, never the least, to the in-person attendees and also to the online attendees. We give you a round of applause for being with us, consistently and persistently for the last 2 days. So thank you very much. It's been real, it's been wonderful, and I look forward to seeing you all in the second week of August of 2027, where we will have our 5th East and Central Africa Regional Workshop. Thank you, and goodbye from Nairobi.